Let me introduce my name is Dr. Agus Susanto Kosasi, Clinical Pathologist Consultant, as President of Indonesian Cytometry Association. Her Honorable Dr. Chan Sian Hua, Clinical Pathologist, as President of Indonesian Association of Clinical Chemistry. And also Her Honorable Professor Dr. An Agung Wiradewi Lestari, Clinical Pathologist Consultant, as Chairperson Organizing Committee.
Ladies and gentlemen, we would like to inform the schedule of the event today as follows. First is the Indonesian national anthem, and then followed by opening prayer. Fourth agenda is welcoming address. Fifth part of today's event, which is the lecture session, and followed by question and answer session. Next agenda is product presentation by Beckman Coulter, and we will continue today's event with parallel workshop.
Ladies and gentlemen, for a brief moment, we would like to invite you to stand up and sing the Indonesian national anthem, Indonesia Raya. Ladies and gentlemen, please have a seat. Before we start the main session, let us pray together according to your belief. And the prayer begin. Amen.
Ladies and gentlemen, let us now move on to the next agenda. For the welcoming address is from Dr. Agus Susanto Kosasi, Clinical Pathologist Consultant, to Dr. Agus Susanto Kosasi, Clinical Pathologist Consultant. You have our full attention.
Very good morning. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi, salam sejahtera bagi kita semua. Shalom, Om Swastiastu, Namo Buddhaya, salam kebajikan. To Honorable Dr. Zozia Masyorowski, the Chair of International Society for Advancement Cytometry, and for Honorable Professor Aryati, the Chairwoman of Indonesian Association of Clinical Pathology and the Laboratory Medicine Specialist, and for Honorable Dr. Chan Shan Hua, the Chairman of Indonesian Association of Clinical Chemistry, and the Honorable the Professor Wiradewi Lestari as Chairwoman of the Organizing Committee, and Honorable all the speaker, Paul Hutchinson, Paul Wallace, Dr. Paris Jane, and David Waterman, and all my beloved colleague and participant of ISAC Bali Workshop 2023. First of all, I would like to thank you for everyone who's being here with us today. And I would want to welcoming to all the speaker, welcome to Bali. Is very, very pleased to be able to become to our meeting today, ISAC Bali Workshop 2023. And I would like to express my sincerest appreciation for the organizing committee, especially for Dr. Wiradewi Lestari and the Life Education Task Force ISAC, Professor Paul Hutchinson, who generously helped us to make this event come together, become successful. And for the information that the first ISAC in Indonesia is in 2015 is in our hospital in Darmais. It is Dr. Kristan is chair of the ISACs. Dr. Zozia is coming, and then in 2016, and then 2017 we attend the Cyto Asia, and then the third ISAC is in 2018, and then 2019 we have a local workshop, and then 2020 is we know all this pandemic. We have stopped the ISAC workshop in Indonesia. So we hope this is a good start for us to continue again today to have ISAC workshop. Recent year that the Flowcytometry in Indonesia is growing rapidly. On the massive progress of Flowcytometry is using maybe the one of the policy of the government to establish a cancer center in the region for efforting the equalized cancer health service in Indonesia. So the government supporting development both item of equipment, procurement, and human resources. And now we have 30 Flowcytometry consists of Beckman Coulter 7 and 23 Beckman Dickinson. So our joint task force for development of human resource related to Flowcytometry to produce to standardize Flowcytometry service in Indonesia. Therefore, in the future, ISAC and ICHA, Indonesian Cytometry Association, and will conduct more similar training, maybe in area diagnosis, stem cell, CTC, transplantation, CAR T-cell, and research. But last not least, I would like to say gratitude to Beckman Coulter and also Beckman Dickinson who support us in this today's event, and would like to thank you for all the committee for especially in Bali. I cannot say one by one, especially to Professor Wiradewi and Dr. Siani and the team. And also in Jakarta, Dr. Liana Dr. Wiwit to arrange this event. Maybe I don't want to take much of your time, so please enjoy this workshop. Shall exit this workshop with abundance of knowledge, since I have an excellent lecture in two days. Assalamualaikum warahmatullahi wabarakatuh. Om Santi Santi. Selamat pagi semua. Selamat workshop. Terima kasih.
This is the speech from Dr. Agus Susanto Kosasih, Clinical Pathologist Consultant. Thank you for your speech.
Now, we would like to invite Dr. Chan Xianhua, Clinical Pathologist. To Dr. Chan Xianhua, Clinical Pathologist, you have our full attention.
Thank you. To our distinguished guest, President of the International Society of Advancement in Cytometry, Dr. Sofia Marciorowski, to President of Indonesian Society for Clinical Pathology and Laboratory Medicine, Professor Dr. Aryati, Specialist Pathology Clinic Consultant, President of Indonesian Cytometry Association, Dr. Agus Kosasih, Specialist Pathology Clinic Consultant Mars, ISAC Bali Organizing Committee Chairwoman, Professor Dr. Vira Dewi Lestari, Specialist Pathology Clinic Consultant, and all the honorable speakers and participants, good morning and warm welcome to Bali. It is an honor for Indonesian Association for Clinical Chemistry to collaborate in this two-day seminar and workshop of ISAC Bali on flow cytometry. Flow cytometry methods has been long used in routine laboratory, but with the advancement of technology, flow cytometry use grows in very clinical condition. And this comprehensive topic in this seminar, from the basic clinical use and research use of flow cytometry, I believe will give a broad coverage of the application itself. Congratulations and great appreciation for Prof. Vira Dewi and his team, and also to the corporate sponsors for arranging this event with very interesting and comprehensive topics. Also, sincere appreciation for all the speakers and lecturers for sharing their expertise and knowledge for the advances of flow cytometry use in Indonesia. Hopefully, Bali Ambience will create good environment for exchange of knowledge and also fruitful discussion. And I congratulate all for this seminar. Thank you.
Thank you, Dr. Chan Xianhua, Clinical Pathologist, for your speech.
Now, we would like to invite Professor Dr. Aryati, Clinical Pathologist Consultant. To Professor Dr. Aryati, Clinical Pathologist Consultant, you have our full attention.
Okay, thank you. Assalamualaikum warahmatullahi wabarakatuh. Om Swastiastu, Shalom, Namo Buddhaya, greetings ladies and gentlemen. Good morning everyone. It is my pleasure to welcome you all to the ISAC Bali Symposium and Workshop on Flow Cytometry. I would also like to welcome to Dr. Sofia Marciorowski as the President of International Society for Advancement of Cytometry, Dr. Agus Kosasih, Clinical Pathologist Consultant as the President of Indonesian Cytometry Association, Dr. Chan Xianhua, Clinical Pathologist as the President of Indonesian Association for Clinical Chemistry. And also I would also like to express my sincere gratitude to our Chairwoman, Professor Dr. Ana Agung Dewi Lestari, Clinical Pathologist Consultant as the Chairwoman Organizing Committee, and also other esteemed speakers for this event, Dr. Paris Jain, Dr. Paul A. Hutchinson, PhD, Professor David Westerman, and Professor Paul Welles, PhD, also all members committee and sponsors who have supported this event. This event, symposium and workshop is made possible through the collaboration of several organizations, including the Indonesian Cytometry Association, ICA, the International Society for Advancement of Cytometry, ISAC, the Indonesian Association of Clinical Pathology, Laboratory Medicine, ISAC, and the Indonesian Association of Clinical Chemistry. As we know, leukemia is a prevalent malignancy that accounts for a significant number of cancer-related deaths. The diagnosis, classification, and identification of the underlying genetic cause of leukemia are critical in its management. The WHO guideline on the diagnosis and classification of hematolymphoid malignancies emphasizes that the diagnosis and classification of leukemia must be based on morphology, immunophenotype, cytogenetics, and molecular tests. Immunophenotyping is essential in the diagnosis and classification of leukemia, and in recent years, the awareness of its utility has increased in Indonesia. However, the capacity for testing with the increased cytometric installation based in many hospitals throughout Indonesia needs to be increased, making it important to enhance knowledge and competence in flow cytometry in the diagnosis of hematologic malignancies. The objectives of this today's symposium and workshop are to improve the skill and competence of flow cytometry users in Indonesia, facilitate opportunities for collaboration, and exchange of ideas with international and national experts in flow cytometry, and enhance awareness of the importance of flow cytometry in clinical and research settings in Indonesia. After going through the COVID period for about 2 years, we were finally able to hold an offline symposium dry and wet workshops, and making it accessible to a wide range of attendees. The target audience includes flow cytometry users in Indonesia, clinical pathologists, also from the pediatrician, and also internal medicine doctors, and also medical technologists with an interest in flow cytometry, hematologists, and pediatricians. I hope that this workshop will help in building the capacity of healthcare professionals in Indonesia and will serve as an informative and engaging platform for attendees to learn and collaborate. I thank all of you for your participation and being a part of this symposium. Thank you. Om Santi 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 Om. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Professor Dr. Aryati, Clinical Pathologist Consultant, for your speech.
And the last, we would like to invite Dr. Zofia Masharovsky, PhD. To Dr. Zofia Masharovsky, you have full attention.
Do you need this step to get up here? Good morning, everybody. I just wanted to say what a pleasure it is to be here in Bali. I can't believe we're so lucky. I think you have one of the most beautiful places in the world here in Indonesia and Bali. So we all feel very, very privileged to be able to come and teach here. I'd like to thank the organizers, Dr. Dewi, Dr. Liana, Dr. Wiwid, who've been so helpful with all of this, and all of the organizers here in Bali. It's just been great so far. Pleasure to be here by the sea. I'm not president of ISAC. I'm chair of the Live Education Delivery Task Force, which organizes these workshops. They originally started in 2002 by Dr. Krishan in India, and then expanded to Turkey and to other places. And as Dr. Agus was saying earlier, we've done 2 in Jakarta, we've done 1 in Kuala Lumpur, Paul Hutchison, who's in Singapore, is crucial in helping organize all of these. So we're grateful to Paul. It makes our lives easier, and he's definitely one of the main drives in this area for the workshops. It's a pleasure to have Paul and Paresh here as well. I'm part of the gang that travels around and does this. And so I'd like to welcome you all to this as well, and thank the sponsors, Beckman Coulter and BD, because we couldn't do these without sponsorship. It just would be impossible, and we're very much reliant and grateful to our sponsors for this. Don't hesitate to ask us any questions, and our emails will be up there if you think of questions afterwards. If we don't know the answer, we know who to send them to. So please contact us and come up to us during the workshop and ask us anything you like. We're happy to be able to help in any way you can. Thank you.
Thank you, Dr. Sosya Mashirovsky, PhD, for your speech. I'm sorry. To officially open the event, we would like to invite Dr. Sosya, Dr. Paresh Jain, Prof. Paul Wallace, Dr. Paul Hutchinson, and also Prof. Wiradewi Lestari to hit the gong as a symbol of opening ISAC Bali on Flowcytometry. Let us give applause for the opening ceremony of ISAC Bali on Flowcytometry.
For all of the honorable speakers and Professor Wiradewi Lestari, please kindly stay on the stage for a moment so we can take a picture. Ladies and gentlemen, now we come to the main session. The lecture will be delivered by Dr. Sosya Mashirovsky, PhD. Dr. Sosya Mashirovsky, PhD, worked in many labs and countries on a variety of different subjects. In the 80s, specialized in solid tumor preparation for multicolor and cell cycle analysis. For 28 years, responsible for the Flowcytometry core facility at the Curie Institute in Paris, France. Co-chair of the Life Education Task Force of the International Society for Advancement of Cytometry. To Dr. Sosya Mashirovsky, PhD, you have our full attention.
So good morning again. I'm going to talk here about the very basics of flow cytometry. I think all of you, how many of you are clinicians? I think most people are clinicians. Can you put your hands up? I see a few hands. How many people work on a flow cytometer right now? So we got quite a few hands. That's good. So I'm going to talk about the basics. I think the basics of how the cytometer work is very important because I think that you need to know what's inside your cytometer in order to know what can go wrong with it and how it can affect the results that you're getting off your instrument. So I've got a laser pointer, but I'm not going forward. Okay, next slide, please. Back one. So what is flowcytometry? I think you probably all know, but I'll just go through this quickly. It's a technology which allows us to measure light scatter and fluorescence intensity on cells or other particles. We use beads, we use micro EVs that are beginning to be important as well. And the cells are one by one in suspension. Next slide. The advantages are speed. This goes very quickly. You can measure data on millions of cells at a time, which gives us good significant statistical significance. Multi-parameter, we can measure very many parameters at the same time. At this point, we can go up to 40 with some of the new full spectrum instruments. And we can sort the cells as well. Next slide, please. So when do we use a flow cytometer? Next again, because I've got a... So we use a cytometer when we want to know if we've got big cells, little cells, whether the big cells are green and pink or red or orange or yellow and red. And so we can subdivide the cells into populations based on their size, their scatter, and the different markers that we put on them. Next slide, please. The applications are huge in both research and clinical. Research in cellular biology, phenotyping, immunology, basic cytometry, basic cellular functions as well. I won't go through all of them. And the clinical, of course, which is what you're most interested in. And over the next couple of days, Paul and Paresh and Dave Westerman will go into detail on a lot of these different topics. Next slide, please. So in a flow cytometer, what do we do? I'm going to talk about first light, what we measure, both fluorescence and light scatter, and how a conventional flow cytometer works. I'll talk about the fluidics, the optics, the electronics, and actually on this one, I don't have time to do the cell sorting. Next slide, please. The light that we use in flow cytometry, we use a specific range of wavelengths. It's expanding now. We're going down to the low 300s, up above 800 now. This is the visible light spectrum. Next, please. And if you remember your basics of physics from high school or undergraduate, next, please. The short wavelengths have very high energy. Next, please. And the longer, next, please. Wavelengths have lower energy. So remember this, lower wavelengths, the shorter wavelengths are high energy, and the longer wavelengths are low energy. Next, please. So we're going to talk about fluorescence and fluorochromes. Next, please. What are fluorochromes? They're molecules which can absorb light at one wavelength, and then they can re-emit this light energy at a longer wavelengths. The structures are generally aromatic rings. Next, please. And two of the standard fluorochromes that we see in flow cytometry that you're probably very familiar with are fluorescein or Phytse, and these are the structures for phycoerythrin and or known as PE as well. Next slide, please. So how does this work? We have excitation. This is a schema of a very basic schema of a molecule of fluorochrome. We have an electron at a resting energy level that's capable of absorbing the light. It jumps up to a higher energy level, which is unstable. it loses some of that energy, and then it re-emits the rest of the energy that it absorbs as fluorescent at a higher, and remember, lower energy wavelength. And so this is a very simplified schema. It's really the fluorochrome's electron cloud that absorbs and emits the energy. But this is basically what's going on. And this is an example of a FITS molecule that can absorb light at 488 and re-emit in the green around 530. Slide, please. Each fluorochrome has a specific excitation spectrum and emission spectrum. The excitation spectrum is the range of wavelengths over which a fluorochrome can absorb light. And so each fluorochrome can absorb light across its range of excitation, but it has a maximum, oops, oh, it worked. It has a maximum excitation. This one is FITSI around 495. So this is working now. Great. Similarly, each fluorochrome has a specific emission spectrum, and it can emit over a range of wavelengths, but it will emit at a maximum here. So for example, FITS will emit all across this emission spectrum, but most efficiently at 520. But as you can see here, it's also going to emit out into the yellow and into the red a little bit. And this is going to be important later when Paresh talks about spillover and spread. The laser light that are lasers that are commonly used on the cytometers that you'll have in your lab, we've got a 488 laser. This is a blue laser, a red laser at 633, and generally we have a violet laser around 407. The newer instruments also tend to have a yellow-green laser around 561, and also a UV laser around 351. And we'll talk about that a little bit later as well. So how do we use the fluorochromes? Very often we have an antibody against a specific marker that's coupled to a fluorochrome. So for example, here we've got antigen A, antibody A against it, coupled to FITS, and then we've got PE that's coupled to an antibody against B. We're also using often in, particularly in research, if the gene has been successfully incorporated into the cell, it will be green because it's also expressing the reporter fluorescent protein, and then we can sort the cells. Sometimes the transfection rates are only 1%, and we want to do experiments on purely transfected cells that we know carry the gene. So we can sort those cells out and give the researcher back a pure population of cells that are expressing the gene that they're interested in. And then there's a lot of other fluorochromes that are specific for different parts of the cell. We've got DNA specific, we've got pH specific, organelle, calcium flux, and a whole range of other fluorochromes that are available. We can also measure light scatter with a flow cytometer. And the light scatter that we measure is a physical property of the cell or the particle, and it refracts or scatters the light when it passes the laser beam. And so the laser beam, say I'm a cell here going through in a fluidics stream past the laser is coming this way. I scatter light in all directions, but we measure the light in two directions. We measure the light over here in the same axis as the laser, and that's our forward scatter. We also collect the light behind me at 90 degrees through a lens. That's called the side scatter. All of the fluorescences are gathered back here as well. And so what does this light scatter tell us? The forward scatter is roughly proportional to the cell surface properties and the size. It's roughly proportional because it's just scatter. A small cell will scatter less light than a large cell. The side scatter is more affected by the internal complexity of the cell and the granularity. And so neither of these can actually be used to quantitate size, but they're still very, very useful for distinguishing different populations of cells. For example, here we see, we can see that we've got a population from peripheral blood. We've got lymphocytes, we've got monocytes, we've got granulocytes, with the forward scatter on the x-axis and the side scatter on the y-axis. And while we can't say that this is a 7 micron or a 10 micron cell. We do know that they're separated out, and we can identify those populations and then look at their parameters separately. So a flow cytometer is not a black box. There's a lot going on inside it. And we're going to talk here about the different aspects of what happens inside the flow cytometer so that you understand how it's going to affect your data. So the fluidics in a flow cytometer position the cells so that they flow one by one past the laser beam. The optics then separate the light emission from the different fluorochromes and direct them towards the appropriate detectors, the light detectors. And the electronics allow the conversion of the light that the detectors see into voltage pulses, which are then digitalized to a channel number, which is related to the intensity of the fluorescence of that fluorochrome on the cell. And so we're going to first of all talk about the fluidics, positioning the cells so that they flow one by one past the laser beam. So this is a very, very rough schema of a flow cytometer. It's a positive air pressure system, and many of the flow cytometers use this positive air pressure system. We have a sheath fluid tank. We've got a waste tank. We have our sample down here. We've got our sample injection system and our sheath injection system, which leads up to the flow cell. And the flow cell is where the cells will pass through the lasers one by one. The sheath fluid can be a saline solution, it can be PBS, it can be water. In a cell sorter, it has to have salts in it, but in many of the flow cytometers today, it's also what can be water. We connect the sheath tank to the sheath chamber, so the sheath is being pumped in here, and we also connect the waste to the output. We have a system of air pumps that pressurize, so the air pump will pressurize the sheath tank so that it's pushing the liquid out here and into the flow cell. This is a fixed pressure, so you don't change this. And it's very carefully regulated because it has to be very stable. We pressurize the sample port as well. So this tube is pressurized. And so that means that the cells will exit through this tube that pushes it up into the sheath fluid, into the center of the sheath fluid, and it's directed through the flow cell where it will pass one by one. And also these slides will be available to anybody. I think that they can put them up on the website. If anybody wants the slides, you're welcome to have them. We'll make them available. And the sample pressure is also carefully regulated, but you are the person that's regulating the sample pressure. So this varies, and it will depend on how fast you want your cells to go through. It's very simplified. Most of the commercial systems have complex pressure regulation mechanisms which carefully control the sheath and the sample injection. And this is what it really looks like. So this one over here has the laser turned on. This is what we saw in the schema there. So we've got our sample that's coming in here. It's being injected into the center. This is the flow cell. And so the laser is coming across here. And here you can see that the laser is illuminating the cells as they're going one by one through. The forward scatter is over here. And behind this, in the Z-axis, there's a lens that's collecting the side scatter and all of the fluorescence to divide it. So there are different ways. We've just talked about this positive air pressure. LSR2 for Tesla Caliber Symphony, all of the BD machines generally use this. The Galios and the sorters also have a positive air pressure. Some of the newer instruments have syringe pumps that push through with a syringe, either the sheath or the sample or both. And other ones also have a peristaltic pump that push it through. So the Accuray, Cytoflex, ZE5 will push through with a peristaltic pump. The sample stream crosses the laser at different points, depending on whether it's an analyzer or a sorter. And so on an analyzer, the cells will be coming from underneath and they'll be injected into the flow cell, into the middle, and the laser will intersect the cell within the flow cell. The flow cells are quartz. They've got very good optical properties, and the lens that collects the light behind it is coupled with an optical gel. So this system is very sensitive. In a stream in a jet and air sorter, there's a nozzle which makes a stream in air. And so the light has to go across the air before it hits the collecting lens for the light and the fluorescence. And this means that for some of the fluorochromes, you might have a log difference in sensitivity. So if you can see it on your analyzer, you may not necessarily see a good separation on your sorter. And so the stream within a stream, the hydrodynamic focusing, is very important. The cells are injected into the center of the sheath fluid, so they'll be positioned in the center of the laser. So this is your sheath fluid coming down, and your sheath fluid is pressuring the cells so that they go one by one through the center of the laser. And we'll see in a minute why that's important. And so if we take a look at it, they're a little bit everywhere. And then as they hit the sheath fluid that's doing the hydrodynamic focusing, then they're in a single cell stream. When you change the sample pressure on your instrument, say you're going from low to medium to high, this can affect your results. If you have your sample pressure running low, as here, the pressure is low enough so that the center core that the cells are going through is very narrow. And that means that the cells are right in the middle of the core and right in the middle of the laser beam. And this is going to affect your results. We'll see why in a minute when we look at the laser beam. But when you're doing something like DNA cell cycle analysis, and you're looking at very tight peaks and you're looking for near diploid or aneuploid populations, it's very important that you run your cells under low pressure so that the excitation is very uniform and the emission is very uniform so that you get very, very tight coefficients of variations on these peaks going through. If you put high sample pressure on, you're pushing it through a lot harder. You put it up to medium or high. It's a very wide core. And so the cells are not going through faster, but there are more of them going through because the core is wider. And you can see that the cells are not necessarily right in the middle. And so the excitation is not as uniform as you'll get over here. And so you'll get something that looks like the same cells going through running on low will have a very narrow coefficient of variation. Whereas if you're running them on a high, you'll have quite a wide coefficient of variation. And when you're doing antibody phenotyping or something like that, this is perfectly acceptable. You can run them through on a high. But if you're doing something like DNA cell cycle analysis, you need to be careful with this kind of thing so that you're always running them on low. The other thing that can happen is that if your instrument is clean and there's no bubbles, your cells will go through the center of the laser. If you have clogs or dirt or air bubbles, those can push the cells to the side. And if the cells are on the side of the laser, they're not being maximally excited, and that means they're not at maximal emission. And so all of a sudden, you may see that your peaks that was going through here on the histogram goes over to the left because you've lost sensitivity. It might also disappear. And so it's very important, we'll talk about this in the lab this afternoon, that you quality control your instrument to make sure it's functioning properly, that your fluidics are functioning properly, and that your cells are really going through the middle of the laser. So we've talked about the fluidics. Our cells are going right through the middle of the laser with the hydrodynamic focusing. Now we're going to talk about the optics a little bit. There are two kinds of optics we're going to talk about: the excitation optics, with the lasers exciting. So it's the lasers, the lenses, and the prisms that focus the beams on the stream. And then, sorry. So down here, we have our excitation optics. We've got a blue laser, a red laser. These are being focused so that they're arriving at different points on the stream, but focused on the stream at the point where the cell will pass through them. So we use lasers in flow cytometry. In the old days, in the 90s, when most of us started here with flow cytometry, they were great, big, huge water-cooled lasers that took up half a room. At a single wavelength, they're very stable light, and they focus to a tight spot on a tiny area, like a sample stream. They're getting much smaller and much cheaper. And there's a whole generation of solid state lasers now that are available pretty much in any color you might want. That said, there are certain wavelengths that are standard on the cytometers that we classically use. And so if we look at this again, the blue laser, which is a 488 nanometer wavelength, is used to excite FITs, PE, GFP, Alexa 488. The red laser, standardly used, and that's a 633 red laser, APC, Alexa 700, APC Psi7, the APC tandems. We have a violet laser usually at 405 that's used for all the brilliant violet dyes, for Pacific Blue, for V500. DAPI is also excited with a violet laser. The yellow-green lasers, and we'll talk about a little bit more later, usually a 561, are better for exciting the PE tandems, PE, PC5, PC7, the red fluorescent proteins. And we also have the UV lasers and near UV lasers that are 350 violets. They excite the calcium dyes like Indo and Herxt and DAPI as well. And so you can see here that these lenses and prisms direct and they focus the lasers on the cell as they pass through. So here we've got a series of lasers that are being directed. Here's our green laser, we've got our red laser, we've got a violet laser, and you can see that they're being bounced through these prisms so that they all arrive in the same axis here, and that they hit the stream at slightly different points, but all focused on the cells. And we'll talk a little bit about how they're focused so that the cells pass through one and then another and then another. We'll talk about that in a minute. So the laser beam geometry is important. The standard laser beam is brighter, has its highest intensity in the center of the laser beam. And this is what it looks like. And generally on the instruments, there's a beam expander that gives us a little bit more leeway about the intensity of the center of the laser beam. But you can see that it's important that the cell passes through the center of the laser beam because that's where the highest intensity excitation will be. And if they're uniformly and highly excited, then the emission will be more uniform and high. And so they should pass right through the center of the laser beam. If they don't, a decreased fluorochrome on that cell. It means that your cell's not passing through the center of the laser. So the fluidics and optics here are very important. And as I said earlier, dirt or bubbles can deflect the cells so that they might be passing over here or passing over here. And so your peak will be lower. You might even have two peaks if you've got some fluidics problems. So your cell's passing through the middle of the laser. The lasers are focused on the stream where the cell is passing through. We're going to talk about the collection optics here. The lenses, mirrors, and filters that separate the wavelengths and direct them to the detectors. And so first of all, this light is collected through a lens. As I said earlier, the forward scatter is collected over in this, the laser beam is coming this way, the forward scatter is over here. The side scatter and all the fluorescence is collected through that lens that would be at 90 degrees behind me. So as I said, the stream and air earlier, here is the lens for the stream and air. The fluorescence has to traverse the air before it gets here. But the flow cell, the lens is coupled directly to the flow cell. So after the collection by the lens, the emitted light then passes through a series of optical mirrors and filters. These separate out the different wavelengths from the different fluorochromes that you're measuring on the cell and direct them towards the correct detectors. And we'll look at how we do this. So we use mainly three different types of optical filters. We use a long pass filter. So a long pass filter, an LP for long pass 500, this defines what wavelengths it lets through. A long pass will let through all of the wavelengths longer or higher than 500. So everything above 500 goes through. And you can see down here the wavelengths. Below 500, there's no transmission. Above 500, it's going through. A short pass filter, SP 500, does the opposite. It lets through all of the wavelengths below 500. So here you can see everything below 500 is transmitted and above 500 it's not. And the third filter, the bandpass filter, lets through a specific range of wavelengths. So this is defined by the numbers behind the bandpass. So bandpass would be a BP 530-30. This is written on the side of each of the optical filters. And so it's going to let through a specific range of wavelengths. The center of the window it lets through is the 530. And this after the slash, this refers to the window of light it lets through. So it's going to let through half above and half below. So it's 530 plus 15 and minus 15. So it's going to let through the wavelengths that are 515 to 545. So over here, you can see this is what the bandpass is letting through. And this will target the wavelengths that correspond to the maximum emission of the fluorochrome that you're interested in measuring. There's a lot of bandpasses available. You can order them. And there's usually one targeted to whatever it is you want to measure. Chromes. We can use these same filters as dichroics. So we can direct the wavelengths that we don't let through to collect them elsewhere. And so the LP filter here, LP 500, that's going to let through everything above 500 as it's supposed to. It's going to, if it's at a 45-degree angle like here, or depending on the instrument, the angles may be different. It's going to reflect all of the wavelengths it's not letting through in another direction where you can collect these for other detectors. You need to know the emission spectra of your fluorochromes. And so here we've got the emission spectra of FITS, the emission spectra of PE, and these are the bandpass filters that generally are targeted for this. So the 530-30 will collect most of the emission light for FITSI, and this 585-42 is nicely targeted to collect PE. Remember that we're looking at FITSI again. Look at how it's tailing off over here. So we'll talk about this a little bit later. Paresh will talk about this, but some of the emission from FITSI is at the same wavelengths that are being collected for PE. So just keep this in mind for the next talk and the end of this talk. So we've got a two-color detection system here, a very simple schematic. We have our laser, our cells going through the laser. We're collecting the light here at 90 degrees. First of all, it's going to see a short past 560. So it's going to let through everything that's under 560, and it's reflecting everything over 560 towards this detector that we want to measure PE. And PE, this has a band pass in front of it that's targeted towards the emission maximum of PE. So this detector, which is actually blind, the detectors are colorblind. They only measure photons of light. So we have to make sure that the photons of light that they're seeing are the photons of light that correspond to our fluorochrome. And so here, this detector is only seeing this range of wavelengths, and those range of wavelengths correspond to our PE fluorescence. Up here, the same thing. This is the detector for FITSI. We have a bandpass in front of it that's targeted towards FITSI. And if we look at a slightly more complicated schema here, we've added in the forward and side scatter. So here we've got, in the axis of the laser, we've got a detector for forward scatter. And remember, scatter is the same, it's not fluorescence. So the scatter is going to be the same wavelength as the laser. So it's 488 in this case. So we've got a bandpass in front of it that targets 488 light. So this is only seeing 488 light. At 90 degrees, we're pulling off, it's seeing along past 510. Everything above 510 goes here. Everything below 510 goes over to the side scatter detector, which also has a 488 filter in front of it. So everything above 510, then it hits a 560. Everything above 560 goes up this way. And below 560, we have our FITSI detector with the bandpass targeted for FITS in front of it, etc. So here, this is along past 610. Everything above 610 is going up here. Everything below 610 is going here. So this is looking at 560 to 610, and it's further refined by the bandpass for PE, a 585-42.
Sorry,
This filter
Doctor.
up here is along.
Just a reminding that your time is about 5 minutes left.
5 minutes?
Thank you.
Okay, I'll hurry through the rest here. So let's just skip through here. Some of the other instruments have slightly different variations on the configuration. This is a linear array, so it's bouncing back with short pass. And the BD instruments have octagons where the light is coming from the flow cell through a fiber optic. It's seeing a 735 long pass. It's bouncing everything below 35 here. And in front of each of these detectors, there's a band pass. So far, we've been looking at a single laser. But remember, I said we have up to 4 or 5 lasers on the instrument. Let's look what happens when we have 2 lasers. So when there's 2 lasers, we have separation in time and space. The lasers are focused to slightly different points vertically on the stream. And so the cell will come down. First of all, it will go through the 488 laser. The fluorochromes that are excitable by 488 will be excited and emit. That light will be collected at that point, and then it continues on to the 633 laser, where those fluorochromes like APC, Alexa 700 will be excited, and the red emission will then be collected. There's a time delay between the first and second lasers. In this case, it's 10 microseconds. And so the instrument will then correlate the light coming from the first laser and the second laser, third laser, fourth laser, so that it's all collected for one cell. This is an area, so you can see it's going through a series of lasers, and behind each of these, there's an optical filter, there's an optical fiber that is collecting the light and directing it towards the appropriate detectors. So I'm going to skip through this, and we'll talk about photons of light. They transform them into electrical current. So the types of photo detectors we use are photodiodes for the forward scatter quite often. And these new avalanche photodiodes, which are on the Coulter instrument called the Cytoflex, which are very sensitive in the reds, or photomultiplier tubes, PMTs, which are used weak light signals. They're very sensitive. So side scatter and all the fluorescence parameters are often used with PMTs. And so a photomultiplier tube has a cathode here. This is where the photons arrive, the photons that correspond to the wavelengths that you're interested in measuring here. So the bandpass will be in front of this. It will see the 530-30 range of wavelengths hitting the cathode. Electrons are emitted for the photons that hit the cathode. And then there's a dynode chain, a multiplication system that multiplies these electrons. So there's a lot of electrons that reach the output anode for each of the electrons that leave the cathode. So you've got a multiplication system here that is multiplying the sensitivity of your PMT. So you've got more signal, also multiplying the noise coming off your PMT. But you can get more sensitivity by changing the gain on this particular multiplication system. And so you get an output current here that's a voltage pulse that corresponds to the cell entering the laser at a maximum of the laser, and then the cell leaves the laser, so there's no more fluorescence. So for every cell that goes through the laser, for each detector, you have a voltage pulse that's generated. And this, you can measure certain things on this voltage pulse. The height is the maximum fluorescence when the cell was in the middle of the laser. The area, which is the total fluorescence as the cell went through the laser and left it, and the width, which will give you more information on how long the cell stayed in the laser. And so these generally are the measurements that we have for each cell, for each detector. And so a small voltage pulse will be digitalized to a lower channel number than a big voltage pulse over here. So in a perfect world, if your cells are exactly the same, you would have all of the cells in one channel if they don't have much fluorescence. The ones that had a lot of fluorescence, a lot of fluorochromes would be over here. But in general, you have a Gaussian curve that's around it that has a certain amount of error around the measurement. So the voltage pulse size is related to the fluorescence intensity and the PMT multiplication that you put. When you set your PMT settings, and we'll talk about this afternoon and how to determine what the best voltage settings are for the multiplication on your detectors. If you have very little, you'll have a small voltage pulse. If you have a lot of fluorochromes, you'll have a big voltage pulse, but it also depends on the multiplication system you put on your detector. And so we'll talk about that. And so the pulse size is recorded as a channel number. So the data is saved as a FCS file, which records all the values for each cell from each detector. So forward scatter, side scatter, all of the different fluorescence. And for each cell, you have all of this data that you can look at as a simple parameter, frequency, how many cells have this amount of fluorescence, how many cells have that amount of fluorescence, or you can look at them against different other parameters to subdivide the populations. I'm going to skip through all this as I'm running out of time. So now we know. So now we can look at gating on these cells. We can look at the small cells, lymphocytes. We can look to see whether they're red or green, and red and green. A single parameter histogram red here, you have a low population and a high population. It looks like there are only two. But if you look at it against another fluorochrome, which is the green fluorochrome, you can see that this high population has some that are negative for green and some that are positive for both green and red. And so you can look at these subpopulations. I have 2 more minutes to explain. Just 2 more minutes. Is that okay? Changing the PMT voltage. So when you change on your instrument, your PMT voltage, let's say we're measuring the same cell as it goes through, but we're looking at 3 different voltages. And so if we run this cell through at 300, the peak will be over here. The same cell at 400 volts will be in the middle. And if you run it at 500 volts, it's over here. So it's the same amount of scatter or fluorescence. It just depends on how you make your settings. And each cytometer has to have a threshold. This cytometer has to know what's a cell and what's not a cell, what's debris and what's a cell. And you're the one that's telling it where to set this line. It sets this line, and so you only collect data on what you've told it is important to collect. Everything else is lost. And so if you set your threshold wrong or your settings wrong, your PMT settings, your cells might be below threshold. If you don't see them going through when you're acquiring it, you're not going to see it next week when you're analyzing the data. So if we run these same cells through with the threshold, and the threshold here, you cannot see it, but this cell would not be visible. This little pulse, if you run that cell through at 300 volts, will be below threshold. If you run it at 400, you will see it. If you run at 500, it will be over here. And if we run through a series of blood cells that are different size pulses, and we look at 3 different thresholds here, here the threshold is very low. All of the pulses are above threshold. You can't even see where the threshold is, but you can see that the population of cells here, you've got your lymphocytes, monocytes, granulocytes, and you've got a whole population here. If you take this threshold up higher, some of these little guys are lost. You can actually see the line where the threshold is, and you can see that you've got a partial population that's probably lost. If you run it up here, you're losing all of these, and you wouldn't even know that this population exists. Now, you may not want that population. It's probably platelets or red cells, but you should be aware of where your cells are and be sure that the cells that you want to collect are actually above threshold here. And so we'll talk about this a little bit later, and I won't summarize here because I'm out of time, but we can do up to 40 colors now with the new spectral instruments. And I think somebody may be talking about those in the future. So thank you very much. And any questions this afternoon, I'm happy to answer. We'll go into more detail.
Thank you to Dr. Sasha Masharovsky, PhD, for sharing with us, and thank you for a great explanation. We will now begin our question and answer session. Ladies and gentlemen, we would like to invite you to raise your hand. If you would like to ask any question. Once selected, please state your name first before proceeding with your question. Thank you. Yes, Ma'am.
Thank you, Dr. Zazir. But I think because you have mentioned that we will have another session that we can have a
Yes.
further discussion about it regarding this light schedule.
Yeah.
So I think I missed that a lot, even though I used the Flowcytometry Gallios, I think, for quite a long time. As you mentioned that this instrument can be used to determine the diversity of the cells.
Yeah.
Say, if I take from very particular gating,
Yeah.
can I just use pick that one gating to examine whether I got the uniform cells, or maybe still I got another cell, say that gating supposed to be NKT, but according to the height of voltage, something else appear. What is in your horizon about it, whether that's possible or not?
So you should be able to gate on the
Yeah.
one population and look at it. You're saying that there are more populations in that than you expected. Maybe, yeah. And so then you have to determine downstream from that gate whether or not you've got the correct markers to separate out the populations of cells that might be in there. So you can add different fluorochromes to see what mix of cells you might have in there. But the side scatter, the forward side scatter, the size of the cell will not give you something pure in general.
Alone can be used if we don't know yet.
The height of the voltage pulse, you can either use the height or the area of the voltage pulse on the forward scatter, you mean? Or on the forward scatter. You can use that to identify your population and then look at the other parameters downstream. If you're not sure where the population of cells that you're interested in, say you're looking at a certain marker that's positive, maybe a CD4, CD8, what you can do is on an ungated population with that parameter, look at the positive cells, backgate them over to your forward scatter, side scatter, and see where they lie on your forward scatter, side scatter to be sure that you've got them all on scale and above threshold. But if you want to show me some of the data this afternoon that you're looking at, that you're worried about, we can discuss it.
Yeah, but I think I got one.
Okay.
Thank you.
Okay, thank you very much for very good about the introduction of basic principle. So I asked about the related to the forward and scatter. You said the forward and scatter. Any difference between the degree of the scatter between instruments, and it is possible can become variation between instruments if they have different degree between
There's
instruments.
difference between the instruments, and
Yes.
even within the same kind of instrument, it depends. There are bars in front of the detector that can be angled slightly different to let more or less side scatter through or forward scatter through. So yes, there are differences between kinds of instruments and even between individual instruments.
Is
You know,
there any
they're pretty
impact
well
to the
standardized,
standardization?
but yes, between a sorter, a BD, a Coulter, another instrument, you will get forward scatter, side scatter that looks slightly different.
Okay, I think you may.
But that's okay. They're all pretty good. Usually it's just you need to know where who is.
Okay, thank you. Maybe second question related to the bandpass.
Yeah.
There is a lot of option of the filters. How we can choose the best filter for us based on our experiments? It is possible we change by ourselves, or we need to choose best and based on our
A
experience.
lot of instruments you can just switch them out. So if you've got a fluorochrome that's not standard and you want a different bandpass, a lot of them you can just take the bandpass out and order a special bandpass that's really targeted to your fluorochrome. Normally you don't have to, but yes, you can. If you've got something that's a little bit not one of the normal fluorochromes, then you can switch it out. You need to be sure in a multi-user thing that you switch it back, so the next person doesn't have a filter they don't know about. That happens to us quite a lot.
Okay, thank you.
That people switch them out and then they don't put them back. But yes, you can. There's a huge range of filters. Practically anything you need, you can find.
Thank you very much.
You're welcome. Is there a question?
Thank you. My name is Ibnu from University of Indonesia. I want to ask about, and at the first I want to say thank you for the very clear explanation about the basic flowcytometry. In your slide, you also explain about what is the difference between current flowcytometry with full spectrum cytometry. For example, with site of Aurora, I think.
With the Aurora.
Yeah.
So in the conventional, I don't know, is anybody talking about full spectrum? Paul is talking about, okay, Paul will discuss full spectrum, but instead of measuring the fluorochrome off one laser just through a bandpass, it's looking at the fluorochrome's excitation and emission across all 3 or 4 or 5 lasers, so that you can distinguish fluorochromes that have emission that's very similar off of one laser, but might be different excitation and emission. So quite often you can use fluorochromes together with the full spectrum that you can't possibly use on a conventional flow cytometer.
Yeah.
And then you can go up to a lot more colors on the, and full spectrum is really the future for flow cytometry.
And if I read in general about full spectrum cytometry, compare to current flow cytometry, we need to perform compensation. It is correct, we don't need compensation again if we're using full spectrum flow cytometer.
You still need to do compensation with full spectrum. It's not called compensation, but it's the same thing. But yes, there's still overlap with the fluorochromes. You don't get away with that. And the other important thing is panel design. And Paresh is going to talk about panel design and spread. You still have to look at that for full spectrum.
Thank you very much.
Thank you.
Is there any other question? Maybe you can raise your hand if you want to ask
This
something.
afternoon, any questions you want.
If not, maybe we can close the question and answer session. And thank you. Thank you to Dr. Sofia Masowski, PhD, for sharing with us. And thank you for the question.
Thank you very much, Dr. Sofia Masowski, PhD, for sharing with us. The second lecture will be delivered by Dr. Paris Chain. As you can see, Dr. Paris Chain have a lot of experience, especially in hemato-oncopathology. He is currently working as Associate Director, Clinical Solution Platform, BD Bioscience Central at South Asia and Japan. He also had trained in hemato-oncopathology at the All India Institute and establishing several BD centers of excellence for education and training in flow cytometry. To Dr. Paris Chain, you have our full attention.
Thank you. Good morning, everyone. I would like to start with thanking Dr. Devi for organizing such a wonderful event here, and for Dr. Liana for the scientific agenda and for inviting us to make a presentation here and share our experience with you. So I think based on the strong foundation built by Sasha on the fundamentals of flow cytometry, I take it to the next logical step, deploying those principles for doing multicolor flow cytometry and discuss some of the 4 core concepts of the panel design. The first question is, why do we do flow cytometric immunophenotyping? Now, what the objective here we are trying to do is define subpopulations of cells based on presence or absence of certain antigens. That is what immunophenotyping is all about. Now, what it does is it allows to reveal the heterogeneity which may be in our cell populations. Now, what I mean by that is, for example, you see here on a forward scatter site scatter plot, this is apparently homogeneous populations of lymphocytes. The moment we put certain markers like CD3 and CD19 in the same sample, what we see is this is not a homogeneous population, but it has 3 different populations. One is CD3 positive T cells, 19 positive B cells, and the double negative NK cells. Now, the same thing can be expanded by adding more and more markers and revealing the further heterogeneity that may be there. Like we may think that this CD3 positive population is a homogeneous population, but it is not. Once you add more markers, one would see clearly that within the CD3 positive T cells, there can be CD4, CD8, that within the CD4, CD8, there can be further subpopulation, which can be characterized by the presence or absence of those antigens. So each specific subpopulation has a unique immunophenotypic signature. And when we put a lot of different markers, it allows one to study and evaluate which of these subpopulations actually correlates with maybe the disease process, response to the treatment, or the final outcome of the patient. So that's why the interest is in being able to use a multicolor panel design and add more and more markers to that. The other benefit of a polychromatic immunophenotyping is that each additional color can significantly increase the number of populations that can be detected and quantified with lesser number of tubes, means lesser sample required, lesser reagents required, and lesser time required to acquire that and analyze that. Now, this is just to give you an illustrative example. In the old days, when we are doing 4-color flow cytometry to do B lineage, ALLMR panels, the same number of markers, we would need a gating markers for CD19, 10, and 34, and you will require here 5 tubes to study all these markers, 45, 20, 58, 38, and 123. Now, when we move to 6-color panel, instead of this 5 tube and the requirement of almost this, what you see here as a 20 markers, we could get the same information in just about 2 tubes and a total of 12 markers used here. The same thing when you move to an 8-color panel, one can see all that information can be obtained on a single tube 8-color panel here. And now, the advantage being that all these populations can be studied simultaneously, giving you a richer biological interpretation from the sample, rather than trying to make inferential reasoning when you're trying to run them as all individual separate tubes. Now, so with this knowledge, now let's understand if we have to make, for example, transition from a 4-color to 8-color panel, what are some of the principles that we need to bear in mind? Now, these rules are common, whether you're doing an 8-color panel or you're doing an 18-color panel, they will remain the same. So your objective is that you have a right antibody conjugated with the right fluorochrome in right combination with other markers to allow the best discrimination between the different subpopulations, minimizing the background and minimizing the spread. Now, the single most important step is to know what you want to do and lay out your experiment blueprint with an analysis hierarchy and antigen density characterization. We'll be going over each of these principles in little more detail in the subsequent part of the talk. The second step is selecting the fluorochromes according to the instrument configuration, which is relatively an easier job with all the tools available today. The third step is matching the fluorochrome brightness with the antigen expression level. The fourth step, the point, and which is the one I would really highlight a lot in this talk, which is often missed, is minimizing the impact of spillover, fluorochrome spillover related spread. And last but not the least, running some fluorescent -1 controls, FMO controls to validate your panel. Now, by analysis hierarchy, I mean that it's important to write down how you're going to analyze your sample eventually once the data is acquired. For example, I'm giving an example of if you're trying to, your objective is to differentiate naive and memory Treg, enumerate, and quantify naive and memory T regulatory cells. So you start with, for example, a CD3 positive marker, CD3 marker as a T cell marker, then you're adding CD4, 8, and CD4 and CD8 to identify your T helper cells. Within that, a 25 and 127 to identify the Tregs, and then using a CD45 array, to distinguish between the naive Tregs and the memory Treg cells. Now, this hierarchy one needs to write not after you have acquired the data, but before you even start with your panel design exercise. And for the markers that you have chosen, then it becomes the next step, which is important, is to understand approximately what is the antigen density. Now, for some of the commonly used markers, that information is more readily available in literature. And for example, we do know that CD3 is at around 30,000 molecules per cell. and CD4 is around similar. CD8 is very high antigen density, 65,000 antigens per cell, and then CD25, 127 one can see are expressed relatively dimly. Now, so there have been some structured approach to qualify and these antigens as either primary antigens, secondary antigens, and tertiary antigens. So what are primary antigens? Which are generally well characterized, easily classified as positive or negative, and they're useful for generally lineage identifications, broad subsets like CD3, CD4, CD45. These are your primary antigens, with a very well expressed, very well characterized. Then there are these secondary antigens. They are also well characterized and also may be expressed at high density, but often as a continuum. You see, there's a kind of a continuum of expression you see on the T cells for a CD45 array here. Now, then there are these tertiary antigens, which are either expressed at low levels, or these are just activation markers, normally absent, but when you activate the cells, they become expressed, or you do not know. If you do not know about the antigen density of a certain marker, then just assume that that is a tertiary one and which will be low expressed, and therefore you assign the fluorochrome accordingly. Now, then we come to the next point of selecting the fluorochromes according to the instrument configuration. Now, this is so here you need to be mindful of what is the laser wavelength available to you for excitation. In the most of the clinical instrument that we see today, blue, red, violet lasers are quite standard. And you also need to know what are the number of detectors that are available of each laser. For example, of the blue laser, on a BD fax lyric, you will generally have 4 fluorochrome detectors. On a Beckman Coulter, DX Flex, or Navios, you will have 5 detectors there of the blue laser. Similarly, on the violet laser, on the BD1, you will have about 5 fluorochromes there. In other instruments, there might be 4 fluorochromes available there. So your choice of panel will depend upon that. What is the laser line available? What are the number of detectors which are available? And of course, you also need to be aware of what are the filters which are used in that, and accordingly, you will make your choice of the fluorochrome. Now, all vendors provide good information as a part of their brochure, spec sheets, that what fluorochromes are compatible. So this is an information which is readily available, but you must make an effort to make sure that it is available to you before when you're doing a final design. Now, once you know what fluorochrome options are available, now the next step then will come around. Next step will be now around matching the fluorochrome brightness. Now, to understand what you mean by fluorochrome brightness, you need to know a fluorochrome brightness of a dye or fluorochrome depends upon how well that fluorochrome is able to absorb energy, and that is known as the molar extinction coefficient. And then it also depends upon the quantum yield, which is what is the ratio of photons emitted to photons absorbed, how well the fluorochrome is able to release the photons. Now, the brightness is a function of both the extinction coefficient and the quantum yield. Generally, it is the ability to absorb the photons is more important in determining the brightness. As you can see an example here, the PE Sci5 dye and the Pacific Blue, they have the same quantum yield. But the brightness of PE Sci5 is much higher compared to the Pacific Blue because the molar extinction coefficient is very high for PE Sci5 compared to the Pacific Blue. Now, you and I have no means to measure the fluorochrome, the extinction coefficient, and the quantum yield for each fluorochrome. So what we do in the practical way in the lab is what we measure a stain index. This is what will be done by Sasha in the afternoon experiment. She'll show you the stain index, how it is calculated. So this is a practical way. So stain index is given by the formula, the brightness of the signal, which is the median of the positive, from which you subtract the median of the negative and divide the whole thing by the width of the negative, which is the spread that you see in the negative. So these values you can easily obtain from any statistical software, which is their part of the Flowcytometry software, and calculate the stain index. Median of positive minus median of negative, and then divide by 2 times as negative, the spread that you are seeing, the noise that you are seeing in this. Now, the median of the positive is a function of fluorochrome brightness, which we have discussed, and of antigen density as well. So when we calculate the stain index, we will use only one antigen. Maybe example CD4 or CD3 conjugated to different fluorochrome. So you have control for the antigen density part, and now the value that you will get on the positive will come entirely from the fluorochrome brightness. The medium of the negative is dependent on the autofluorescence or the non-specific antibody binding. And if you are using the same cell, for example, lymphocytes as a population of interest, then you know the autofluorescence is same and the background is practically the same for all of them. And the spread is coming from the multiple factors, the instrument factors, and the spread which come because of the spectral spillover, and that's something we'll be studying more later. Now, for several years, for almost 2-3 decades, the PE was the dye which was considered as the brightest dye, had the highest stain index. But of late, in the last about 8 to 10 years, there have been many new dyes which have been now discovered and are commercially available, both from BD and from other vendors as well. And again, these charts, they are quite helpful of what is the approximate stain index value available. Mind you, stain index is instrument dependent. It will depend upon the laser power you are using, the actual filter you are using, and each individual, although it may be a blue laser in instrument 1 or instrument 2 from the same company, there is a slight difference there. But it is helpful, it is important to know the stain index, to do that exercise once. But from a panel design perspective, it is appropriate as long as you are able to qualify your dyes into the broad categories of very bright, moderate, and that is good enough. You do not need to know the exact stain index value and number as such it is, So that's not very critical for the panel design. So, your very bright dyes, for example, are some of these Cerigen dyes, Horizon dyes are very bright. And then the CP, PSI7, and some of these Cerigen dyes are quite bright dyes as well. APC is a bright dye. Among the moderate dyes comes the BB510 per CP Psi4.5, and among the dim dyes are per CP, and for example, APC Psi7, APCS7, these are the dim dyes. Okay, as long as you know this broad categorization, that is good enough. And then the next step is pair the bright fluorochromes with low expressing antigens and dim fluorochromes with high expressing. Now that's common sense. Okay, so that's not very hard as well to do, as once you have qualified your antigen densities as low, medium, high, and then you have your the fluorochromes, the very bright, moderate, and dim. And even if you have a 3-scale bright, moderate, dim, that will work quite well as well. There need not be a 4-scale ordinate to distinguish your dyes. A bright, moderate, dim will work quite well, and then you pair them accordingly. Now, then comes the next step of minimizing the impact of spillover related spread, and that is where we'll spend most of the time in the talk today. Now, let's understand this here. It's showing per CP Psi5.1, Psi7. We can clearly see here that there is some spillover that we have per CP Psi5.5 into the Psi7 channel. The boundaries that you see here are the boundaries of the band pass filter. And so this spillover into the Psi7 channel, what it does when you do a single stain control, when you have a single stain control of Psi.5, it introduces a background into the Psi7 channel. As you see here, the positive population uncompensated shows a background signal of about 3000 intensity. Now, this, along with the background, what you also see here is that this population has a certain amount of spread of 291 channels compared to this unstained population. The reason why it looks still so tight, although the spread is very high, is because you're looking the data on the y-axis on the logarithmic scale. Now, as you compensate, the background is subtracted in this process. So once you compensate appropriately, then what you see, once you have compensated adequately, what happens is the background became equivalent to the background that you are seeing for the negative population. But what you observe is the compensation is able to correct for the background, but the spread introduced by the spillover is not removed by the compensation. And this is the one which will eventually be reducing the resolution of any double positive cells. You can see here you will be able to resolve the population, but here you will not be able to resolve a double positive population. And as you add more and more fluorochrome, this becomes more and more pronounced there. Now, it's important to understand at this stage, what are the sources of spillover? Now, I like to use this kind of a chart to mentally guide me when I'm doing the panel design. I put the laser lines available on the one row, the kind of filter options broadly. Again, it is all broadly is okay. Approximately this dye is going to emit at this channel. Let's say we know FITC is going to emit at around 500 to 530 nanometer. So I just put them in the order here. Now, what you see, the most commonly source of spillover is adjacent detector. As you move within the certain laser line, as you move from left to right is what you see. For example, in the blue laser, you have FITC and PE, most common example. Because PE is sitting just right to the FITC, there will be a substantial amount of spillover of FITC into PE. There is also some amount of spillover of PE into FITC, but that's generally lesser. So from the right to left, there is less amount of spillover. From left to right, there is a more amount of spillover. Now, would you imagine that? How much do you think will be the impact of FITC spilling over into P size 7? Very less, because they are spectrally very far from each other. So even if you don't know, if you have this chart, okay, so FITC going to spill over into P will be more, slightly less into PCF594 channel, even further less into the Percy P Psi 5.5 channel, even lesser into the P size 7 channel. Okay, and would you think that PE size 7 is going to have a spillover into FITC? No, they're very far from each other. There's no business for it to have a spillover there. So that's how you remember the adjacent detector, more spillover. It's from left to right, and the immediate neighbor, there is more spillover there. Then the second source is where the emission spectra is same, but the laser lines are different. Now, what, for example, a classical example here, you see PE Psi 5 on the blue laser and APC on the red laser. They have almost the same amount of emission spectra. The laser lines are different, but there is some component of the Psi 5 which will get excited by the red laser as it passes, the cell passes through that, and it will directly spill into the APC channel. A huge amount of spillover and huge amount of spread. So again, while this can happen in both the directions, generally you will find, therefore, what will happen? A BV711 will have a high amount of spillover into the Alexa 700 channel or APCR 700 channel. A PE size 7 will have a spillover into the APC size 7 channel. Okay, although they are different laser lines, this spillover is happening because of similar emission spectra. We also call it as a cross excitation laser expression. It's getting excited by two different lasers, but still you are seeing some kind of an emission overlap there. The third source is what we call as a tandem dye spillover. What it means, so that it skips. So till now you are talking anything which is adjacent, that is where you have the problem. Now what is happening here is PE size 7 is a tandem of PE. There always will be some spillover that you will see in the PE channel because of the what we call as the tandem is not complete. There is some residual spillover, you will see that. Similarly, APC size 7 or APCH7 will have some spillover into APC. And a BV711, it is not a standalone dye. This also is a tandem dye. It's a tandem of BV421. So a BV711 will also have a spillover into BV421 channel. It does not matter whether in the violet channel here you are using, suppose you say, oh, I am using V450 dye and a BV711. BV711 will still have an impact on the V450 data because it's a tandem, and the tandem will still give you some background spread in this channel, whether you are using BV421 or you are using V450. That dye itself is going to give you a tandem effect there. So there are only these three sources of spillover: adjacent detector, common laser excitation, common emission spectra, different laser excitation, and the tandem dye. So irrespective of what the source of spillover is, what one need to understand now how this spillover introduces the background on spread. So the amount of spread that you see is a function of, first of all, amount of spillover. And we have already seen from the previous slide, how do you calculate the approximate amount of spillover? I mean, even if you don't have the numbers. Now the PE will have a huge spillover into P psi 5. Why? Same laser line, and psi 5 is just sitting right side to the PE. Okay, so there is a certain amount of spillover, which is around 10%, and you see a certain amount of spread in the data here. However, if instead of PCA594, if you had a FITC, if instead of psi 5 you had a FITC, now FITC is sitting left to the PE in the same channel. Because it is in the left to the PE, the amount of spillover is less. Therefore, you do not see the spread in the signals here, in the PE positive signals. If you now move, instead of P psi 5 or FITC, you go to P psi 594. Let's have a quick look. Where was P psi 594? See? Here, if you see, this is PE, this is FITC, and this is P psi 594, and this is P psi 5. So where do you think this maximum impact of the spread will be from PE? It will be in this channel PCA 594. Correct? So this is what we were seeing here. So in PCA 594, the spillover is higher, and therefore the spread impact is also highest. Now, the second function is the function of reagent brightness. Now, the reagent brightness is, now suppose the signal was at a certain level here as 7000 intensity for the PE, and you see a certain amount of spillover. If, however, the signal of the PE was lower, it was a dimmer antigen, now you do not see as much of spread. If, however, the PE becomes very bright, okay, you may have seen CD15 signal in the neutrophils, or CD16 signal in the neutrophils, CD38 in the plasma cells, very bright signal. So very bright signal means, again, you will have a very high spread. So in multicolor flow cytometry, bright is not necessarily always good. It should be just enough bright to resolve the population, but you make it more brighter, and then you will start seeing all kind of spread related issues. Okay, so this itself is the function, as I mentioned, is a function of antigen density and the fluorochrome brightness. Now, the population resolution for this PCF94 is decreased by the increasing spread due to the P spillover, and you will not be able to resolve the populations there. So to maximize the resolution of a given double positive population, you need to minimize the fluorescent spread in that detector that defines that population. So if that population is primarily defined by the expression or the lack of it in this channel, then in this channel you should have the minimum amount of spillover, and therefore the minimum amount of spread. Now here again, looking at an example here, what you have CD3 and CD56. What is you see here? Here the CD3 positive, 56 positive population is not very well resolved. Why? Because it is this pop in the 56 channel, there must be many fluorochromes which are spilling their signal, and therefore this T positive population is showing a lot of spread, and you're not able to resolve. Once you correct for that, now you are able to identify and enumerate the population with clarity. And more the number of colors that you have, more is the chances of spillover and greater is the spread. And that is why it becomes, whatever was working well in your 4 color panels, the moment you move to 8 or 12 color panels, these complexities start appearing there, and one has to be mindful. Okay, so I think something coming up. Great. Okay. So I think the HDMI needs to be just put harder. Can you just tighten the HDMI port? No, this is okay. So I think I'll continue. Maybe try to just HDMI port, try to tighten it further. Okay. So now the population resolution given fluorescence is reduced by increased spread. Now this is an important slide. Now what you see here is this: there is a certain fluorescence fluorochrome X here, and there is certain fluorochrome Y here. Now, the population resolution for this fluorochrome Y is reduced by increased spread due to spillover from the fluorochrome. It could be X to XN in that panel. Suppose you have 8 colors. So any of those 7 fluorochromes, if they spill into the fluorochrome Y, they will cause a spread, whether you are showing in this plot or you're not showing in that plot. Now, so the impact of spread can be understood by, for each fluorochrome X1, how much is the spillover into Y channel? Then you multiply also that by the reagent, it is directly proportional to the reagent overall brightness of the signal that you are seeing in this channel. Brighter the signal on the X channel, more will be the spillover impact on the Y channel. And the impact of spread is inversely proportional to the brightness of the signal you are seeing on the Y channel, which itself is a function of fluorochrome brightness and the antigen density. So if you have a highly dense expressed antigen here, then the spread may be there, but the impact would be reduced. And I'll show you all this with examples later on. Ignore this point for the time being. And of course, all this is valid if the antigens are co-expressed. If these are CD3 and CD19, which are not co-expressed, then you're not worried about the impact of spread at all. Or if it is CD4 and CD8, which are not co-expressed in most normal cells, in leukemias, we do see they can be co-expressed as well. So, but then if they are not co-expressed, then there is whole impact of spread is 0. Okay. So if the spillover is 0, impact of spread is 0. If the antigen co-expression is not there, again, the impact of spread is 0 for that combination. Now let's go back to our example of CD3 and CD56 and see if you have to improve the resolution of this double positive population, how would you do that? Now let's look at the spread equation and see where all you will make a difference to improve it. Now, one option is, if let's say in your panel, instead of 56, you use something brighter, like say CD8. I showed you that CD8 was something which was highly expressed, 65,000 molecules per cell. Now, the moment I use CD8, keeping the fluorochrome same, where I have changed in the equation? I have put a change in the equation here. The antigen density of the Y-axis channel is much higher now. Okay, so although this population is still spread, I am able to quantify my CD8 positive T cells very easily. The other approach could be now I am titrating the CD3 reagent to reduce the spread. Okay, now where am I playing in the equation? I am playing in the equation at this point. Now I have reduced the antigen density in a certain antigen density is same. Because of the titration, I'm using less molecules. So overall, this signal has reduced. So I'm able to resolve it much better now. The other approach could be, just use a fluorochrome on CD3, which has a less spillover into CD56. So where in the equation I am working now? I am working on this part of the equation now, where I know that the spillover of this fluorochrome on the x-axis is not high into the y-axis there. So once you have the spread equation in your mind, you can see that where you need to work on the equation to improve your panel and improve your signal resolution. Last comes the FMO controls to validate the panel. Now, what do you mean by an FMO control? Now, FMO control, they inform you about the impact of spread on data analysis. Okay, now take an example. Suppose there is an 8-color panel. If you have to run an FMO, it means you need to add all markers except that one marker. So if you have to run a full FMO panel for an 8-color panel, you will need to run 7 different tubes. Now, in strict sense, one should do that, but in a practical way, it becomes very impracticable, at least in a clinical setting, to do that. So what I would do is I would focus on those combinations where I know the marker expression is critical, where is likely to be high spillover into that channel, and run FMO controls just for that purpose. So it depends on your context, case to case, how many FMO controls you really want to run as a part of your panel validation. I'll give you an example around that. Now, see here, this is a cytokine panel. Now, gated on lymphocytes, this is 3+4 positive. This is gated on 3+4 negative. Now, what do you see here? in the 4 positive population, you are able to see interferon gamma signal very well. However, in the 4 negative, that means it must be the CD8 positive cells. You see there is a high spread that you see in the FITC channel. And that spread is coming, why? Because the AMP-cyan dye is spilling into the FITC channel by cross laser excitation. AMP-cyan is violet laser, FITC is blue laser, but they have similar emission spectra. So on that similar emission spectra, because of that, you are seeing this spill over here. And you are seeing the spread and you are not able to identify the population. How you identify that? One approach is you run FMO control. You run a panel where you have not added AMP-cyan. It's a fluorescence -1, and that -1 is minus AMP-cyan. And we find that once you do not add AMP-cyan, then in the CD4 positive and in the CD4 negative, you are still able to see the interferon gamma positive cells very well. Well, you can run an FMO control. The other way I would say is just remember your fundamentals of flow cytometry. You know AMP-cyan is going to spill over into the FITC. How do you know that? From that spillover guide that I showed you. You know AMP-cyan will have a spillover, then why make a panel where there it is, you know, that they're going to have this problem there. Now, this is something now, let's build on that, the knowledge that we have done till now, and we build on that to see how we make panels and how by changing the panels in a real world setting improves your data quality. Now, example, I'm going to take the same example that we had the six color panel on a simple 2 laser instrument, blue laser and red laser. Now, experimental goal is to identify the T regulatory cell subsets as naive and memory. So you need markers 3, 4, 8. You need markers for Treg 25 and 27, and then you have 45 RN as positive for naive and negative for memory Tregs. Now you classify your markers. CD3, 4, and 8 are your primary markers, very well expressed, very well characterized. Your secondary marker is CD45RA, and your tertiary markers are which are dim express or activation markers CD25 and 127. Now, the first example I'll show you is when you do not follow any rules. The previous student in the lab, he had some reagents, you had some reagents in the lab and in the fridge, and you just use those available reagents without paying any attention to fluorochrome brightness or antigen density. The second example we will take when you follow some rules. You pair the bright fluorochrome to dim antigen. And the third example is the best practice example, and we'll see how the results will improve. Okay. Now, in this, what we see, when you simply just run with whatever was available with you without paying attention to the panel design principles, okay, and you have this choice of the panel there. So CD3 is what we see is very well resolved. Then CD4 and CD8 are very well resolved here. Within CD4, we are looking at CD25, 45RA, and we are looking at CD25, 127. You can see your you can see your Tregs a little bit, but overall you do see that 25 is not very well resolved here. It's not very well resolved. Now, why? Now, what happened? Not following the panel design rule resulted in, you can see the 127 is very dim, and it's a dim marker and a dim fluorochrome. Okay, it's poorly resolved. So the high antigen density like CD4 and CD8 have been incorrectly paired with the bright fluorochromes. Why would you want to put them with the bright fluorochromes? And the low density antigens like 25 and 127 are incorrectly paired with the dimmer fluorochromes. So you are not able to resolve the populations very well here. And you may think that, oh, the Treg numbers are, you will under enumerate the Treg numbers. Now, when you start putting some rules into practice based on the fluorochrome pairing with the antigen brightness, now what you see, some caveats to remember are that the same antigen can have different level of expression on the different cell types. Now, this is quite commonly your CD7 may be bright expressed on the lymphoblast, T lymphoblast, but may be dimly expressed on your myeloblasts as a antigen aberrancy. So you need to be mindful of that. CD38 in plasma cells may be very bright. 38 on an activated cells or or on a BCLPD cell may not be super bright. And antigen levels can also vary due to activation state and functional differences. Again, becomes important. HLA-DR in the TCLPD panel is an activation marker there. HLA-DR on a monocyte is a very bright marker there. Now, markers can also be expressed over a continuous range. Now we have already gone through the fluorochrome ranking. Now let's start pairing it. Now, what we do, how do you assign? C25 dim, we give it to the PE, bright fluorochrome. 127, we take it to P size 7, which is also a bright fluorochrome. 45 RA, it's a medium antigen, okay? And we give it to APC, which also is a bright fluorochrome. Then CD4, bright antigen, moderate fluorochrome. CD8, very bright antigen. I think it's got stuck. Good. Okay. CD8, very bright antigen, dim fluorochrome. CD3, bright antigen, dim fluorochrome. Now see how it improves your data quality. Now CD3, although it's a dim fluorochrome, it is still very well resolved. We see that. CD4 is, we see now again very well resolved. The change of the fluorochrome has not made any difference. But now what you see, the populations have started becoming better defined. In the 25, clearly we are able to see two populations now because it is assigned to a bright fluorochrome. Similarly, the Treg population here, because 127 is there on a bright fluorochrome, you can see just much better population here. So
Then,
on
Dr. Pares, the time is about 5 minutes left.
Sure. Thank you very
Thank
much.
you very much.
So what we see here, first example on the top, second example on the bottom, you see 25 is better resolved. And here also 127 is also Treg is better resolved. So using dimmer fluorochrome for CD4 did not impact the resolution of CD4 T cells, but using brighter fluorochrome for low density 25 and 27 markers improved their resolution. Okay. Now, but it is still remaining challenging to gate on the Tregs. You see, you still have some population which is going into the axis here. The reason is let's study the impact of spread, because the spillover and co-expression were not taken into consideration. Now, if you were to go by the panel design principles, you would have first laid out your analysis hierarchy. And you see here, if you conjugate CD4 to per CP psi 5.5, and then downstream in co-expressed molecules, you have CD127, which is dim and which is on P size 7. Now, there will be a spillover of CD4 per CP psi 5.5 into 127 channel, and therefore the resolution ability will be diminished. So and this is what you can reveal either by that exercise, or you can run a FMO control. What is FMO control telling you? Now when you had an FMO of per CP psi 5.5 where you did not add CD4, and now if you look at the signal, you see this is so very well resolved. Okay, so you know that per CP psi 5.5 was the culprit which was not allowing you to resolve the dim 127 with the moderate 127 expression. So either you run a FMO control, or you simply, as a part of the analysis hierarchy on a whiteboard, you understand where the impacts are likely to be there. And even if you may still need to run FMO controls in certain settings, FMO controls are still quite important. Now, if we want to optimize again now, so we keep CD3 same. We CD8 instead of APCH7, now CD8 we will take down to per CP psi 5.5, and CD4 we are making APCH7. And we make changes so where the CD127 now moves to P. So CD127 is now moving actually to the Alexa 647 here, to the APC channel here. Okay. And CD45 RA is the one instead of this. So the CD1, so in P size 7, we will put CD45 RA and we swap them here. And 3 and 25, we keep the same. Now let's see the impact of this. So now what you see, again, CD3 gated, CD4 gated, then within that we look at 25 and 45 RA and 25 and 127. Very well resolved population with clear centers of density that you see in your target population. So now we see second example. This was your second example, and this is your third example. And you can see compared to this, in our population here, the resolution at this level is much better compared to what you are seeing here. So populations are very well resolved. Now, looking at all of them together, can you move one slide forward? Maybe you can manually move it. Can you move one slide? Okay, resolution of both are improved. So now you see all the three examples here. The first example, the basic rule of panel designs were not applied and the T-reg resolution was suboptimal. In the second example, panel optimized to use bright fluorochrome for low expressors. So we saw that the resolution of 127/25 was improved. The third example, panel optimized to minimize the loss of resolution due to spillover, and we see it is fully optimal panel now. Your proper density you are seeing for your T-reg population. So this is how, just to show an example, why it is important to follow the principles of panel design in that order so that you get the best outcomes in your actual data resolution. To summarize, so the steps in multicolor panel design are: lay out the experiment blueprint with analysis hierarchy. That is absolutely must. And then selecting fluorochromes according to instrument configuration, matching the fluorochrome brightness with the antigen expression. And the fourth step, again, a very important step as you add more and more number of colors, which is minimizing the impact of spillover due to spread. And lastly, for some selected examples, you may run FMO control to validate the panel mostly. So we have covered what is fluorochrome brightness. We have covered what is fluorochrome spillover and the sources of spillover, adjacent detector, similar emission spectra, tandem dyes, and the importance of spread. Lastly, but not the least, spread equation, which forms an easy, back of the envelope tool to understand how good or a panel is likely to look at, and you follow this principle, I think you should do, quite well in your panel design. Thank you very much.
Thank you very much, Dr. Paris Chain, for the beneficial lecture for all of us. Moving right along, we will begin our question and answer session. But while we have a question and answer session, we also will have a coffee break, and light refreshment will be served to all of you, to all of the participants, and also for the speakers. And for ladies and gentlemen, we would like to invite you to raise your hand if you would like to ask a question. And once selected, please state your name and before proceeding with your question. Thank you. Is there any question? You can raise your hand.
There's a question here.
Yes.
Okay, thank you very much. Very interesting. It's not easy to optimize the experiments based on this information. A lot of things we can do now. I have some question related to do you ISAC have tools how to make easy to us to combine to optimize to reduce spillover? Do you have tools for that? So if we have experiments, we can add all our experiments at that tool, so we can easy to make to choose the combination. Thank you.
Yeah, see, the only way I can see is follow the principles in that order. So often what we try to do is, just want to make up, we get the, antibodies and we try to run it. But if you follow the steps as given here, you will find that most of your panel design issues will be solved. And it will take longer time in the beginning because you may not remember which fluorochrome spills into what. So these slides will be there with you as a handout. The spillover guide that I showed you will kind of guide you around that what fluorochromes will have a problem or not. If you see the two fluorochromes are lying far apart, you can easily pair them there. So knowing your biology of what you want to check and why you want to check, knowing your instrument, what fluorochromes you can run, then knowing your brightness of the dye, knowing the spillover, that knowledge, if is all there in the head, the panel design process is quicker. Till the time you reach that stage where it is all in the mind, have those tools and charts in front of you, sit on a big whiteboard and then try to look at that. Oh, and follow the principles strictly in that order. You will find it will work. It will take some time in the beginning, but you will get to it.
Okay, thank you. Because sometimes related to supply chains, not all reagent we can buy from one. Sometimes we need to buy from BD, from Beckman, or from BioLegends, so we can combine for fit CPE. Or any difference from fit CPE from that's all companies, or how we can optimize for that?
Good question. So by and large, FitC is a FitC dye or a P is a P dye. From two different vendors, it will not make much difference there. The difference at most would be that how much of PE molecules they have bound to that antibody. That may have a bearing on the your brightness of that signal there. Okay. So now in that case, if you may want to run the stain index in your experiment using those dyes, but most likely, you will not find that difference there. And as I mentioned to you, each vendor generally provides you qualitative information, whether that dye is very bright, moderate, dim. You can ask the vendors that. You say, you want to sell me this reagent, conjugate that to CD, show me the CD4 data of that fluorochrome in your instrument, or show me the CD3 data of that fluorochrome in your instrument. Compare that across different vendors. Ask that to all the vendors. Okay. And then you do not be worried about it is slightly brighter, low brighter. Just you need to qualify, are these bright? Are these moderate dyes, fluorochrome brightness, or these are dim? That's more than enough. You don't have to be a little bit worried about here and there. And as I told you again, bright is not necessarily always good in a multicolor panel design. At times, you may purposefully want to use a dim dye as long as it resolves your population, because dim dye will give less headache as spillover into other channels and less headache of spread in other channels there.
Thank you very much. Thank you.
Sure.
Thank you very much. Is there any question? You could raise your hand and state your name first.
Oh, good.
Hello, Dr. Paris. I'm wondering about, but I forgot what slide it is. You show about the 3 panel to the right, 3 panel to the bottom. But in the central panel, if I'm not mistaken, there is one top panel in the central panel on top. There is the gating cross about a very distinctive population, and this I'm not sure, is it 7 prior to the last slide maybe? So I'm not quite so what is the gating about, because that's obviously 2 distinctive populations, but getting into one.
So I think it's not allowing me to move the slide. So what I'll do is in the break time, I'll just come to your desk and see what question you're asking, which slide you're talking about. Oh, is it this one?
No. 3 panel. No.
No. Let me know once you see it.
Not this one, not this one. Before
This
that.
maybe? No.
Oh yes, that one.
Okay.
Yeah, the center on top.
Here.
Yeah, that's quite, I mean, this
This
on
is
the
not a
left.
gate. This is just to highlight the population.
Oh, okay.
This
I
is
thought
just to
one
highlight
gating.
here that you see here you are not able to resolve it. And this is here you are able to resolve the 2 populations. You see the contour plots is showing you 2 distinct populations there.
Oh, right.
So
Okay.
this red circle is just to highlight here on the plot. This is not the gating.
Oh, okay.
Yeah,
Thank you.
got it. Okay, good question. Thanks for the question.
Thank you very much. Is there any question more? I think it's enough. Thank you very much for Dr. Parichain for your lecture. Giving applause to Dr. Paras.
Ladies and gentlemen, let us continue our session. For the next agenda, let us start the 3rd lecture that delivered by Professor Paul Willis, PhD. To Professor Paul Willis, PhD, you have our full attention, which PhD got from the Medical College from Pennsylvania in 1993, and Master of from Idaho State University in 1979. Past president of the International Society for the Advancement of Cytometry and past counselor of the International Clinic Cytometry Society. Recipient of their 2028 Walsh H. Coulter Award for Lifetime Achievement in Clinical Cytometry. To Professor Paul Willis, PhD, you have our full attention.
All
Thank
right.
you.
Well, thank you all very much. I really appreciate the invitation here. Paul, thank you very much for that. Leanna as well. And it really is a pleasure to be here. We got the day yesterday to spend a little time on the beach, and what a wonderful place it is. And the food, my gosh, you guys know how to eat and eat well. I'm gonna, we have to find my slides. I'm going to talk to you about it. We're continue on sort of in the introductory material here. I know tomorrow it gets really advanced. And so what I'm going to be talking about is really clinical analysis, setting up a clinical analysis in the lab and looking at some of the gating that we would use. And then I've got an example of a situation here. So it's an introduction to clinical flow cytometry, and we'll focus a little bit in on the gating of those populations. There we go. So the objectives are to use the advantages of using flow cytometry. We'll look at an example of CLL and AML. And talk a little bit about how it can be used in the diagnosis monitoring of different types of tumors. Yeah, maybe if I point it there. Okay. Oh, there we go. All right. So how do we use it? Well, really in the diagnosis, the classification, detection of residual disease, monitoring different types of treatments and potential therapies like for CD20 and rituximab. It's got a very good sensitivity, so we can do minimal residual disease and reproducible. At Roswell, where I formerly worked, we had 3 different types of samples. Ones that were considered routine, and they typically took us 2 or 3 days to report out. We had ASAPs, which we tried to get out in that same day, and lots of stats. Stats we did 24/7. So we didn't cover the lab at night, but if a stat came in, we would set it up, have somebody come in, we'd set it up and report it out really within 3 or 4 hours. And these are the types of testing that we did. A lot of different types of cell enumeration that included T cell subsets, CD34s, PNH was a big one. Here's our clinical workup, which was the majority of the work. And that included a lot of your routine leukemias, lymphomas, and multiple myeloma, as well as panels for NK cells, T lymphoproliferative disease. We did a lot of work on immune reconstitution post-transplant, and then we had assays for ALPS and what Paresh just showed you, an assay for T regulatory cells. What makes it really a great technique for the clinical flow cytometry lab is that it is rapid. You can use relatively small samples. It's sensitive, semi-quantitative, and we think of flow cytometry as being a quantitative science. But when you get down to, is it quantitative, semi-quantitative, qualitative? The distinction between quantitative and semi-quantitative is that for a quantitative assay, something where you need to have a standard curve. Like, you would need to know a defined amount of glucose or something like that. And in flow, we really don't have anything that's a defined amount. We may have a range for CD34s or a range for CD4 cells. So that's why we refer to it as being a semi-quantitative, or in the jargon of some of the people back in the States, a quasi-quantitative assay. I think we'll spend actually a lot of time talking about the fact that it's multi-parameter, can do lots of colors. It's visual, and in my mind, it's actually there's a reduced interpretation selectivity, in the sense that they're often, usually these samples are very easy to interpret, and many times, on where there was a question, is this acute leukemia? Is it myeloid or is it lymphoid? Flow can really give you a definitive answer. And of course, we can detect very, very small populations. The disadvantages are that you really need a fresh specimen. I know people use frozen, but in the clinical lab, you're really much better off with a fresh sample. Some diseases, such as Hodgkin's, can't be easily identified. We have to disaggregate the tissues. We have to clean up the bone marrow a little bit. So there's some issues like that. And as a consequence of that disaggregation, sometimes it really won't be representative of what was actually in the tissue. And that's probably best illustrated with multiple myeloma, where you lose a lot of the plasma cells. How do we do it? We typically stained up 100 microliters of blood. It could increase to 200 microliters if we were trying to do something where we're going to acquire like 1 million events for MRD. We place that in the tube, then we throw in our antibodies, and these antibodies would be fluorochrome conjugated. This is FITC, and those, of course, are attached to the antibody. I know Jasha just went over this. She talked a lot about the FITC and its fluorescence, and I'm sure everybody here remembers their quantum mechanics from school. But if you don't, then your light is going in, your laser light's going in, your electrons are being excited, and as they decay and go back to their steady state level or their ground level, fluorescence is given out, and that's what we measure in the cytometer. So we've got a whole bunch of antibodies in the tube. We incubate those, and they're going to bind to the cell. Then we would lyse. And I spent a lot of time on this, so I want you to see those red cells in that tube just disappearing. I hope you can appreciate that. Much too much time on my hands creating these things. And then we wash, and the ones that aren't attached to the cell are removed, and then we can analyze it on the flow cytometer. So that's our staining technique. Some people will lyse first, others will do like a bulk lyse, others will do a stain and then a lyse. To do the analysis, I would usually start off with a forward by side scatter gate. And it kind of depends on whether you grew up using BD equipment, in which case this would probably be your default, or Beckman-Coulter equipment, in which case this would be your default. But in either case, it's looking at the same thing. And here we can easily identify the lymphocytes. Down here, this is probably some unlysed red cells, and as we move out here, this might be some dead cells. Here, your granulocytes, and then in here would be your monocytes. It really doesn't matter whether you look at side scatter in a linear space or side scatter on a log space. Either is fine. And normally I'll start off with one of these. I'm more comfortable with this one, but we'll start off with one of these. And then we'll move into a 45 by side scatter gate. This is classically just about everything starts off with some sort of a 45 by, I would run it through a forward and side scatter and then into one of these, or vice versa. But in the 45 by side scatter, what you've got here, your lymphocytes here, you've got your monocytes in here, and you've got your granulocytes here. And again, it really doesn't matter whether we're looking at side scatter in a log space or in a linear space. These two are identical. These are probably some decaying cells down here. They're 45 negative, maybe some erythroids. Anybody know what these populations are here? or this one kind of in here. Well, these are the hematogones, different stages of B cell precursors. And over here, we've got some of the myeloblasts that are falling in there. You can also throw in a CD19 region or a CD3 region if you're looking in lymphoma, if you're looking for T cells or B cells. And in the example that I'm looking at next, we're looking at a CD19 gate. So when I started in this field, which was back in the 80s, everything was one color, and we were kind of left with this type of a histogram. We've got a CD5, say in FITC, and a CD19, say in FITC. And you see this, and you can certainly see that there's a large populations of B cells here. But what's going on with this one? I mean, where do we actually draw the line, and where do these B cells actually fall in here? With just one color, you can't tell. It's really, you can make inferences, and you can say, well, I think they're probably this is a large population, and here's a large population, but you really can't be sure. Although some outstanding work was done with just one color. So really, what you need is two color. And Presh talked about setting up and compensation and everything and all that. I want to tell you that when I moved from one color to two color, I didn't understand what was going on. Everything was on an angle. I mean, it just didn't make sense. And then we learned about compensation. So the compensation is really a bane of just about everybody's existence, and we'll spend a lot of time on that during the course. But here, this is, of course, properly compensated. And what we can now see using this two-color approach is that our CD19 cells look like they some are maybe negative for CD5, but a large number of them are dim for CD5. And then these, of course, would be our T cells. So this two-color approach allows us to really determine the phenotype of different abnormal populations. And that's probably, we actually might base our regions on this population here, but that's looking at the gates. So this is what that cell looks like under the microscope. It's a lymphocyte, clearly. It's got some sort of an odd-looking nucleus and more cytoplasm in some of them than you would expect in a normal lymphocyte. Here's a little bit more data on the population. We saw it's CD5 dim. It's got light chain, lambda light chain expression. There's some CD20 as well as some CD23. And so anybody doing clinical flow, this is probably enough information right there to give you an identification that this is going to be a CLL. It's easier when you've got 3 colors, when you've got 4 colors, when you've got 8 colors, than it certainly was when you had the one color. And right now, we are exploding in the number of fluorochromes that we have available to us. Paresh talked a little bit about this. Tomorrow, I'll talk about spectral cytometry, where currently there's a really good paper that was put out by Joanne Lenigan on doing 40 colors in one tube. Clinically, we're not quite there yet, but I have certainly seen, and we published on a clinical panel that was 27 colors. So we really are in a golden age, and it's amazing. It started out, it was 1 color, 2 color, we jumped to 4. Next thing we knew, we were doing 6 and 8. It's really been like a logarithmic growth in terms of the number of fluorochromes that are available to us and the possibilities. So here, this is this is a panel with a lot more colors, and we can see that this expresses CD45, fairly bright. And expresses CD19. It's actually pretty bright for 20, this particular one, but that's what it is. Expresses CD5. This one is fairly bright for lambda. It's negative for 79B, and it is co-expresses CD200 as well as CD23. So that's clearly is a CLL. If we go through the phenotype, those are the characteristic markers that are sort of distinguishing it with the 23 and the 5. And then you can follow an algorithm. This one was CD5 positive. It expressed CD23. In our case, it was 20 bright. but that really means that puts us right into this classification here as being a CLL or a small lymphocytic leukemia. Of course, it gets a little more complicated than that. If it's CD5 negative, then we can look at different combinations of FMC7 or CD20 and CD23. We can look at CD10, CD38, and we can define all those into whether it's a follicular cell, a diffuse large cell, a mantle cell, a Burkitt's, or even a marginal zone. So using this flow chart, we can basically come up with a diagnosis. Now we can also use expert systems. We can use artificial intelligence, and by creating banks of these different cases and looking how our panel falls into that bank. It can also do MRD. So here's a sample where we have only 1% of the cells available that are, there's a lot of normal B cells, but there's only 1% of the cells that express the CD20, the CD5. There's our CD19, and you can see this particular one. They really jump out at you: 43 positive, 81 negative, dim for lambda, or dim for lambda, negative for lambda. And then CD22 dim, negative for 79, and then they co-express CD200 and CD23. So what's nice about this, I don't know if it's on there or not, but this is, I guess not. That population was only 1% of the total cells, and we can go lower and lower. We'll talk about that some more tomorrow. Sorry, oops, there we go. So it turns out that doing MRD on a CLL is actually very informative. And you can see in this slide here that the ones that have a high number of cells, this is greater than 100, you can see these patients do very, very poorly, whereas those that have a low number of residual cells do very well. And they're actually, what's illustrated in this is there's actually maybe a slight improvement if you include rituximab in the treatment. So the treatment here was effective, and then monitoring it was particularly good doing the MRD. So in terms of making a clinical diagnosis by flow cytometry, it's really nice to have the case history, the symptoms, and understand what's going on with the patient. And that certainly is ideal, but it's not necessary. You can usually make a pretty good diagnosis just based on the flow by itself. What we would normally do is we would begin with a differential from the report. We'd look at basically the CBC or the differential. We'd evaluate the fluorescence data and then mentally or physically compare it to normal. And then look for different patterns, holes in the histograms, different things that we recognize as being asynchronous antigen expression or different from normal, and then report it out and develop around that an interpretation. So we've got a case here with a 68-year-old male who comes in complaining of fatigue. Significant bruising is evident. It's actually got a normal white count, but the platelets are low. And this patient came into Roswell for a workup. Here's the report off of an automated CBC, and you can see here we've got 88% blasts that were done manually. By flow, what we had was a granulocytopenia, so a low number of granulocytes. We had lymphocytes were low, but not unreasonably low. And we had a large number, 71% of CD45 dim of a dim population. Looking at the report a little bit more, and honestly, I was often criticized for putting too many numbers on my report. I like looking at numbers, so my front page of my clinical report often had a lot of numbers. They really weren't necessary. All we really needed to give them was the number of blasts on the report. In this case, it's CD34. And in terms of the gated, that was 72% of the cells. But I like looking at the numbers, and what you can see here is it's an elevated CD4 ratio. Kappa lambda ratios look very good, whether you're looking at the CD5 negatives or the CD5 positives, but we clearly have an increased number of CD117 positive cells and CD34 positive cells. So I'm already thinking that this is some sort of a leukemia. Here's some flow data from the patient. There's our forward and side scatter gating. You can see down here, here's a CD45, and there's this green population that you can see in these first two tubes that is fairly evident and clearly abnormal. Dim for 45, and it's sort of sitting on top of or to the right of the lymphocytes. Another one that I often look at is this HLA-DR by CD45, and this is where our blasts would typically fall. Taking it another step, you can see it expresses a little bit of CD4. And then down here, we've got a CD13, so we're thinking myeloid, that co-expresses CD7. So I guess that's probably best shown here, but you can see the 13 and the CD7 population. You know, it's not unusual for myeloid cells to have some 7 on them, but it's really unusual to see that much 7 on this population. So this is triggering me to think that it's an AML, or I'm sure it's an AML. So here, just looking at the patient versus normal, you can see that the difference here between the two. It's fairly obvious. You know, I want to just come back to that 45 region and ask again, well, what's falling in that CD45 region? Here we've got a number of populations in here that are falling in this region that we would use. It doesn't matter how you look at it, it's always going to be the same. Whether you're looking at log or you're looking at linear, whether you're looking at it from top to bottom or left to right, you're going to get about the same thing. What falls in there? Well, yeah, the blasts, as I mentioned, the hematogones and the myeloblasts, but you have to be careful because also falling in that blast cell region are going to be, yes, the blasts and other myeloid progenitors, B cell hematogones, basophils, and plasmacytoid dendritic cells. So you need to be careful really about these two populations here because they can confuse you. So looking at this blast cell region on the CD45 by side scatter, what we've got is basically a population of myeloid blasts, which are pretty easy to identify versus CD34 and 117. We have the hematogones that are falling in here, and they are going to be 19 positive with variable amounts of 34, so sort of differentiating in this type of way, as well as the basophils. And here we're looking at DR by 123, and you can see the plasmacytoid DCs and the basophils right there. All that is falling in that region, and your challenge is to discern what your population actually is in that. So here, going back to our case, we've got, you know, clearly an abnormal population, the top hat here, that's falling on the forward by side scatter. What you can see here, is 13 positive. It's 34 positive in this case. It is also expressing quite a bit of 7, not as much as the normal T cells, but these CD34 positive cells are co-expressing CD7, not something you would see in an AML. Here's looking at that actually looking at another combination where we're looking at 11B and 13 and 33 and 34. Again, you can see they're 34 positive, negative for 11B, whereas a normal not blasts, but normal granulocytes and monocytes will express 11B and 13 as well as 33. Looking at that a little bit closer, this is really one of my favorite combinations, and just how informative analyzing this type of data can be. The 13 really is an unusual marker. Basically, the myeloblasts are going to start off as being 34 positive and not really express anything. Then they're going to pick up their expression of CD13. They're going to move on. So here's our 13 coming up. The colors are all the same in each of the plots. And then what happens to the CD13, you begin to pick up 11B as they mature, but you lose the CD13. So you have this very, very characteristic expression, really what would be considered like a left shift, where the 13 comes up, then it's lost, and then it comes up again as they mature into the more mature granulocytes and monocytes. And it's this pattern that is the same in me, it's the same in everybody in this room, that often you can use to differentiate normal from abnormal. So while I like to look at abnormal antigen expression, like we were seeing the CD7 expression on these cells, what's also very informative to me is actually looking at what we refer to as asynchronous antigen expression, or the expression of antigens on cells that you really wouldn't expect to see them. An example of that would be really with the 11B. So if all of a sudden we start seeing a lot of CD34 positive cells that are also positive for 11B, then we know that they're abnormal, because we wouldn't expect to see CD34 co-expressing with 11B. So going back to our patient, clearly, we've got something abnormal going on here with the 117 and 13. We've got that population here, whereas our normal, we wouldn't see the co-expression of the mature 13 marker on the CD117 positive cells. All right, so this is an example of a case where we've got our forward scatter. You can sort of follow that over time, starting off in 2015 in January and moving out to October of 2015. And this patient underwent a transplant. And initially, we actually saw where the blast cells came back. And then the patient was treated additionally, and we lost him. So really, one of the major applications is just to follow how well that particular treatment plan is going to determine whether or not you need to have additional treatment. So to summarize this brief introduction into clinical flow cytometry, we talked about the applications that we use at Roswell. We talked about the gating strategies, including forward and side scatter, as well as the CD45. We talked about the fact that it is really a powerful technique to basically identify your abnormal population, whether it be a lymphoma or a leukemia or a multiple myeloma. We talked about the power of increasing from one marker out to 8, 10, 16 probably is the sort of limit now in the clinical marker. The major components that we think about are the fluidics, the lasers, the optics, and the computer. The major applications are going to be the diagnosis, prognosis, and detection of residual disease. Where we really need help, I think, as a community, is in the standardization of flow cytometry. We tend to everybody has a different panel. Paresh gave the example of the guy who goes in the refrigerator and pulls out whatever antibodies are in the refrigerator and sets up a lousy panel. I think we all suffer from the fact that we do not follow a standardized methodology, a standardized set of panels that would allow us to be more convincing to the clinicians about where we're going. So we still have a long way to go with that, but wow, and we'll talk about this more. The future is really bright. We've got more colors than we know what to do with. Sony has now built an instrument that is capable, in theory at least, we don't have the fluorochromes, but is capable in theory of displaying over 100 different fluorochromes simultaneously in a single tube. Good luck adding that to the tube, but that's what it's capable of. So with that, I'd like to acknowledge all the people at Roswell Park, who over the years really made the clinical lab happen and made everything happen, particularly Kat Young and Joe Tario. He's the current supervisor. Jan was our past supervisor. Ed, we were lucky to have Ed. He just kept everything going. And I know that this American football is not something that you guys are very interested in, but Roswell's in a place called a snow belt, and they're all playing playing football here. And I don't know if you can make it out, but these are people. These are the stands. This is the way we play football in America. In a complete whiteout in the snow. So thank you very much. And if there's time, I'd be happy to take some questions.
Thank you to Professor Paul Willis, PhD, for the great explanation about getting strategy and data analysis about clinical leukemia and lymphoma. We will now begin question and answer session. To all the participants, please raise your hand and mention your name before proceeding your question. Thank you. Is there any question? Yeah.
Thank you very much, Dr. Paul. I'm Miswar from Prodia Clinical Laboratory. I have question related to data analysis. We know for leukemia phenotyping, some like for Euroflow, they have a library to doing analysis. So how important library for that to doing analysis? It is possible we have our own library to make the secondary analysis to make more precision, so to minimize variation between operator. Thank you.
So I understand the question correctly. Euroflow has a library that they create using the Euroflow panel. And are there other options out there for people that don't use the Euroflow for sort of creating that type of library? And honestly, what I was thinking about when I made that remark was the Euroflow system. I mean, they've really taken that to a higher level. I know other vendors are working on the similar types of platforms. I don't think anything is really commercially available. But what I had was the old-fashioned way, where I had a ring binder. And I had the cases in the ring binder, and this was these were like 3 or 4 examples of normal, 3 or 4 examples of a CLL, and I would, in particular when I was learning this, that's really what I used, was just to sort of create that ring binder. The truth is that a lot of companies are now working on artificial intelligence to really address this type of thing. So there are large studies that are ongoing. One of the companies is one called DeepCyto, and I know they're publishing in Cytometry B about creating a artificial intelligence, essentially using a library that would then say, okay, with a 99% probability, this is an AML with monocytic differentiation, or this is a diffuse large cell lymphoma. So we're headed that way, but what you know of and what I know is the Euroflow example is really the best done that. The reason this has been so hard is because of my comment about the standardization, because your panel is probably different than my panel. And so to create a library at that a vendor would create for everybody to use is really difficult, because you have to have some way of crossing over between those. So good question, and I thank you very much for that.
Okay, thank you very much. Another question related to the standardizations, because this is very interesting. We know a lot of variation between the laboratory. They have their own panels. The difference, any initiative to doing standardization for each disease for in flow cytometry, are ISAC doing for that or not? Thank you.
It probably wouldn't fall to ISAC. It would probably fall to an organization like the International Clinical Cytometry Society, and they have standardized PNH. They've standardized multiple myeloma MRD, and I'll talk about this tomorrow. But within the multiple myeloma, we standardized a panel. There's 2 tubes, 8 colors, so it could be run on the Canto, but also on at that time the Navios. And within a year, somebody else published on a panel that was 10 colors in 1 tube. And while we had thought about it and thought very hardly about, do we want to just make this 1 tube, the decision was to go with tubes, so it could be more universal. It's just like herding cats. I mean, everybody has a different idea of what is best. I think Paresh is right. If you follow the guidelines, in terms of, well, if you follow the guidelines, you're pretty much going to come up with the same panel design, depending on the instrumentation that you're using. But no, it really, there's hope, I think, in the future for standardization, but we're not there yet. I understand the state's system pretty well, and Joe Biden, the president, has just ordered, mandated that everything be, that we share our data and we move along that line. The FDA is in the US is looking at saying that everybody must validate their assay. So, and what that means is you have to validate it, but you have to submit it to the FDA. So if you were offering a test in the US from your lab, you would have to submit all of your validation data to the FDA, and then they would approve it. That's going to be a lot of work. It's going to be a lot of work for the FDA. It's going to be a lot of work for any lab that wants to meet that criteria. And this isn't just for flow. This is for everything. So there's a lot of discussion within Congress and within the FDA about this. What I think that will end up doing is making everybody say, I don't want to go through that hassle. I am going to go with an approved assay, something that has already been approved, and that process will standardize it. It'll make a lot of people mad. It will probably also limit the ability for people like academic labs to investigate new areas. But I think that's really where it's going. Thank you. You got great questions. Appreciate it.
Is there any other question? Maybe? Yeah.
Thank you, Dr. Powell, for the explanation. I want to ask, according to your experience, how many gating events that should we use or we run for MRD detection for acute lymphoblastic leukemia using peripheral blood? And what should we do if we have very limited sample?
Right.
Thank you.
That's a good question. It's a tough question. There are standards along that area, and what you really are looking for is a minimum of 500,000 events in order to get the sensitivities that you want. Ideally, you would collect 1 million events. For multiple myeloma, it's 5 million events is what people recommend, and 2 million being the minimum. Really what it comes down to, and I'll go over this tomorrow, but what it really comes down to is what level of sensitivity you want to have. How do I want to make this sensitive at 1%, 0.1%, 0.001%, or 1 in a million? What is the sensitivity that you want to have? And what you want to be able to do is to analyze and acquire really a clustered population. So a population that are all related to each other, of a minimum of 20 events. Now you may be really good and you may say, I only need 10, and okay, I'll accept that. 10 may be enough. And somebody else, I may say, no, I'm really insecure. I need to have 30 events, but 20 is the number that we typically aim for. So what that means is you need 20 events, and if you want a sensitivity of 1 in 100,000, then you're going to need to collect that 2 million events. The other number that's a classic number is 50. You need to, if you want to be really quantitative about or semi-quantitative about it, then in order to get semi-quantitative, you want to be around 50 events. So when it comes to the second part of your question, well, three questions there, but the second part of your question would be, what do you do if you don't acquire enough events? Then you report it out. You say, we strive to get a sensitivity of 1 in a million or 1 in 100,000 or 1 in 100, but we were only able to achieve a sensitivity of 1%. So we tried, but this is the sensitivity of the assay. So you can report out what your actual sensitivity was. Again, knowing that you want to have 20 events and the actual number of events that you acquired. So it's just a straight division of the 20 divided by the total number of events. And then if it's a paucicellular sample, if it's a low cellularity, then what we would do is to use 200 microliters as opposed to 100 microliters of blood. Or, if it's really, really important, you can stand it up in multiple tubes and combine the tubes, or you can do that bulk lyse. A lot of like the Euroflow group, they will do a bulk lyse up front, and then they'll count and make sure that they have enough events in the tube. So those are the approaches that people take, but it's a question that haunts everybody. Thank you.
Maybe there's another one question. Participant.
Thank you, sir. The limitation for flowcytometry method is fresh sample needed. How long to prepare sample can running by flowcytometry, and how to solution this problem? Thank you.
I'm sorry, the question was how long to collect a sample, or
stability of sample? Thank you.
the viability of the sample? I'm sorry, I'm having a hard time understanding. Oh, sample stability. Thank you. Yeah, the bottom line is that every lab should establish their own sample stability. And it really comes down to what you're collecting it in and what the sample is. So if it is say a blood or a bone marrow that's collected in EDTA, the published stability of that is 48 hours. So in an EDTA sample, 48 hours is the maximum stability of that sample. If it is say peripheral blood and or bone marrow and it's collected in heparin, that gives you another day. That gives you 72 hours. So you can go out to 72 hours. And that's what's been published. And those were, we actually dropped it down a day. EDTA, we would say it was good for a day, and heparin was good for 2 days, 48 hours. But people do go longer. And you can do things like put some media in there, some RPMI in the sample to go longer. So those were the guidelines that I tended to use for tissues, for a lymph node or something like that. We really would try to get those done within about 6 hours. Again, it could be longer, but we tried to do those relatively fast, at least to get it into the single cell state. And then we would keep it in RPMI, and we could keep it there for 24 hours to the next day. But the initial processing, we like to get that disaggregated in one day. A spinal fluid is completely different. Those samples are stable for minutes. I mean, they start to deteriorate right away, and really the faster you can get that stain, the better. We set that at 4 hours, but Mary Alice and Bruce Gregg have published that if you take your spinal fluid and you put it in the media without fetal calf, just in the regular media, then it's stable for a much longer period of time. There are fixatives out there. There's like Transfix and Streck makes a fixative that you can put the sample in that work well. They tend to increase your background fluorescence and decrease your actual fluorescence, but they work well. They work well also. It also depends on your temperature. So some of the, imagine here, but certainly in Africa, transporting samples is a real problem, just because of the heat. That sample's stored at 23 degrees is completely different than a sample that's stored at 30 degrees, as it transfers to the lab. So stability is something that you would want to establish, but guidelines, 48 hours for an EDTA and 72 hours for a heparin. Okay.
Thank you.
Do you have a particular sample type in mind?
Due to limitation of time, we can close the question and answer session. So thank you to Professor Paul Willis, PhD. It's such an honor for us.
Thank you very much.
Thank you very much.
I can't tell you, I'm sure I speak for all the speakers, how wonderful it is to get the questions. That really, you guys are really good with the questions. You should be proud. Thank you.
Thank you, Professor.
Finally, the fourth and fifth lecture will be delivered by Dr. Paul Hutchinson, PhD. Dr. Paul Hutchinson, PhD, have a lot of experience in flow cytometry, completed Master of Science degree from Monash University on microphage function, and got PhD degree from Monash University on the use of flow cytometry to quantify the level of immune function in renal transplant recipients. Since 2009, Dr. Paul Hutchinson, PhD, has been in charge of the core flow cytometry facility at the immunology program of the National University. To Dr. Paul Hutchinson, PhD, you have our full attention.
Okay. All right. Thanks to the organizers for inviting me and the other speakers. It's always good fun and a pleasure to come to Indonesia to give a talk. So I've got two talks, one on extracellular vesicles and then one on about flow cytometry and infectious diseases. So a bit more of a research focus, but some of these assays might be coming into clinical use in the future, hopefully the near future. So I think we're going to start with the extracellular vesicles first, okay? So let's load it up. Yep, that's the one. Okay. All right, if we just move on to the next one. Yes. So I'm from Singapore. I work at the NUS there in the Life Sciences Institute. I run the core flow lab. And we're going to be talking about extracellular vesicles. Okay. All right. So is that going backwards? Okay. No? Yes, here we go. So what are extracellular vesicles or exosomes or microparticles? So a cell can secrete a small particle enclosed in a phospholipid membrane, and these can be released from the surface membrane, or they can come from the in-cell, from intracellular organelles. So they're a phospholipid membrane, and sometimes within them, they can have bioactive molecules such as DNA, microRNA, and different proteins. And particularly if they come from the surface, they'll also express the specific markers from the parental cells, so you can identify where this microvesicle come from. Okay. So what are they? They are small, and we are talking sometimes down to less than 50 nanometers. And because they're small, they're tricky to measure. They can be found in any solution that cells are in contact with. So that can be plasma, bronchoalveolar lavage fluid, cell culture medium, etc. The fact that they contain bioactive molecules indicates that they could have and very likely have important physiological functions. Studies have found that they are upregulated in a number of diseases. Whoops, sorry. Just let me get right. In diseases such as thrombosis and some cancers, etc. And that they're also perhaps got a role in some hemostatic processes too. It's thought that higher levels of these microparticles can be indicative of cellular stress and also related to disease severity. So because of all this, it's thought they may have potential clinical applications as biomarkers and what we can call a non-invasive liquid biopsy. Okay, moving on. Try again. go to the next slide, please. Okay, how do we measure them? Well, electron microscopy. It's probably the gold standard. Electron microscopy is very good at looking at small things, and you can actually visualize and localize the microparticles. Atomic force microscopy, that's another way you can look at very small things. But both of these, not very standard in the clinical lab. Electrical impedance, cooled to volume measurements can be useful. Bulk methods, where you first purify and enrich, and then you can do an ELISA or Western blot, or actually beads to measure the microparticles. And then there's flow cytometry. So flow cytometry is good for single cell analysis, but can it accurately and reproducibly be applied to microparticle measurements? Because these things are very small. They're in the debris that you normally gate out when you do your analysis. So we know that flow can be defined as measuring individual cells or particles as they flow past a series of detectors. We can measure light and fluorescence from these particles, and then this is just a quick example of it. You've got your fluidic system, your lasers to excite and for light scatter, fluorescence. Pick up the fluorescence and the scatter with your optics, measure the different fluorescences and light scatter, and then display it on your computer. So that's the basics. You've heard all about that. Hydrodynamic focusing, Zosha went through this, but basically we can focus the particles or cells so they go through the laser one at a time. And as they go through the laser, you'll get a pulse measured in your detector. So here comes your cell, enters the laser. The voltage output will be from either the scatter detector or your fluorescence detector, and the size of the pulse proportional to the size of the signal from that cell, either the light scatter signal or the fluorescence. And pulse size related to the size of the signal. So a small particle, for the scatter, for instance, will have a small pulse. Larger cells, larger particles will have a large pulse. Now, an important part of this and the measurement is the event triggering or the thresholding that Zosha spoke earlier about as well. But just to go through it again and give you some examples. So the flow cytometer has a threshold or trigger level set on a particular parameter. It could be forward scatter. You could also use fluorescence. And it will only make a measurement on a particle which has a signal above that threshold. So you can see here, a big pulse above the threshold. We do this because we want to eliminate noise or background from our measurements, but you have to be careful because if you increase the threshold too high, you can exclude things that you want to measure. Which parameter we use for the triggering threshold and at what level depends on what you want to measure. So if you want to look at all particles, you look at the light scatter because all particles will have a light scatter. But if you only want to look at particles that are positive for a particular fluorescence, whether that could be Hirsch staining of the DNA or in microparticles, some sort of stain on the phospholipid membrane, you can trigger off that fluorescence. So as microparticles are small, it's very important to set the correct trigger threshold to maximize the discrimination from any noise. So if your threshold is too high, you might actually miss the pulse of the microparticle. You have to bring it down. But the problem then is you might also get the noise coming through. So with flow cytometry, we can measure the microparticles with light scatter. So remember, the scattered light is at the same wavelength as the incident laser. It's not exactly cell size. It's related to the illumination wavelength, the angle of collection, the refractive index of the particle, and its cross-sectional area. So different particles of the same size won't necessarily have the same light scatter signal. Differences also in the angle of light scatter collection and the size of the laser beam and power means that some particle measured on different instruments will also have different scatter signals. Now, normally for intact cells, it's not much of a difference. But again, once you start looking at small things like microparticles, things start to change. This is just an example of looking at, on the top here, we have the signal from a fax caliber, and on the bottom, the EPIX system from Beckman. And these were beads of a particular size, 3, 4, 5, 6, 7, 8. And you can see that on the caliber, the 4 micron had a lower signal than the 3 micron, while it was the opposite for the XL. So that doesn't mean the caliber's measuring the scatter wrong. It's just that particular optical setup on that machine was different to the epics. So just highlighting the differences there. What about using fluorescence to identify your microparticles? Fluorescent light is at a longer wavelength than the incident laser, and we can use that to detect any fluorescent probes that you put on your particles. And things that we can measure on the microparticles include we can have fluorochrome tagged antibodies. So if you're interested in a particular antigen expressed on the microparticle, you could use a fluorochrome tagged antibody for that. Membrane phospholipid binding dyes, and an XM5, which binds phosphatidylserine, which is part of the cell membrane that can be exposed on some of these microparticles. So again, I emphasize these are small, so the signals from them are small. So it's important to know what is the minimum level of fluorescence your instrument can measure so you can measure the fluorescence on these small particles. So how low can we go with that fluorescence? Okay.
Pardon, doctor.
No, that's
I'm
okay.
sorry, we have any problem, few problem.
Okay.
Can I fix it for a moment?
Yes.
Thank you so much.
All right. That look good?
So, we're so sorry.
No, that's fine. We can work it out. Okay. So if you're looking at a microparticle from the surface of the cell, imagine it's budded off. It's sort of, the membrane is sort of peeled off from the surface of that cell. Let's say it's from a monocyte. So on a monocyte, we know that you have CD16, 11A, CD14 expressed. And on average, per square micron on the cell, this is the number of copies there is. But if you look at extracellular vesicles or microparticles, they're very small. So the amount of that particular antigen on a microparticle, it's going to be less than this. It could be 2 or 3 or 10 if you're lucky. So you've got to be sure that if you're going to label it with an antibody, that you've got enough fluorescence from that microparticle to measure, okay? Something like PE, it's a bright fluorochrome, or BV421. But even then, you know, you're only getting down to 10 or 20 molecules per particle. So if the antigen expression is below that, you're not going to pick it up. Okay, can we go to the next slide? Try again. Okay. All right. Another factor that you have to be aware of if you're doing microparticle measurement is what we call the swarm. So here's your sample stream that I showed you earlier. Now, normally a cell will take up just about the whole size of the laser beam. So one cell at a time is possible. But when you're looking at small particles like we're looking at for microparticles, often you can have multiple particles in the beam at the same time. So when you make your measurement, rather than measuring one particle, you're measuring the signal from all of the particles, okay? So you would see this signal, but it's actually not one microparticle, it's multiples of that. So, as I said, it's possible for multiple microparticles to be in a laser beam spot coincidentally. And if this happens, the flow cytometer can't distinguish the different particles, and a measurement made is a single value that is the summation of the signals from all the microparticles in the beam at that time. And that leads to an overestimation of the signal and an underestimation of the number of microparticles. So how can we get that? The trick is to dilute your sample. You need to dilute it enough so there's enough space between your sample, between the microparticles as they flow through the laser, so it's measuring only one at a time. And how can we do that, or what does it look like? So this is just an example when we looked at beads. So this is concentration of beads, increasing concentration of beads along this axis, and this is how many events we counted. Now, you would think you increase the concentration, your event rate should go up, and it does to a certain level. But once you reach this very high concentration, because the beads, multiple beads are going through the laser at the same time, your event rate will eventually level off, okay? And this is reflected in the forward scatter, because what happens when you've got that linear relationship between increasing concentration and event rate, we're measuring one bead at a time, the forward scatter will remain the same. But as soon as you start to measure multiple beads at the same time, your forward scatter will go up. Same with the fluorescence. So this is an example here. This is at a high concentration, and this is very high, 2.8 by 10 to the 9 per mil of beads. You can see it looks like they've got a fluorescence and a high forward scatter. But as you dilute the sample down, you can see there's a reduction in the forward scatter and the fluorescence. So you need to do this dilution of your sample until you see this linearity between your concentration and the event rate. That's what it looks like for beads. This is an example of exosomes. In this case, they're from mast cells that were cultured for 48 hours in EV depleted medium. And then we collected it after those 2 days, and they stained the EVs and measured them. So increasing concentration, you can see most of the time, increase the concentration, there's a linear relationship, but eventually you reach a point where there's saturation, okay, where you get that swarm effect. And again, at that point, the forward scatter goes up and the fluorescence goes up, and that's shown there. So that swarm effect is very important. Start with your exosome sample and then dilute it until you reach the point that the signal that you get that you're trying to measure, for instance, the fluorescence and the forward scatter, is the same with your reduction in dilution. So how can we standardize? And like all measurements in flow cytometry and other things, you do want to standardize them. So, you know, you compare your results from one lab to another, from one machine to another. And unfortunately, because it's a new field, there's lots of different methods. So we do need to standardize the methodology of measurement for extracellular vesicles. Not only the flow, we've got to standardize the sample preparation. To start, if you're interested in doing extravesical measurement, this is a very good paper to work with. This journal covers a lot of the issues. There's some very good people working on how can we standardize the reporting and the measurements of flow cytometry, microvesical quantitation. This is an example for antibody labeling, the different things you need to do. You've got to isolate the vesicles. You've got to label them with antibody. You could label them with a fluorescent membrane dye. The PKH dyes are very good for that. You can float them on a sucrose gradient to separate them out, look at different density fractions and so on. That all depends on which particular extracellular vesicles you're trying to measure. After that, you've got your sample. What about the flow cytometer? You've got to, you know, start up your flow, do your regular QC, check the laser alignment. Maybe you have to adjust a few things for nanoparticle analysis, microparticle analysis, run some small size calibration beads. So you've got a reference point in your forward and side scatter, your fluorescence, antibody detection, optimize that, and then acquire. So what should you trigger on? What should you threshold your measurement on? Should it be forward scatter? Should it be side scatter? Triggering on scatter means that you can detect the particles even if they don't have a fluorescent label. Choosing which scatter to trigger from depends on the resolution of your detectors. Best to choose the one which can detect the smallest particle above the background, and that is actually usually the side scatter rather than the forward scatter. However, it also means that any non-microparticle particle in your sample and in the sheath liquid that has a signal above your trigger level will be detected, and they can interfere in your measurement as well. So if you've got non-microparticles in there of 100, 200 nanometers, they're also going to be measured. You know, most instruments filter the sheath at 0.2 micron, but you might want to look at nanoparticles of 100 nanometer size or 0.1 micron. So best to filter your sheath at a lower level than what you want to measure. So if you want to measure nanoparticles 0.1 or 100 nanometers and above, filter the sheath liquid below 0.1 micrometers. If you want to trigger on fluorescence, triggering on fluorescence means you only measure particles that have a fluorescent signal above a certain level. This is a good way to avoid measuring those non-microparticle debris, because if you have a specific probe, it should just label your microparticles and you can measure their fluorescence and exclude those without it. However, you have to be sure that the fluorescent label you will use does tag the microparticles you're interested in and at a level, at a brightness that is above the trigger level. Cell membrane binding probes such as PKH, CSFE, they're fluorescent levels that are bright and that people have been using for this type of measurement. So this is just an example of triggering on fluorescence. So in this case, we set our threshold on the fluorescence. In this, they were looking at nano-sized bead vesicles by fluorescent thresholding. First, the threshold was down here, so you can see the noise and the beads here. So to get rid of the noise, raise the threshold and you eliminate that noise setting, but you can still see those 100 nanometer beads. If you're using microparticles, so this is looking at nano-sized vesicles released by LPS-stimulated mouse dendritic cells, labeled with PKH67, and then we can see forward scatter and the PKH dye. So we've got a clear fluorescent population, which corresponds to our microparticles. Sometimes you need more than one dye because maybe the particle's very small or it doesn't stain that well. Whoops. So in this case, they used a combination of CSFE and the PKH dye. So originally not stained. So if they're not stained, no fluorescence, triggering on fluorescence, you won't be able to measure them. CSFE, okay, you can see a population arising here with some fluorescence above the threshold level. Add in another dye, okay, now we can see more of that population. So some triggering from fluorescence issues though, as I said, are the microparticles you're interested in able to be labeled with those fluorochromes, and will the signal to be strong enough to be detected? I mean, these things you can only try by trial and error. Also, I mean, it's subjective where you cut off that level. You know, the difference between a 100 nanometer stained particle and a 90 nanometer stained particle. Maybe the 100 is above, the 90 is below, but you do have to draw the line somewhere. The thing there is to be consistent across your experiments. Okay, so now I'm going to just go through a couple of publications where they've done some microparticle measurements and just give you some example of the data that people are generating and some controls you can use as well. So this one was a paper published in PLOS One, and they were looking at the quantification of microparticles from different cellular lineages by flow and looking at the effect of secreted phospholipase A2 on microparticle assessment. So the secreted phospholipases A2 are the family of soluble proteins, and they comprise of enzymes that can destroy membrane phospholipids. So if you have more of these enzymes, maybe they're going to affect your microparticle measurements. So it can hydrolyze phosphatidylserine, a phospholipid frequently used to assess microparticles. And it may be that with this enzyme, it can even clear the microparticles. So they used what they called an improved flow method to measure microparticles and what happens when you add those enzymes to those microparticles and what effect that has on the quantification. Their improved flow method basically consisted of all the buffers they use, the washing solutions and so on, filter them through the 0.2 micron, not millimeter, pore size membrane. So it's the same as the sheath filter. If you're filtering your sheath through a 0.1, filter your buffers through a 0.1. They used Fax-Canto, and then rather than using a diode, which is of less sensitivity than a PMT, they put the PMT in to measure the forward scatter. They used a laser with higher laser power, and the forward scatter blocker bar, which is normally used to block the light scattered from the laser, was at a higher thickness. So that meant that the forward scatter was at a higher angle. The higher angle, the more sensitive the scatter measurement. They used fluorescence as a trigger signal, and the positive fluorescence events were displayed in the side scatter, forward scatter graph. they used microspheres of varying diameters to set their gate of detection. So it's reference particles. They used some standardized beads. And the microparticles were acquired at a low speed, 10 microliters per minute. So basically meant they run it on low and done various things with the setting up of the antibody labeling and so on. So microparticles described as ranging from 100 to 1,000 nanometers, expressing the phosphatidylserine and also exposing surface markers originating from their cellular origin. So they hope to do this, identify their microparticles using an XM5, which binds specifically to phosphatidylserine, and then specific antibodies against surface markers coupled to fluorochromes. They also labeled their microparticles with another cell tracker dye, CMFD. It passed, but this one doesn't stick to the membrane. It goes inside the membrane and then becomes a fluorescent marker. They actually stain the cells with this dye and then stimulate the cells to release the microparticles. So the idea is there's fluorescence within the cell. When the microparticles release, it's going to take a bit of that fluorescence with it. So first, this is some data here where they show forward scatter versus side scatter, and this is their reference beads from 100 nanometers up to 3,200. So they would use those reference beads to set their gate on the forward scatter. Okay, so gating here, that's how they defined their microparticle gate. You know, it's a subjective place, but at least they've got some beads, some standards that they can reliably put from one experiment to the other. They were looking at human platelet microparticles. So this gate was to the left, below where the intact platelets were. They also included a counting bead gate because they wanted to quantitate how many microparticles, what the concentration was per sample. Whoops. I don't know why they used height instead of area for their measurement. I would have thought area is better, but they did use height. So to confirm that the microparticles contained were genuine microparticles and not protein aggregates, what they did was they would split the sample in half. And one half of the sample they treat with Tridon X detergent, which would solubilize or dissolve the membranes. So it's a way of giving you a negative control. So if there's any microparticles in your sample, you treat it with Tridon, it's going to break them down. You shouldn't see any staining. And the other way that they tried to do a sort of negative control for the staining, an XM5 binds to phosphatidylserine in a calcium-dependent manner. So if you put the NXM5 with the microparticles in a media that chelates the calcium, or you add EDTA, it should block the NXM5 binding. So it's a negative control for the staining. So platelet microparticles detected using fluorescent conjugated NXM5, and they also used anti-CD41 antibodies. This is the control, the untreated microparticle samples here, forward scatter, side scatter. So that's that gate they set. The intact platelets would be here, microparticles here. Gate from there, you can see, yes, there's some CD41 staining. There's some NXM5 staining. They defined their microparticles as being double positive for both those markers. And here, they gated on them. Actually, no, it's anything 41 positive, and they were able to quantitate that using the counting beads. What happens with the Tridon? You can see they're much smaller. The debris is there because basically it's dissolved the microparticles. No NXM5 and no CD41 staining. And if you gate on whatever was there, and you know the data display is a little bit not so clear, but you would just have bits of debris there. So it's a very good control of your microparticle sample, the Tridon. EDTA, that just blocks the NXM5. So you can see the microparticles are still there, but they're negative for an XM5. There's still that CD41 population. For their swarm controls, they did something that was quite smart. They did the dilution, but also to confirm it, they would add beads to the sample. And those beads would be of a particular fluorescence, similar to, you know, or actually the beads would be coming through with the microparticles. So if there was a swarm effect, if the beads were there with the microparticles, you would expect a lot of double positives, but that wasn't the case. So that's another way you can quantify a swarm effect. Put in something which shouldn't be positive for the same marker as the microparticles. And if they look double positive, your sample's too concentrated. Dilute it down until they're negative for the microparticle marker. And that's just showing how when they diluted it down, that double positive reduced. Okay, and then the results they found, they looked at human platelet microparticles, standardized their measurements along the lines of what I mentioned, and then they were looking at the numbers of CMFDA microparticles and the effects of the different phospholipase on those markers there. So microparticle measurement by their improved method seemed to work. Good control data on the effects of the swarm, as well as reverse negative controls. So sample was positive. How can you knock out that positive staining to be sure it's a specific staining? And they did find that some of these phospholipase can decrease specific markers, as well as reduce the annexin 5 binding. Lastly, let's talk about attempts to standardize this measurement across labs. So this is a paper where they did a study on that, attempt to standardize. The aim was to have a calibration bead mixture and also platelet EV samples that they sent out to 33 different labs. Beads and a light scattering model were used to set a standard extracellular vesicle diameter gate across all instruments in order to measure the concentration of these CD61PE positive platelet microparticles. So this is a bit of a QAP sample, if you like, for microparticle measurement. I won't go into this too much, but basically they had what they called the Rosetta calibration system. Consists of software and a mixture of 7 types of polystyrene beads with traceable mean diameters between 100 to 1,000. This bead mixture is analyzed in the flowcytometers with the settings for that's normally used for EV analysis. And then they put that on a standalone software package that would try and standardize the light scatter across all the different instruments. This is just to show something interesting here. So this is the diameter of the particle, and this is the scattering intensity. 3 different instruments, forward scatter on one and side scatter on the other 2. The black line is the bead measurements. So a particle here of about 0.2 micron or 200 nanometers up here would have a scattering intensity here. And as you'd expect, increasing size, increasing scatter intensity. This is the EV model size. So you can see the signal from a bead this small and the signal from an EV of this size was actually the same. So what that indicates is, even though this bead might have been, let's say, 200 nanometers, the signal from the EV bead was of the EV particle was the same, but it's actually larger. That's because they consist of different materials. They have different scatter, different refractive index. That was on one machine, and you can see the differences on the others, okay? Small particle, small bead, same scatter from an EV. So when it says, you know, the bead is 100 nanometers, doesn't mean that the EV is 100 nanometers. Use them as a reference, not a quantitation tool. Whoops. So they looked at the particle concentration, and they had these frozen aliquots of platelet EVs, and then they sent them to each labs along with the antibodies to do the staining, and to do the staining following a standard protocol. Sent it out to 33 labs, but only 23 out of the 64 flows detected could do the EV measurement using this particular protocol. Had to be able to measure a minimum size of that bead. But of those 23 labs, what were the results? So how did it work? Sorry. First, the beads were run to define size gates for the EV measurement, and then stained EVs were then measured and the concentration of samples within the gates calculated. So this is the side scatter used to define the gates. This is the 1200 to 3000 nanometer EVs. This is the 400 to 800 nanometer bead gate there, because remember the bead size is smaller than the extracellular vesicles, and then anything below that would be here. Okay, so they would set their gates and then try and quantitate what come up with the CD61 staining. So how did the different results compare across the different labs? This is divided up into different machines. These are the individual results. So, okay, you can see some groups were able to measure a high concentration, others not so great. Or maybe these are the correct measurements and these are wrong, because this is where most people measured it. That was for the larger extracellular vesicles. Only a few labs were able to get results for the smaller particles. So just to show you that it's tricky, yes. If you're interested in doing it, you got to spend a bit of time and work on your methods and to get things as accurate and as sensitive as possible. So, in conclusion, it's becoming apparent microparticles do play an important role in normal and disease physiology. In principle, flow does seem a technology you could use to identify and quantitate these particles. But because of their small size, you know, they're at the limits of detection for most instruments. You're right where the debris is. So normally we ignore debris. Microparticles, you want to measure the debris. So we need to have standardized preparation and measurement protocols so that you can have accurate and reproducible results. Okay, so that's the first talk, and I'm happy to take some questions on that.
Thank you very much for Dr. Paul Hutchinson, PhD, for the first lecture about extracellular vesicle analysis. We may begin our question and answer session. You may raise your hand.
Thank you. Good morning, Professor. I'm Dr. Fitri from Dr. Stomo General Academic Hospital. My question is this microparticle specific for particular diseases or maybe for particular diagnosis, or maybe is there any particular microparticle for a specific disease? Thank you.
There may be particular microparticles associated with particular disease or increased numbers of microparticles associated with particular disease. I think at the moment, particularly for flow cytometry measurement, we're still at the point of trying to clearly define the measurement, to standardize the measurement before you could really take that as a clinical test. You know, there's a lot of work still need to be done there. And actually, there's newer technologies, newer machines that are coming up, which are more sensitive to measure the microparticles. I think flow cytometry might still have a place for it, but I'm sort of leaning to the view that there's more specialized machines, which might be better at the microparticle measurement, which will hopefully make it more standardized. It's one of those things, you know, there's indications that yes, this might be useful, but I think there's a lot more work that needs to be done to use it as a clinical test.
Thank you very much, Doctor. Is there any question? You may raise your hand. Thank you.
Yes, thank you very much. A very interesting topic. We have experience to doing measure exosome base, but not yet using flow cytometry because we know that's not easy to measure with flow cytometry base for exosomes. Based on your presentations, to increase the sensitivity, we need to use the 9-degree or different degrees because some flow cytometry they already standardized the degree. It is all flow cytometry can be set up for the degree of the first the degree of the scatter. So we can increase the sensitivity or not, or it is, and the second question is related to, it is possible we can separate the exosome or microparticle using flow cytometry. Thank you.
Okay, so I think that you're talking about the scatter measurements being a bit more sensitive and the side scatter in particular. I think in general, yes, side scatter does seem more sensitive than forward scatter in picking up the small particles. And usually whenever we've tried to measure small particles, if we're like looking at small bacteria and things like that, we always look at the side scatter and we either trigger off the forward scatter or the side scatter, and they see which one gives us the clearest measurement of the population of interest. So I think if I was going to make a blanket statement, side scatter better than forward scatter. There are ways you can manipulate your forward scatter to make it a little bit better, but that means, you know, you have to get inside the machine and do a few things, whereas the side scatter is already there. The second part was how can we purify and isolate these particles? Now, I don't think we talk about self-sorting in this workshop, but if you can measure it by flow, you can sort it by flow on a self-sorter. But the question with sorting small particles is, what about the particles you miss? So you might think you've sorted exosome CD14 positive, so it's a monocyte exosome. And on your measurement, it's nice and clear. But there's going to be below your threshold, below your triggering level, other microparticles in there, very likely. And when you sort, they're going to be included as well because you didn't measure them. So that can interfere in your purity. So sorting, if you want to enrich, yes. If you want to purify, difficult. But another good way to purify and enrich your particles is, and it's another technique that can be useful, is to use beads. So you have a bead with a capture antibody on it, say against CD14 or an exon 5 conjugated to the bead. You add it to your sample, the microparticles will stick to the bead, and then you can counterstain, and then you have your microparticles on there. It's a way of quantitating them. Yes, you're not measuring them one by one, but it is a measurement of the amount in the sample. And you could also use it to purify those as well, or purify to a higher level than cell sorting. So I would look at that technology. If you want to isolate particular microparticles expressing a particular, you know, are they from the monocyte, are they from the platelet and so on, those beads, microbeads with the capture antibodies could be a good way to do that.
Thank you very much.
Thank you very much, Doctor. Is there any more question before we continue to next lecture? Okay, maybe this is the end of question and answer session. We may continue to another lecture. Doctor, thank you.
Okay. Go to the next one. Eventually, come on. Okay, so in this talk, get away from the little things. Let's talk about intact cells, which I feel a bit more comfortable with. I'm going to talk about the applications of flow cytometry in infection. And in particular, I'm going to talk about T cells and how we can measure them in infectious diseases and get some useful information from that. So the immune system, you know, it's responsive against infection can be split into two groups, the innate, the adaptive arms. The innate arm acts immediately, quickly responds to non-specific signals. So these are things like neutrophils, macrophages, complement. The adaptive immune response develops after the initial innate response, and it's more of a specific response directed against the organisms that actually cause the infection. And there's two main cell types in the adaptive arm. Oops, come on. The B cells that make the antibodies and the T cells that drive the cellular immunity. So T cells, and that's what I'm going to focus on in this talk. We have the CD4 and CD8. CD4 T cells are the helper cells, so-called because their main role appears to be that of helping or directing other immune cell responses in infection. CD4 T cells can release cytokines that will also enhance the innate immune response. Interferon gamma can activate the macrophage to directly target the infectious organisms. They also talk to the B cells. You know, they switch the antibody production from IgM to IgG by binding to the B cell. CD8 T cells are known as killer cells, cytotoxic T cells, because they can directly kill other cells, such as tumor cells or virally infected cells. And there's other subgroups of the T cells. There's the Tregs, there's a mate T cells, and so on. The more you look, the more of these different subsets you can find. All of these cells can be identified and have their function measured by flow cytometry. So what's the T cell response to infection? So remember, a T cell develops with a receptor specific for a particular antigen, particular peptide of a protein that's related to an infectious organism. When it encounters that antigen, either via infection or vaccination, it'll induce a proliferative response. So that's your naive T cells, binds its antigen, it'll proliferate, it'll differentiate. Once the antigen is cleared, they'll remain in the cells as the memory T cells. And these memory or antigen-specific T cells can drive the rapid recall immune response if that antigen is encountered again. Flow cytometry, it's an ideal technology to quantitate and characterize these memory antigen-specific T cells. And that in turn will give us an important insight into the status of the immune system and how it clears and controls the response to foreign pathogens. So how does your memory T cell respond to antigen? It will specifically bind it, and we can measure that with things like the MHC tetramers. It'll proliferate. So binds the antigen, that memory T cell will proliferate again, start dividing, so there's more of them to fight the infection. They'll produce cytokines, and we can measure that with ELISpot and intracellular cytokine staining. They'll upregulate new markers or activation markers, CD154, CD137, etc. And for the CD8s, there'll be some sort of degranulation. They'll start to release cytotoxic effectors. So how can we measure these different steps? I won't go through them all, but for proliferation, a good way to measure proliferation is to use a dye like CSFE or the violet, cell trace violet. These dyes get into the cell and stain it with a uniform brightness. So when that cell divides, those daughter cells will have half the amount of fluorescence of the parental cells. And by that, using that property, we can count how many times did they divide and what's the proportion of dividing cells. So we can stimulate your sample with your memory T cells and see if they proliferate after stimulation with the antigen of interest. So this is what it looks like. You start off with your staining, stimulate the cells to proliferate. That could be an antigen stimulus or some sort of polyclonal stimulus. And you can see these peaks are rising. This is 1 division. 2 division, 3, 4, 5, 6. Eventually they dilute out because they've divided so far. And as an example of using this, so this is looking at your CSFE or your CFSE, the cell division marker versus CD4, a healthy donor here, a patient with common variable immunodeficiency. Unstimulated, you can see they retain that parental peak of the CFSE, so there's no cell division. If you add strep pneumoniae or antigens of that, a protein preparation of that, you can see in the normal, there are some cells that recognize it and start to divide. Whereas in the CVID patient, very little of the cells responded to that, even though very likely they've been exposed to it. Same with Klebsiella. In the normal, there's strong proliferation from the T cells in the healthy subject, but not in the CVID patient. So this patient has an impaired immune response against these diseases. Was it some functional problem with the T cells? They don't proliferate properly? Well, no, because if we stimulated with a polyclonal stimulus, super antigen, we did see cell proliferation in the CVID patient. So it wasn't an issue with the actual T cell proliferation. It's something else causing the problem. Another way you can detect antigen-specific cells is their cytokines. T cells will rapidly, within hours, produce and secrete cytokines after stimulation by antigen. And we can measure these cytokines by flow by blocking the release of the cytokine. Use a Golgi transporter inhibitor, Menensin, brifaldin A. You can then do surface staining, followed by intracellular staining to measure which T cells are producing the cytokines following the antigen stimulus. This technique is known as intracellular cytokine staining. So first you start off with your T cells, you activate them, you stimulate them with your protein of your antigen of interest, but you also add an inhibitor of protein export so that when they make the cytokines in response, it's trapped within the cells. Give them a few hours culture or overnight, they'll start producing cytokine, but it'll be trapped within the cell. At the end of the culture period, surface stain, fix and permeabilize so they can stain your cytokines and then run it on the flow cytometer. So people use this to detect antigen-specific T cells, and the first paper was published about 25 years ago. In this paper, they used profeldin A to block the secretion of cytokines by antigen-active T cells and then stain them with the anti-cytokine antibody. So this is peripheral blood mononuclear cells stimulated with a control antigen, that is an antigen that shouldn't induce a response. CD69 was used as a generic activation marker. This is no staining for the cytokine, gamma interferon, IL-2, and TNF. So you see there's few scattered dots, that's your background. But if you stimulated with antigens derived from the cytomegalovirus, and this particular subject was infected with that virus, so they had memory T cells, you could see that there's this population of T cells, CD4 T cells, making interferon gamma, making IL-2, TNF. So you could quantitate the amount of memory antigen-specific T cells against CMV using this technique. This technique was later adapted to use in a whole blood assay. So rather than isolating the cells, you can just take the blood, heparin blood, add the antigen, add your profeldin, culture, and then do your staining, which is a lot easier than isolating the mononuclear cells. You could also look at activation marker expression. So T cells responding to antigen will have induced expression of new cell surface proteins, activation markers. These markers include things like CD134, also known as OX40, CD137, and CD154, also known as the ligand for CD40. So in these assays, incubate the T cells with antigen. The time of culture depends on the activation marker of interest. CD154 goes up quickly, CD134 takes longer. So 6 to 48 hours, you should see upregulation of them all. At the end of that time point, stain the cells with the antibodies against your antigen-specific markers and then do your measurement. It's always quite useful to also combine them with some non-specific activation markers, CD25 and CD69. These antigen-specific cells are quite rare. They can be 1 in 100 CD4 T cells or 1 in 100,000. Looking for rare cells, always good to have more than one marker. The more markers to define that cell population, the more accurate your measurement. So this is just an example using CD154 staining to identify antigen-specific cells, different donors, and then stimulated with different antigens. So this is looking at Birch antigen. So this is looking at an allergic response. So you don't see, there's a little bit of IL-2. So this is the 154 expression. So those few dots there, it's about 0.02% of the population, but they're the T cells specific for the Birch antigen. And they're not positive for interferon gamma in this case because it's not a Th1 response. If you look at CMV though, you can see, yes, 154 positive, interferon gamma positive, no IL-10, no IL-4, but a bit of IL-2. So a Th1 response as compared to a Th2 response for the different types of antigen. Tetanus toxoid, the same, so your cytokines are 154, and then super antigen, which serves as a nice positive control for your assay, where you can see there's a lot of 154 in cytokine production. So, and you can also look at other active antigen-induced activation markers, such as CD137. So as I said, we're looking for rare cells. So you need to count a lot of cells to measure this. It's very similar to doing, measurable residual disease in leukemia, lymphoma. Rare cell measurement, you need to count a lot of cells to identify that rare cells. If you're going to do short-term stimulation to measure antigen-specific cells, say less than a day, best to use whole protein antigens for CD4 and peptide mixes for CD8, though you'll still see some CD4 with that peptide mix. How do I identify those rare cells? How can you determine, specific from non-specific staining, real antigen-specific cells from, background? So as I said, you need clear markers to identify those antigen-responding cells. Ideally, it should include both positive and negative markers. It's a good idea, I think, to combine that intracellular cytokine staining with the activation, CD154 or CD137, okay? So you've got two markers you can be sure identify the antigen-specific cell. You need correct controls. Isotype antibodies, probably not. Fluorescence -1, yes, depending on your staining panel. No antigen stimulation, definitely, because that gives you your background level of the cytokine staining of your activation markers. So always a no antigen stimulated sample as your control. How many cells should you count? As many as possible. Paul spoke about, needing 20 counts to identify that measurable residual disease leukemic cell. Very similar for antigen-specific cells. It's positive for 154, positive for interferon gamma. 20 counts is good, 100 counts is better, 1,000 is better. Count as many of those CD4 and CD8 cells as you can. So now I'm going to give you some examples in particular diseases, focusing a bit on tuberculosis, but we'll talk a little bit about COVID at the end as well. So actually, the commonly used tests in TB diagnosis are measurements of antigen-specific T-cell response. When you put a little bit of PPD in your skin, you're going to generate a delayed type hypersensitivity response, which is driven by your memory CD4 T-cells. Whoops. The quantiferon, interferon gamma release assays, they measure the antigen-specific T cells because they add to the blood the TB antigen. If that patient, that subject has been exposed to Mycobacterium tuberculosis, they're going to have memory T cells that'll release interferon gamma into the supernatant, but they don't quantitate those antigen-specific T cells directly like the flow assay does. So can we do that by flow? And the answer is yes. And it depends what you're trying to measure, but the antigens you can use is, if you want to use MTB-specific proteins, use ESAT6 and CFP10 like they do in the quantiferon assay. Or if you want to do more characterization of the response, you can actually use PPD, which is the same thing that they put into your skin for the skin test. Problem with PPD is it does cross-react with other Mycobacterium species, but it depends on what you're trying to measure in your study. So how do you do it? Stimulate your T cells with the TB antigens of interest. They can be whole proteins or peptide mixes of the protein, the protein chopped up into the peptides. You can then identify the memory T cells by staining for interferon gamma, but it's also good to look at other cytokines and specific activation markers. The advantage with doing it by flow is you're looking at the cells one by one. You're directly identifying those cells. You can measure, are they interferon gamma positive, but also are they making IL-2? Are they making TNF-alpha, GM-CSF? Do they express other markers which could help? What can we use this assay for? Can it detect prior infection exposure to MTB, much like the quantiferon assay? And the answer to that is yes. I'll show you some data for that. Can we use it to discriminate latent from active TB infection? May be possible. And again, I'll show you some data on that. Can it measure whether anti-MTB treatment is effective? Once you're on a TB treatment, it lasts many weeks. Is there a way we could use this assay to identify early in the treatment course if the treatment is working? And maybe we can. As I said, PPD test is used to determine if an individual has been exposed and infected with MTB. However, if you've been vaccinated with BCG, you're going to have a positive response as well, because there's a big crossover in the amount the proteins expressed by these two organisms. But there are some specific proteins which are only expressed by MTB and not BCG. and that's ESAT6, CFP10, and a few others. So if you stimulate with these proteins, they should be more specific at detecting the recall responses against MTB than the PPD skin test. And that's ESAT6 is one such protein, and we did an early study nearly 20 years ago now, where we used ICS to measure CD4 T cell responses against this antigen in patients suspected to have TB. And so we wanted to see, could it differentiate between TB infection and non-TB infection? For this study, we used the whole blood culture technique. So the whole blood, I like to do whole blood assays because I'm lazy. I don't want to, phycol the cells, wash them and so on. Whole blood is a lot easier. Take the blood, set the assay up. But it does mean you have to do it straight away. You can't put the blood and leave it till tomorrow. Has to be done, and the sooner the better after the blood draw, the more accurate it will be. It needs to be heparin blood. You can't use EDTA because that chelates the calcium, impairs the immune response. Has to be heparin. So we take the blood, we add co-stimulatory antibodies which amplify the response, and we set up and what we set up for this assay or this study, we had a no antigen negative control. So they got the co-stim antibodies, but they didn't get any antigen. So that was our background. Then we looked at our antigens of interest. We looked at PPD and we looked at ESAT6. Take the blood, incubate for an hour before we add the Brufaldin A. You need time for your antigen presenting cells, take up that antigen, process it, put it on the class 2, and then present it to the T cell. So 1 hour without the Brufaldin A, and then for the first study, we incubate for 5 hours. You can also make it go longer, give them 2 hours without the Brufaldin A, and then leave it overnight, about 16 hours. You can't go much longer than that because eventually the Brufaldin A will have a toxic effect on your cells. At the end of that culture period, lyse your red cells, do your surface antibody labeling, your CD3, your dump channel and whatever, then fix and perm and do your intracell staining of the cytokines. Also good to do your CD4 during the intracellular staining because there is some downregulation of the CD4 on the surface with the antigen stimulation. So include it in your intracellular staining. Same for the CD154 if you're using that as your activation marker. After that, you run it on the flow cytometer. So we looked at 3 groups of patients or subjects in our initial study. Patients who were adults with newly diagnosed active TB, either culture positive or a high likelihood of having TB, and were going to get anti-TB therapy. We looked at controls. So this was done in Melbourne, in Australia. We looked at those who also had pneumonia, but had confirmed alternative microbiological diagnosis or low clinical probability of tuberculosis. So it should be negative for that ESAT response. And then we had our normal controls. So we took the blood and we did the whole blood assay. So this shows you the interferon gamma response to the PPD. So in Australia, everyone's immunized to BCG, so we would expect a response from the normal. So this is a proportion of CD4 cells positive to interferon gamma after stimulation with PPD. So we can see there's a measurable response. The controls, not so great, but that's probably because they're sick with pneumonia. So maybe their immune system overall is a little bit, depressed. And then the TB cases, yes, we could clearly see that interferon gamma response. But what about to ESAT6? ESAT6, remember, specific for TB, not expressed by BCG. So our normals who had been immunized with BCG didn't respond to the ESAT6. We couldn't measure any positive or very few CD4s making interferon gamma response to the ESAT6. Same for the pneumonia patients, the non-TB pneumonia patients. But on the TB patients, yes, not in all of them, but in most of them, we could measure an interferon gamma response. We also looked at CD154, the activation marker, and again, we found a similar thing. Normals, controls, very low response to the TB-specific ESAT6. The TB cases, we could have a measurable CD154 response. So yes, we can use this assay to identify patients that have been exposed to MTB. So Studies have moved on from that. Now they're more interested in, can we use this measurement to identify specific signatures that can differentiate subjects with active or latent TB infection, and also possibly to predict the effect of anti-TB treatment? So what are the parameters people are looking at to do that? Is there particular cytokine combinations? Do the MTB specific T cells have a different phenotype? Do they have a different memory or activation marker expression between active and latent TB? There was an early paper 10 years ago, which sort of promised a lot. Just look over here. They looked at 3 different cytokines. And what they published was, if you had your memory T cell against PPD or ISAC6 was positive for TNF, but negative for IL-2 and interferon gamma, there seemed to be some differentiation between active and latent TB. However, further studies have found different. They didn't see the same effect. So if you look at this as your TNF only, and the red is active, the blue is latent, there is no difference. So why one study one way and one study the other? Well, it comes down to there's no standardization in this assay. So the bane of our life is standardization, particularly for rare cell measurement. And there's a lot of differences. One study used whole blood, one used PBMC. How long did they stimulate for? I think one was 6 hours, one was overnight. Did they use co-stimulatory antibodies? What about the antibody combinations? Okay, so those things can affect the staining that you see. But there are some other markers which seem to be a bit more stable from one study to the next, which are a bit more clearly expressed on the cells that we can use. So this was an early one where they looked at CD27, which is a memory cell marker. So it's a bit counterintuitive, but this is CD27 negative on your PPD specific CD4 T cells. So the higher the signal means there's more CD27 negatives. These are the active TB. These are the healthy donors, unexposed. These are highly exposed healthy donors. So these were nurses working in the TB clinic. And this is latent TB infection. So you can see that the active disease tended to have more CD27 negative on their TB specific T cells than the latent disease. So there's a separation, but it's not, 100%. So we did a study where we thought this is good, but why don't we combine the CD27 with the cytokine staining? Is there a combination of CD27 and cytokines that can help differentiate? So we did a study where we looked at samples from the Singapore Tuberculosis Control Unit for the evaluation of active TB, and for the latent population, we used their contacts. Combined cytokine measurement and activation markers and memory markers to determine if we could distinguish between these two groups MTB-specific CD4 T cells that could discriminate active from latent TB infection. We also tested another interesting MTB-specific antigen called heparin-binding hemagglutinin, which tends to be, if you have latent TB, you have a higher response against this antigen than inactive. So we wanted to measure that. Again, whole blood assay. I like the whole blood assays. So whole blood stimulation, 6 hours. We use PPD stimulation. We combined ESAT6 and CFP10 protein preparations. And we also had the HBHA, the latency associated antigen. End of the stimulation period, lysa RBCs, and then we did the staining for the cytokines. And we compared the results between those subjects with clinically defined active disease and those with latent infection, and the analysis was unblinded to those results. So this is gated on the CD4s and CD4 T cells. This is no antigen stimulation, CD154 expression, TNF expression. You can see there's a few dots there, but it's normally pretty minimal background. Stimulate with PPD, you can see it's quite a clear positive. And, in some of the active TB patients, we later found they would have, up to nearly 2% of the T cells PPD specific positive. So we would define these as our antigen specific cells, gate on them, and then we would look at things like GM-CSF, interferon gamma, IL-2 and interferon gamma, and then CD27 as well. So this was all done simultaneously. So we looked at the different combinations of these 1, 2, 3, 4, 5 different markers to see which could best discriminate active from latent disease. And what we found was the best discrimination we found between the active and latent disease was those antigens responding cells against PPD that were CD27 negative and GM-CSF positive. So this is active disease, high proportion CD27 negative and also GM-CSF positive. The latent disease with the low CD27 expression, lower GM-CSF staining. And this is the area under the curve staining there. So it looks pretty good. But, you know, as a clinical test, not quite there. There's still some overlap between the low ones and the high ones there. But getting there, we hope. Whoops. And we also looked at the HBHA response. Now, remember, this is meant to be a higher response in the latent compared to the active. So this is the interferon gamma positive responding cells to HBHA. You can see the active is lower than the latent, but there's an overlap between the two, okay? If you had a high HBHA response, then probably, yes, definitely latent disease, but that wasn't on all the latent patients. So other people have been doing this, and there was a paper that introduced other activation markers to look at. So what they did was stimulated the T cells. This was a PBMC preparation, gate on the CD4s, and look at the interferon gamma positive cells. So they defined their antigen-specific cells, CD4 and interferon gamma positive. Then they would look at those cells. What's their expression of CD38? HLA-DR and Ki-67, a cell proliferation marker. And this is the differences they found between latent and active TB, whether stimulated with the cell wall preparation of MTB or the ISAC6 and the CFP10. And you can see there's daylight between the two groups for this particular study. CD38 positive, interferon gamma, class 2, okay, and the Ki-67. So clear separation. It was really, it's quite striking. They also monitored the expression of these markers over treatment. So this is time of treatment, and this is the expression of CD38 there. So you would expect successful treatment of TB is basically taking you from active disease to latent disease. So, and you see a reflection of that with the treatment. And even after 2 weeks, so this is just one individual, but the trend was there. Even after 2 weeks, you're starting to see a reduction in these activation markers, which indicates possibly the treatment is working. So if you can monitor the treatment early, you might be able to find those patients who aren't responding to treatment, who perhaps have drug-resistant forms of MTB. So we took that, and actually I did a study with Prof. Jusak at Erlanger University in Surabaya, and with one of his students, Dr. Harry, who was up in Banda Aceh. So we wanted to apply this CD38 class 2 measurement on the T cells and see if we could differentiate before and after treatment. So we wanted to measure if there are changes in the phenotype of peripheral blood specific PPD, specific CD4 T cells, in active TB patients before and after treatment. So we took them from Dr. Harry's outpatient clinic in Banda Aceh. It was a cross-sectional study. We did this study over 2 weeks, 2 weeks of collection, and then the blood was transported to Surabaya, where we did the staining and run the samples. And this study was supported by Erlanger University and some sponsorship from Cytec as well, because we ran the samples on their Aurora analyzer. And this is the paper that we got out of it. So small study, but just to demonstrate we can do this here in Indonesia and other places. So what were our results? So this is PPD specific CD4 T cells before and after treatment with the CD38. So this is the after was 6 weeks after they started treatment, and they were people who successfully who it seemed to be, the treatment seemed to work. So we're not seeing that super clear separation like you do in that original study, but overall there is a decrease in the CD38 expression on the before and after. Remember, it's cross-sectional. It would have been nice to make it longitudinal to say, you know, this patient went down to here, but that's not what we did, cross-sectional. Class 2, a little bit better separation. So the trend is there, not as clear as the other study, but you know, certainly worthy of further investigation.
Pardon, Dr. Paul, the time is about 5 minutes left.
Okay, I better hurry up. And then we also found some differences in the cytokines produced. I do want to go on and just finish up on talking about applying these assays to SARS COVID-2 specific T cell measurement. Okay, we all know it's been a bit of a problem lately. It would be nice to measure the immune response to this. Will we be able to get, you know, some understanding of the disease, why some people are more affected by the infection than others, and so on. So how important is the response to the virus? Is it protective? Is it part of the problem? Can we measure the efficacy of vaccines by quantifying and characterizing these T cell responses? So it's basically the same assay as we do for the TB. The difference is you just put in the COVID antigens, okay, the spike protein, the nuclear capsid, and so on. And this was done very early in the pandemic. This was published early in 2020. They looked at SARS spike glycoprotein reactive CD4 T cells in peripheral blood of patients with COVID-19 and also unexposed healthy donors, stimulated on cryopreserved PBMC preparations with 2 different peptide pools that covered the spike protein and that corresponded to the expected class 2 epitopes. In this assay, they used the upregulation of 137 and 154, the activation marker, to define the COVID-specific cells. So it's a little blurry, but you can see over here, this is the CD154 expression, and this is the CD137. So these up here are the activation markers. Down here, they also did some intracellular staining. But let's just focus up here. Patient 3, yes, 0.2% of the CD4 cells when stimulated with the spike 1, the N-terminus preparation, they could see upregulation of those activation markers. They're the specific cells there, and that could be measured across most patients. They also looked at the C-terminus of those patients, and they could also measure the interferon gamma response. Interestingly, they also found responses in some of the unexposed healthy donors as well. So unexposed, but with a pre-existing response against COVID. How's that possible? Well, it's because it's a coronavirus. We've got lots of coronaviruses that are infecting us. There's co-expression, you know, of the same antigens across different viral species. So that might be where that's from. And it seemed in this study, those healthy donors that responded to the stimulation, it was mainly directed against the C-terminus of the spike protein. So that's interesting. They think it's overlap of specificity against other types of coronaviruses. The real question is, if you got that pre-existing T cell specific against the virus, does it give you some protection? I don't know if they followed up on this study or not on those individuals, because remember, this was done very early, I think May 2020. But they hopefully have the information on those individuals and what was their course along the pandemic. Did they get it? If they got infected, was it severe or not? So we still await on that. The other interesting thing is they also found, like in TB, those patients who had active disease had that higher CD38, HLA-DR and Ki-67 expression compared to the normal. So this is HLA-DR expression on the COVID-specific cells. So in those healthy donors that they could measure it, very little to absent class 2 expression, whereas in active disease, it's very high. Not surprising, because those markers reflect activation of the T cell. Those patients who got activated active disease, there's going to be an ongoing immune response there. Can we measure CD8 T cells? The answer is yes, we can do that. You just see here, interferon gamma, CD69 on some COVID expressing cells. People have looked at severity. Is there some sort of response that corresponds to severity? Yes. More cytokines are made by the T cells in severe cases, maybe. There's a lot, again, no one's got a standardized assay, and that really impacts the reproducibility of the studies. Not to say these people did it wrong or whatever, it's just because they stimulate with different antigen preparations, different period of time, frozen cells versus fresh cells and so on, you get a difference in the result. But basically you can detect it. Can we detect them post-vaccination? Yes, you can. This was a study where they looked at the AstraZeneca vaccine, and they could measure responses CD4 and CD8, different types of cytokines being made, including some Th2 cytokines they were able to measure. There's a paper where they're looking at simultaneous analysis of both B and T cells after SARS-CoV-2 infection and vaccination. So you can take the sample, look for T cells and look for B cells. Basically, take your cells, P and PMCs, stimulate them with the peptide pools, leave them overnight, and you can also look for the B cells by having a spike protein fluorescently labeled. The B cells that are specific will be making antibodies against that, so they'll bind that fluorescent spike protein. And then you can stain with the different surface antibodies and measure it by flow cytometry. So this is just the B cells. Because we're looking for rare cells and to be sure of specificity, what they do is they have half the spike probe labeled with APC and the other half labeled with BV421. So to be sure that those B cells are specific, they should bind both. And so you get this diagonal staining. So it's just another specificity control within the sample. And then they were looking at the activation marker expression on these cells, on the T cells as their measurement of the T cell response at the same time. I'll just skip over the mate cells there, and I'll just finish to say flow cytometry, ideal technology for analyzing the role of T cells infection and also B cells. I didn't go over B cells, but you can use that technique to identify, to separate out, to sort those B cells and that are specific for a particular pathogen. Can quantitate and characterize the function of pathogen-specific T cells during infection. The measurements are likely to have, hopefully, some near future clinical applications in infectious disease diagnosis and treatment. And I'll finish there, and I'm happy to have any questions. And thank you.
Thank you very much for Dr. Paul Hutchinson, PhD, for your great lecture about flow cytometry application in infectious disease. Because of time limitation, maybe we can have only one question. If there's any question, you may raise your hand and state your name before you give question.
You can always come up at lunchtime too, or any other time, and ask a question. That's fine. I think we're all hungry for lunch.
Okay.
Yep. Question there? Yep.
Thank you, Dr. Paul, for the nice experience and presentations. My question is about what is the function of co-stimulation antibody? If we don't use the antibody co-stimulation, what is the effect to the experience?
So the co-stimulatories antibodies, CD20, anti-CD28, anti-CD49D, what they do is amplify the cytokine response is what we expect them to do. So what it means is the memory T cell, when it responds to the antigen, it's going to make more of the cytokine. It's giving it a boost. So if they make more, you can see the staining more clearer. So instead of making 5 interferon gammas, it's making 10 and so on. But in reality, I think for a lot of the measurements, you can get away without using them. They don't increase the background. I mean, the concern is you add those antibodies on your no antigen ones, it's going to make your background go up. There might be some non-specific stimulation, but usually, I mean, we've been doing this for a while. I never really see that. So it's just an extra, not a control, but an extra boost that helps you discriminate the staining better. But I would say certainly you can try it without them. And if you're measuring something, then that's probably okay.
Thank you.
Thank you very much, Doctor. Is there any question more? Maybe we will finish this question and answer session. Thank you very much to Dr. Paul Hutchinson, PhD. Thank you very much. Now, ladies and gentlemen, now we will move on to the next agenda. We will hear about product presentation from Beckman Coulter that will be delivered by Miss Lily Chang. To Miss Lily Chang, the time is yours.
Good afternoon, everyone. I'm Lily Zhang of Beckman Coulter Life Sciences as a flow cytometry specialist. I'm from Korea. Thanks for having me today to share about machine learning algorithms in cytometry data analysis. Oh, yeah. Well, the title of today's talk is From Terminology to Insight. Okay. I can't wait. I know it's time. You may be the most sensitive when you are hungry. Maybe I will try to finish this quickly. Okay. So yes, today I will talk you through the process how we can convert terms such as machine learning algorithms, dimensionality reduction, computational visualization into the experiment insight, which you can apply in your daily data analysis and accelerate your research progress. Yeah, before my presentation starts, this is a reminder that Beckman Coulter Life Sciences has a proprietary right to the information in this presentation. Please do not disclose, use, copy, or distribute this information without our written consent. Okay, thank you. Yeah, let's get started by looking at the first term, machine learning. What is machine learning? It's a type of artificial intelligence where computers use complex algorithms that can learn from data to perform analysis. It focuses on the use of data and algorithms to imitate the way that human learns, gradually improve its accuracy. You can see from this graph that machine learning is a subset of artificial intelligence. So and AI means to incorporate human intelligence into machine. Machine learning is a set of tools or algorithms to realize AI. And deep learning brings machine learning to a next level. Some real life examples, including Netflix's recommendation engine, or are you using YouTube or Instagram? Why every time you log into there, you will find similar genre or types of movie being recommended to you? This is because there is an algorithm learning what you have watched, learning your preferences, and based on the data it collected from your watching history, they can predict your preferences and recommend relevant shows to you. Okay, next slide, please. Okay, so former IBM CEO here, Virginia Rometty, also says such smart machine learning, it will increase human intelligence instead of artificial intelligence. So now I'm going to start talking about flowcytometry, which I'm going to talk about today. As you know, flowcytometry is an analysis method that is used in a wide variety of fields, and the technology continues to develop. This technology makes us analyzing very large numbers of target marker sequence simultaneously and rapidly, and it is continued to evolve. As a result, the data analysis is also changing very rapidly in terms of methodology. So it means the trend of the method of presenting analysis results are also changing very rapidly. This trend can also come from in the paper. This is because, as you know well, researchers make an effort to show their results using the latest trend. So if you search for machine learning and Flowcytometry together in PubMed, like this, you can see that the number of published papers has increased significantly in recent years. Oh. Yes, after we understand what machine learning is, let's see why do people start to apply machine learning in high dimensional data analysis. I'd like to use the blind man and an elephant as an analogy to illustrate high dimensional data is difficult to visualize. You probably are familiar with this story about a group of blind men who have never come across an elephant before. They try to learn and conceptualize what an elephant is like by touching it, but each blind man could only feel a small part of the elephant's body. Depends on the body part they touched, they now have totally different images on how an elephant looks like. So high dimensional data is kind of like this elephant. It's enormous amount of information, and we need to combine information from all dimensions in order to make the true discovery. Using machine learning, the algorithm can learn the data pattern and perform more accurate analysis. Another term you might often hear is computational visualization. Let's use a data set as an example to compare traditional visualization and computational visualization. If you have a set of high dimensional data, let's say just 20 colors, this is how we visualize it in a traditional way, using y-axis plot to consider the combinations of all markers in your panel. You need to go through so many plots just for one sample like this. It is laborious, time-consuming, and error-prone to compare across different samples. Therefore, people start to move from traditional to computational visualization. These are the example of using machine learning algorithms. We can generate various visualization and understand the data from different perspectives. There are different types of machine learning assisted analysis too. There is dimensionality reduction algorithms, including BISNI here, and UMAP, TSNI, CUDA, and OPSNI. Dimensionality reduction is a tool we use to reduce the number of dimensions in your sample and provide us a two-dimensional visualization like this. SPADE and FlowDOM, on the other hand, are the unsupervised clustering algorithm. The purpose of FlowDOM is to cluster all the events in your sample and maps the clusters to a self-organizing map. And SPADE performs clustering and also allows the comparison of relative expression level of specific biomarkers. Lastly, there is a Citrus, which is an algorithm that can perform both clustering and also identification of differences between groups of samples. Today, I'm going to use specific application examples to demonstrate how we can use BISNI and Citrus in our data analysis workflow. Now, let's look at the first type of algorithm, dimensionality reduction. I would also call it just DR from now on. The definition of dimensionality reduction, DR, is to allow for the creation of a 2D map like this, where each dot on the map represents each event in your sample. Cells that are close to each other on this map have more similar expression levels of multiple markers. I believe most of you have seen this kind of graph quite often nowadays, either in journal article or in a post presentation. As DR is one of the most popular and most commonly used machine learning algorithms these days. When do people use this dimensionality deduction? When you have a new panel, you are not sure which marker is significant or what is the best way to sequentially gate your populations. You can use BISNI to visualize all the events with all markers. Or if you are dealing with high dimensional data set, it is too complex for you to use the manual gating. There are multiple ways to show a BISNI map. On the left, you can see a BISNI map colored by density of population. If you compare samples side by side, you will see how the density of certain populations change due to different experimental conditions. Another way is combine manually gate population with BISNI result. For example, when we do immunophenotyping using PBMC, we know there are many well-established immune compartments. We know the markers which can help us to define these immune populations well. What I can do is perform some gating using my existing knowledge with immune cells, such as CD3 positive T cells, or I can use 19 B cells like this. So after I gate the population, I can overlay it to my BISNI result. And these help me identify those big immune subsets within known phenotype. There is another way of showing BISNI result. In the previous slide, I showed you a BISNI map colored with density and a BISNI map overlaid with manually gated population. Here, let's look at a BISNI map colored by markers. Expression level. Until now, you might notice that at axis X and Y axis, we are having TSNI1 or TSNI2 like this, instead of the fluorescence channel, such as CD3 or CD19. TSNI channels are the dimension channels, and it has no biological meaning. It shows how different events are from each other in this map. As I mentioned in the previous slide, each dot on the map represents a single cell or a single event, and each island is a group of similar cells. They have very similar phenotypes. When you choose to color the map with biomarker expression, you can see the name of the channel. In this example is CD4, and here is a scale to show the range. Red means high expression, and yellow means intermediate, and blue is low or no expression. So the islands in red colors like this are the CD4 high-leveled, high positive populations, and blue islands are CD4 negative populations. If you select any other markers in your panel, for instance, CDA, then you would be able to observe which island expressing CDA on this map. Currently, Cytobank has a choice of DR types. The BISNI algorithm as well as involved algorithms such as TSNI, TSNI-CUDA, ALPSNI, and UMAP. These different types of algorithm allow the result to be displayed in optimized your data to visualization. Now let's look at an example of it. The first one is for identification of treatment-induced changes. Normal human PBMCs were stimulated by Dynabeat T-cell activator, and lineage markers and cytokine expression was assessed with a 13-color antibody cocktail, and data were acquired on a 3 laser side effects of Beckman Coulter. So here is the result of it. We have 8 FCS files derived from 4 different healthy donors with T-cell activator. As you can see, the results are very clearly visualized and distinguished the changes between each experimental condition. Especially here, if you find out specific population that you can gate it manually yourself on this business map result, and you can display the result by heat map like this to examine expression level of all markers. Or you can also display the result by bar type graph with statistical test to verify each level. All these functions are in the same page of Cytobank software. You don't need to travel several software. The aim of this second study is to assess the changes in immune profiles of normal and COVID-19 positive donors. And PBMCs were collected from healthy and COVID-19 positive donors with different degrees of symptoms, and add the cells and drop in antibodies into drug clone tubes, and add viability dye here, and run samples on Cytoflex of Beckman cultures and perform data analysis using Kaluja and Cytobank. Here are 3 sets of panels being used in this study and the respective drop-in antibodies. The first panel is for basic immunophenotyping, and second panel for T cells, and third one is for the B cells. I'd like to briefly introduce this division here. Everyone, basically, when you mix multicolored antibodies, you combine all the antibodies in the black vial one by one to make a cocktail and treat it into cells. This drug clone is a reagent in which fluorescent antibody cocktail required is already put into one tube. It is very new system with new technology. It is not freeze-dried form, but rather mixed cocktail coated in a tube with Beckman's unique patented technology. All of these panels here are already made like this to explain the advantage of this tube a little more. What are the benefits? It is a room temperature storage product that does not require refrigeration or freezing storage, so it can be used very stably for a long time, and human error is reduced so that no matter who conducts the same experiment, it starts under the same conditions. In particular, what I'm most proud of is that I don't have to worry at all about which marker to attach and which fluorescence to experiment with. This is because these panels are all highly verified panels by a group called One Study. This group aims to prevent immunological rejection for the effective treatment of organ transplant patient in the early stages. So it is very reliable because they standardize the panel. And it makes the workflow very simple, just mix and acquire data. That's it. Of course, it depends on the sample tube, sample type, but still it is very simple and easy. But this is for the RUO. So here you can also get this IVD type you are interested. That's Clear Lab. It is also very simple workflow. Please see these details. It can be used for here leukemia, lymphoma, myeloma, MDS, and or MPM. And this panel also fixed configurations like this. But if you want, you can make your own panels by customizing panel by Beckman Coulter experts. Actually, at this point, I've got a lot of pricing inquiries from audience. But if you are really interested in these, please find Veronica or over there. I will support you to optimize your panel. And we also provide this kind of casebook for using ClearLab users. This casebook has been designed to assist in the analysis of flow cytometric immunophenotyping data generated using Beckman Coulter's ClearLab panels reagent for leukemia and lymphoma analysis. As you know, all the samples are not showing the same results. Sometimes you can find unexpected populations, or sometimes you have struggle with gating depends on cutoff percentage. So we can help on here to you by these kind of references. Okay, let's go back to the second example of business result again. After data acquisition, we use both manual gating and computational visualization for data analysis. Here is the slide to show you manual gates were created based on the existing knowledge we have for major immune cell population. And here is the result business analysis. When you perform DR and overlay the manually gated population on this business map, immediately you can spot on the difference between asymptomatic and severe symptom groups. As compared to the manual gating method in the previous slide, this is the fast and straightforward way to compare multi-dimensional data. After that, you can display the all of data with different degrees of disease severity like this. If we had to graph every single samples for these 12 markers in the traditional way, it would get very complicated. By the way, by using this algorithm, you can simply check and compare which population have changes at a glance with one graph. So as a result, it is very easy to visualize which population has the most change compared to a normal donor for each patient sample for each symptom. I keep telling you it's easy and simple. This is because after uploading your access data to the software, then adjust or just check the scale or compensation like that so that the samples you measured are displayed well. And then just click the business analysis button to get these results. You don't even have to wait for this analysis to end in front of your computer because it is a cloud-based software. So you can simply turn off your computer after clicking Analyze button. This is because it automatically completes the analysis and later inform you of the result by email. And I'd like to briefly introduce the data of Flowdome as well. These algorithms can be used when you do not want to use subjective analysis when phenotyping cells and want to directly compare a subset of cells in a new sample with a previous analysis. Unsupervised clustering, that is the top-down relationship between each event is analyzed with an algorithm so that each population can be distinguished without manual gating. And it is displayed in the form of DROM here. DROM is short for self-organizing map. In particular, the size and color of these zones can also express the expression level of the population. In particular, in Flowdome, when we want to observe how the same object changes over time, we continuously reuse the existing zone in addition to the existing file so that the algorithm can analyze its kinetics. So Flowdome overlay business on the clustering data obtained without manual gating for the populations anew in a separate a dictionary in this way to recognize what each population is on the business graph according to the Flowdome analysis. In this way, it is also expressed as a heat map like this. In this way, which markers change for population that show differences between samples to be compared. This conversion is as simple as clicking on the heat map in the software. In this way, you can display it in the form of a bar graph, or you can go for and compare the markers you want to see by processing statistics. Once again, same with previous example, all these functions are in the same page of Cytobank software. So you don't need to travel several type of software. And let's move on to another machine learning algorithm called Citrus, which can help you automatically identify predictable markers. This algorithm is particularly useful for biomarker discovery, and when you need to find differences with statistical significance. For example, in immunotherapy for cancer, you know that we always have different level of response rate. Some people react very well, have complete remission, but some people are not responder. As an example of the use of Citrus, I would like to show the result of a study on chikungunya virus disease announced in 2018. This is a study on the differences in the innate immune response to chikungunya virus infection seen in pediatric patients. Chikungunya virus is a type of new human pathogen that continues to be reported and spread even as of 2022. There were 42 pediatric cases in a hospital in Nicaragua, and total of 37 cell surface markers were analyzed by focusing on monocytes by sampling PBMCs from acute patients 1 to 2 days after the onset of chikungunya virus disease, and patient 15 to 17 days after the onset of the disease who had entered the recovery period has been implemented. Now, Citrus is analyzed with this workflow. First, it is a population we already know. The population positive for CD45 is gated in advance. And then after deciding which group to compare and specifying, if you click the button once to analyze with Citrus, then the algorithm analyzes similar cell populations while automatically clustering based on this result, overlaying using a BISNI display. It is visualized and analyzed for easy comparison at a glance. Here I said that clustering is automatic. The result is expressed like this. All populations for each samples are classified like this. And the algorithm shows the clustering that shows significant differences between the two groups I wanted to compare. This form is similar to the algorithm called frozen or spade, but the most significant differences is expressed in colors like this. And it's an indication that all children groups grouped in this way are in a parent-child relationship. So proceed with the analysis by selecting the clustering O. And this is because the clustering of this higher level contains all the lower concept. So it's also displayed in the form of a box plot like this to show how much differences is actually showing between each sample. So only for clustering with such a significant differences. And you can also visualize what kind of population it was by overlaying the BISNI image. In this BISNI data, you can check which island clustering population that shows a significant differences it belongs to. Although this population is finally invisible in the graph legend here now, it can be determined that it is a CD14 high and CD16 low monocyte. So the field of flow cytometry has entered an era of data analysis using machine learning. ML analysis can certainly help to quickly and easily identify and analyze a biological subset in a sample that the researchers are interested in, or they're so rare that manual gating can be error-prone. If computational visualization is used, experimenters can derive results with insightful data that can be analyzed for raw data containing vast amounts of information. It's just showing the first implementation of the Cytobank platform. You can see updates to the Cytobank system are being made annually. This keeps us waiting for some very interesting features to come in the future. So what I want to say is it's really good. Cytobank is the only product within Beckman culture to evolve so quickly. Really, I mean, the development is phenomenal. So this is the vision of Cytobank within Beckman culture to show there's a real commitment with Beckman culture. Cytobank is evolving to meet these two strategies for analyzing flow cytometry results. And it's very much with discoveries with the citrus and the dimensional reduction, but also with the guide analysis. And now we are taking a bigger dimension in a sense that we can do the guided analysis with these machine learning algorithms. So unsupervised with the flow zone, but we can also do the guide analysis now with the automatic gating. So kind of supervised, and then so we've got some kind of choice now how to do this analysis. And this is so powerful. So that's, we developing the platform, and that's nice. And here supporters are always waiting for your perfect and successful results. 24-hour help desk allows real-time data discussions between us. This is possible because Cytobank is cloud-based. And that means you can share and analyze data in real time, and not just with us, but with anyone else you want to discuss in real time. And certainly, if you have a data set, if you want to analyze by Cytobank, or if you want to try, you can create a trial account to try it for 30 days and try it. Even better, without having to analyze it yourself in this way, on our homepage in a space called the Learning Center, here's everything you need to know about Cytobank, very well guided. All of the tutorials are manuals with videos, so you can freely look around. And there are so many references and guidelines and also webinar materials. So please feel free to contact me and ask me anytime. I will wait for you. Okay, that's it. Thank you for your attention. At the afternoon workshop session, we prepared the topic of multicolor FCM panel design, instrument setting, and compensation by Dr. Joshua with a Cytoflex instrument. So please join and enjoy. Thank you.
Thank you very much, Ms. Lily Chang, for your presentation.
Moving right along, before we have a lunch break, we will take a picture together. We would like to invite all of the speakers, committee, and all of the participants to join us in front of the stage.
Kindly reminder, for all of the materials and lectures is already inside of the USB on your name tag. And for all of the participants has been divided into 2 groups. Kindly check your own name and room for workshop. Thank you very much.
kepada seluruh peserta diharapkan ke depan panggung karena kami ingin mengambil foto. Terima kasih.
Boleh. Nanti kami fotokan.
Izin mengingatkan sekali lagi untuk semua materi sudah ada di dalam USB, di dalam name tag masing-masing peserta.
Thank you. We will now have lunch break before we continue parallel workshop. Terima kasih. Terima kasih. But first, I would like to remind you about the group for workshop. All of the participants has been divided into 2 groups, and kindly check your own name and room for workshop and in your USB. Thank you. Good. Ada dua. Yang webnya di sana. Yang di WA group itu berbeda. Yang di share terakhir. Ikut
For lunch break, we will take about 1 hour. Untuk jam makan siang akan memakan waktu kurang lebih 1 jam. Untuk peserta yang memerlukan untuk ke musola, untuk sholat, musola terdapat di bagian atau di sebelah. Untuk tempat sholat sudah disediakan tempat di sebelah kiri stage. Untuk musola terdapat di lobi hotel. Terima kasih. Mohon izin untuk mengingatkan kembali bahwa workshop yang akan diadakan di ruangan ini adalah Bali workshop. Jadi bagi para peserta yang akan workshop nantinya akan dilakukan di sini terus. Lalu akan dilanjutkan di ruangan sebelah untuk mengikuti workshop. Terima kasih.
Halo. 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 Halo.
Ada imbannya ya.
Hello, hello. We will go very slowly, very in the beginning. We open a blank sheet, we open a plot, and right from scratch, we'll see how we create magic and how we make the dots talk to us. Okay, what kind of data we can see, what analysis we can do, the difference between a region and a date. What do you mean by a region? What is a date? What is a median? What is a mean? When you use what, why you use what you want to do, what's a histogram, what's a density plot, what's a contour plot. We'll see the differences around that, and slowly we'll build on the software. So do not worry about what I cover and what I do not cover in the next 1 hour. I am assuming that most of you are new to Flowcytometry. Are you? Okay, so then you will not, so whatever you learning is a continuous journey. So you cannot learn everything in 1 hour, 1 hour, 90 minutes. There's no need to also stress yourself about that. Important thing is whatever you learn it well. And then you will have your own instruments in your lab and your own software, you will play with it and you'll go by it. Okay, this is a workshop, interactive. As I do something on the screen, I keep on speaking. Try to do the same thing on the laptop if you want to. If that is your style, you learn more by doing. If your style is you're comfortable seeing what I am doing on the screen, focus on that. Okay, and every now and then I give you 5-10 minutes to just play around on the software again. Up to you, depending on your speed, depending on your goal. Okay, we'll go by that. So, what I have with me today, first of all, to show you is a normal blood sample, normal peripheral blood sample, which has been stained. We will not go how it has been stained. We have the FCS file. What does FCS stand for? What does FCS stand for? FCS, forward scatter is FSC. What is FCS? When I say there is an FCS file, what do you mean by an FCS file? Anybody? What is FCS? Forward scatter is FSC. Side scatter is FSC. I am saying I will be importing FCS file. So if you see, when you create a new experiment, what is FCS? Google quickly. When I say import FCS file, what do you mean by FCS? Flowcytometry standard, very good. Google knows everything, right? So you need to take help of Google, very good. Okay. So, Flowcytometry standard file meaning that these data files are based on some internationally accepted standard which includes a certain basic information about the data that has been acquired. It will include the voltage information, it will include the fix whatever it has done, to give the sample name, field name, to include lot of information. Now, I have imported already a file which is the one flow for acute leukemia orientation flow. And for that, we will be now looking at a normal blood sample. Okay. Now, suppose there is no blood there. What happens? Of the software. Okay. So this is how the software will look like for you. You see this guy? Tracks you 1.0. So you have to demonstrate the software. When you demonstrate the software. Let me close this and I can open it again to you. So you need to double click this. So Amitabh, are they password to this? You are doing for all of them? Okay. So then you need to put in the password. Okay. Then it opens the software. Then in the software, what do you see now? Experiment. What is an experiment? An experiment will include maybe number of tubes in the same patient that you are running, tube 1, tube 2, tube 3. Each tube and experiment will have lot of analysis template. It will have plots, region, gate, statistics. Okay, all of that it will have in that. So now what we go is, we go into the Bali workshop folder that I have created. I built a practice folder in this. So if you see BD administrator somewhere, you can right click and say new folder or just say new experiment. If you say new experiment, okay, it will create a new experiment like this. Experiment, maybe it will call for you experiment 001. Okay, then within that experiment, it shows you one tube which has no data. You see this tube, this is blank. There is no data in this. And then it shows you a default plot. Now, then you can go into file, import FCS file, and then you can go into laptop, in your laptop, wherever the files are, and import maybe just one file, normal peripheral blood one flow a.fcs. Open. And it opens the file. Now you see this tube has data in it. It is filled, black color. Okay. Repeat again. At this step. Creating experiment. Have you imported the FCS file? Okay. Okay, so let's go again. In the BD administrator, right click, new experiment. Okay, you got the new experiment. So the new experiment we had was experiment. For me, it was this. For whatever, for you, it may be experiment 001, right? So now, this was for you like this, blank. Correct? You don't have what? You've already had an experiment. You already experimented. So are you? I don't know. I can't. Do we have one spare laptop of this kind? Then I can show them exactly what they have. Or the already been given. It's okay. Okay. Okay, so I was showing importing HCI file, but you already have an import experiment. Okay. Huh? Yeah. Okay, so you see like this case one normal TV, case two you see, so you click on case one. Okay. In this you see some plots. Okay, highlight all of them by just all of them and delete. Okay, you have many plots, right? You say control A, okay, control A, all the plots will be highlighted and then you say delete. Either you can click delete or you say right click and say delete. Okay, everybody has screen like this now? So delete all plots. So this is the blank worksheet which is default now. No, you can just delete the plots in that worksheet. No, we don't want to. Yeah. This is basic data analysis. They don't. Yeah. Delete all the plots. So you can also, instead of I would want them to delete. I want to start with a fresh worksheet. Whatever the worksheet elements are, just delete them. I can show you, for example, if you are on the managed experiment, suppose you click on case number 2. You click on case number 2. Okay, you click on case number 2. You are seeing all of them like this, case number 2. You see like this, case number 2. Okay, do control A. You come to the worksheet and say control A. Everything will get highlighted like this. Right? Right? Everything highlighted? Okay. Then right click and say delete. Okay. Okay. Yeah. So I want to show you how to create that. Something in the lab also you need to create the plot. Correct? So how to create a plot and template? Case number 2, yes. Case number 2. Same way you will do for case number 1 also. Okay, I think I will have to change the format because at different groups we cannot. What I suggest is you first focus what I'm doing. Okay, everybody. Hello, everyone. Do not look at your laptop now. You just focus on what I'm doing. Otherwise, this is not a software training. This is the purpose is to understand the logic of what has to be done. Okay, the software training, Hasan, Adi, they will do later on for you in your own places. Because if you don't, otherwise you spend all the time just doing this. Okay, it will not help anyone. So focus first of all what I am doing. Then I give you some time, 30 minutes in the end. Then you play on the laptop at that time. Okay. Okay. So we are on the case number 1, normal table blood sample. We have 3 tubes, A log, acute leukemia tube 2, acute leukemia 3. In leukemia analysis, always for the first time when you do, you should run normal blood sample to know what the expression of the population is or the cell marker is in the normal. If I have to create a from scratch, this is what I see, create dot plot. I click on this, and when I click on this, I get a dot plot. Okay. Now in this dot plot, because this was not from scratch, there is already some information which is being there in this. So what I will do is I just delete all of this and we create this from scratch. Because this is how in the real life for you do not have any information available to you. Okay. So you click on dot plot, you create a dot plot. Then you can right click on the dot plot. If you want to take pictures, you can make video, you can make it for your own level. I have no problem. Okay. I click, see on the right side properties. I go to, it's a dot plot. I can go to parameters, forward scatter you take in linear, side scatter you take in linear. And I have a forward scatter, side scatter. Okay. Usually when we do leukemia flow cytometry, we first try to clean up the population, and commonly we use a time plot as well. So what I can do here is on x-axis there is option of time. So I can choose that time. It shows me how the data was acquired over a period of time. And on y-axis, generally I can take any parameter. Maybe I can take CD45. Okay, this is showing as the data was acquired over a period of time, whether it was consistent or homogeneous or not. If you see a lot of populations going up and down, that means the fluidics were not stable. Okay, so once I have done this, now what I'm going to do, I'm going to right click this properties in the display. Many of these will be new to you, but slowly as I keep on doing it, you will become familiar with it. In display, the grid outline, I will call it full. So now you see, if it is half, I do not see this. This is my preference. I like to see it full. And now I am going to, this is my time plot. In this again, what I can do is, on the title of the plot, here, in the plot title, I will say just show me the tube name for the first plot. I will just call it tube name. It is showing me what data is coming from what tube. This is OF A log tube. Now in this, I'm drawing a region. Okay, it says gate. What you draw on a plot is typically a region. What you draw on a plot is a region. So I am clicking on this and I'm saying, okay, I want to analyze all the okay, I'm going to analyze all of them. That's my P1, population 1. Okay, then I can right click and duplicate this. I have one more plot now. In this plot, I say show me forward scatter height and forward scatter area. And again, for both of these, I can make them as linear because the scatter signals could be ideally seen in the linear. And I have my population here. Oh, I think, sorry, I should have height. I can right click here, height here, and area. Linear, and there. So these population that you see generally is coming from doublets, two cells traveling together. In this population, in this plot, again, what I want is to show the events which are gated on this population P1. In plot editor, I go here and make it P1. So now, and in this I say show me population. What population is this gated on? This is gated on P1 population now. Correct? If I want, I can also change the color. Usually I have a color coding that I like to use. In the time 45 plot, I'd like to use only the color as black. Okay? And how do I get that black color? For my P1 population, right click, properties, and I can go to the color, and this is my black color. Okay, so this is your gate properties because now you are on that gate. Correct? So now once I have drawn a P1, I can draw one more. I can draw one more region here. And all these are singlets. This is P2. And what do I want to color as P2? P2 I like to color as light orange, which is P2. I click on the plot, on the gate, right click, properties, and here I change the color to here. Okay. Then I can again right click on this, close this, right click on this, close this, and say drill down. Drill down, it will create a subsequent plot only gated on the population that I want now. I want forward scatter and side scatter. It has created this. And again, I can change this back to linear. Okay. So this is forward scatter, side scatter. Here I will draw one more region now, and that region I will color it as slate gray color. And in this, you see here, okay, I'm drawing a region of interest. This is somewhere I will need to exclude the debris. And this is P3, and right click, properties, and I want the color here, slate gray. Now, I will give you a tip. How do I remember what color to use? The choice of color is your own, whatever color you like. But whatever color you do like, it is easy to remember that color. I like to give it by the name. You see, how did I do? This is row 1, row 2, row 3, row 4, row 5, row 6, row 7. So I call, this is what I call as slate gray. So to me, the slate gray color is equal to 72. 72 means what? Row number 7, column 2. Okay. Otherwise, all the columns look very similar. It is very hard to remember that. So I go here, row number 7 and number 2. This is my straight data. It's a good way to remember to color and convert into a number. At the same time, you can label this. Now, the label that I put for this is I call it as NDCE, non-debris cellular events. Okay, you can call it whatever, cells, NDCE, this is just my way of doing it. Similarly, for this gate, we will relabel this as stringlet. Okay, this means that these are the stringlets that we are seeing. Now, what happened? Why did this color change to gray? Because this is a population of stringlets. So as you add more colors, it keeps on changing. If you want to see the original color of that plot, whatever it was, then in the properties, in the dot plot option, you say show population with gate shown on this plot only. Now see what happens. The moment I click on this, it shows the original color of that gate that you had made, the region that you had drawn. Correct? Okay. So similarly, in this plot as well, I can say in this plot also, I can say show plot with gate shown on this plot only. It will show it as black. But generally, in the time plot, it is okay to see all the colors because it tells me whether they have been acquired uniformly or not. Now I have the NDCE. I can make this by this icon. I can click and make the plot bigger inside. Okay, you saw how I did? I click here and make it bigger inside. Now I see, this looks like debris. This is my normal lymphocyte, monocyte, neutrophil. So I can move it like this. Okay. I'm putting the cursor and I can move it. I can also double click on the region and move individual points like this. I can do that also. But this looks okay to me. So I'll say done. Now I double click on this, drill down, it will create one more plot. And now I want to look at CD45 on x-axis, side scatter on the y-axis. Okay, it has given me one more plot, dot plot. Now in this again, I can make parameters, side scatter back to linear, and it is showing a side scatter 45 plot. Now you see, starting to see how the analysis is done. And we'll keep it safe there. And within this now, we will create more regions. These regions will look like this. If you want to take a picture of this, just take a picture of this first. Can you take out your camera? Just take a picture of this. Take a picture of this. We'll take a picture of this. Okay. The color scheme we will use from here. So you see now, within this, we will create a blast region, then a lymphocyte region, then a mono region, and then a gram region. So I need here polygon gate. I can put the shape later on. I will need one blast region. I will also need one for lymphocyte region. I need one for monocyte region, and I need one for gram region. Correct? Now you see, I increase the size. Now I will put them to the right place. Mono and then gram. Okay, so this is what I am going to call as okay. This is the so-called my glass region. I can reduce it inside. You see this? I have left the debris out. This is the glass region. Correct? P2. Then this is, and we'll change the name later on. This is the lymphocytes. Okay. And some of these may be large lymphocytes, little bit here. Okay. Then this is monocyte. Okay. And granulocytes is okay here. You can take these and drag them. This is the P4 was your monocyte. Let's see it here. Where is P3 gone? Yeah, here is your P3. So P3 I keep it here, that's your lymphocyte. I put it back. Then P5 is here, and the P2 is last. Done. Now we get the colors right. So we wanted for glass, we wanted the color 108. 108 means right click, properties, go here, 108. 108 means 10, 8. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Then this is 1, 2, 3, 4, 5, 6, 7, 8. Red color. Okay? It makes it easy to remember. So glass are this color. If you remember the color like that, sometimes you forget otherwise. You know, I like to write this is glass, and I'm calling this as glass. Right? Okay. Close this. Then this one, the color is 48. 1, 2, 3, 4. This is lime. Okay, this is green, lime. And I call it lymph. Okay. So 45 is on the x-axis, right cutter is y-axis. What is the question? The lymphocyte should be right 45, right? How do I? That is my wish. You can use whatever color you want. My rule, so I like, so this is where you have a picture. This is my rule. Glass, I keep red, lymphocyte, I keep lime color, I will make it bigger in size. So my rule, I follow this. Glass, red, lymphocyte, lime, which is 48 number. Monocyte, I keep blue, which is 82 number. Yeah, this is GRA, neutrophil, granulocyte. I call it violet, 96 number. This is just my rule. You can have your own rule, no problem. Okay. But this is one rule I have seen it works well because the color, they contrast with each other very, very well. This is one rule I have seen it works well. Okay. So now we go to monocyte and we make it 82. We click on the base, properties, here, 82. 1, 2, 3, 4, 5, 6, 7, 8. This is 1, 2, 3, 4, 5, 6, 7, 8. Yes. This is blue. This is monocyte. 82. Okay. And I label this mono. Okay. Then for neutrophil, what is the color? What is the color for neutrophil? Anybody? What is the color for neutrophil? 90 something. 96. So this was 8, 9, 9, 1, 2, 3, 4, 5, 6. Okay. 96. That's neutrophil. And labeled as GR. Now you see, this looks nice. You can adjust the gate a little bit. Okay. Now I will draw one more plot here. I'll say duplicate, right click and duplicate. And I'll keep it here. And in this, I will call it site scatter 34. Okay. Now you see, also as I did the region gate here, in the coming plot, it is showing you the color of all the population. That all the populations were acquired uniformly without any problem. Correct? Now, in the site scatter 34, my interest is in this 34 positive population. But this is normal. You don't have one of 34 positives. In an actual patient sample, you will have 34. In a case where there is clearly 34 positive population, for that, I like to have a plot just for my 34 positive population, and I will, so that I call it like this. Okay, this is P2. So this, what is the color for 34? I will keep it close to red. Not red, I am keeping it tomato color. Color number 16. Okay. So for this P2, right click, properties, color 16. This is, I think it's already 16, tomato. And I'll call it CD 30, CD 34 positive. Let me see what I call it here. CD 34 positive. Let me call it CD 34. Positive. Okay, increase this in size, and this is your okay. No, this should not be so. Okay, now what I see, by mistake, I do it. Properties, TV 34, and the color is already 17. Okay, this is how it should look like. So you should all be seeing something like this. Okay. Remember, a normal blood sample, although what I am showing here is 34, but this is not actually 34 positive population. In normal blood, this population is generally your basophils. This population of low side scatter 1045 is generally your basis. It is not blast. When you will start seeing the patient sample, then you will see your blast population here. Okay. So now what do we do? What is the next step? The next step is now, okay, we can copy the first row plot and paste to page 2 and we will take more and more to do them. I think the charger is going. So first, you are here. Looks okay. So I am going to add now more page. Okay, in your computer, do you see this option? Were you able to get 5 plots on the same page? Were you able to get 5 plots on the same page or no? Now, okay, anyway, doesn't matter. You just watch what I'm doing. See, this one changes the page orientation, portrait, and you can make back to landscape. Portrait, landscape. This option, change page orientation. I like to keep the landscape option. I like to keep the landscape option because it allows me 5 plots in a row. And this top row is my row for identifying my population of interest. Okay. I can click on the add page. The add page. Add page, and it will add new page at the bottom. I will add one more page and add one more page. I will add one more and one more. So total I need 5 pages. I need 5 pages. 1 page for each new data. 1 page I will use for my pasting my population hierarchy in the end. And 1 page I will use for that. So 3 cube, 5 pages. 1 page for 1 cube. The 4th page for analysis hierarchy. 5th page for that. Correct? Okay. So now that I have, and this is what I mean by hierarchy. All events, then P1 is, I can click on this P1 and change this to, I can change the name to time. This is time. Okay, time, singlet, non-debris cellular event, and within that I have blast, lymph, mono, gram, CD34. Correct? Now you see a pattern emerging there. Now, within this, what are we going to do? These plots are going to be common in all the tubes, because all the tubes have CD45 and all the tubes have CD34. So I can say Control A. I can again right click. Either I can duplicate or I can copy and come here, click at the top left, right click and say paste. It has pasted everything. All of them it has pasted now. I can similarly now go here, next page as well, and say paste. Correct? Just, and then what do we do next? Now what we are going to do is, now there is something we call as, in the next page or in the next row, we are going to get more slot. I first duplicate this slot. I keep it here And in this plot, I will say show me marker on the x-axis here, and I think it will be 79. Okay. In the plot properties, so I have taken to a FITC PE, and I go to property and make sure that these two plots are bi-exponential for the fluorochrome channel. So the marker data should always be seen only in bi-exponential scale. I think that's what I was telling in a class about bi-exponential. So bi-exponential scale, it is okay. Then what is the next step? Then I am going to create right click, one more plot by duplicate. Okay. And here I am going to look at the population for CP Site 5.5 and maybe 19. Site 5, Site 7. We go in this order: blue FITC, blue PE, blue Site 5, blue 7. Now you see I can highlight all two of them together and do that duplicate. And slowly just touch the bottom part, these two will get highlighted. And here I can drag all of them together. Okay. Now, what happens here? I can say, show me data of 7 and CD3. Correct? And in this plot, I will look at the data, maybe cytoplasmic 3, and I can look at the data of CD3. Okay. I can keep, so generally what I will do is these 4 plots I will not change. I will add one more plot based on case-to-case basis in case I need to look at some other marker combination. I can always play around here and I say I put here 34 and right now maybe I just put here 20. But this is kind of flexible. And now see what I do. I highlight all of these by just touching somewhere on the plot and I can say top align the top. Align top. Now see very close to what happens. As I say align top, they move slightly and they're all aligned very well. Similarly, I can go and on all of these and I say align on the top. Properly aligned. Correct? Now, the way I created this, I highlight all of them and say copy, and I come here and say paste. Okay, so now it has pasted all of them, and I put them on here. Okay, so this is how my cube 1 data is looking like. In this, generally what will happen? 34 will be your blast will be your population of interest. Like in acute leukemia, blast is your population of interest. So on this plot, I will say right click, property, and show me blast and CD34. So now it shows if it's showing only blast or CD34. Correct? And here I'll say show me the population, my internal controls, lymphocytes, monocyte, and neutrophils. This is showing the population, which is my internal control. As these cells are basophils, there is some bit of NK positivity you are seeing. Okay. So you see this now? So you understand, now this tells you how much of background is there. It tells you that even when in the neutrophil there is no CD7, there is some background signal coming from autofluorescence. Okay. Okay. So let me slightly. You see this now? Now, similarly, what I can do is I can create similarly plots for, and now I don't have to repeat the same, I can just highlight all of them together, right click, copy, and paste them here. Okay, can come down, kind of move it. Right? And similarly, they have already been copied. I come here, paste. I take these up here. Here, up here. Okay. So I have data for all the three fields now. Now I tell, okay, Mr. Software, for these plots, I want to make sure that the data is always coming only from the cube A log. So run pointer means as if the run pointer is split, it means that every time when you move to a next cube, now see here, if I move to cube number base, the data changes. The data changes. If I move to cube number 3, data changes again. Why? Because this is on run pointer. Wherever my pointer is, show me that data. But when you are creating your template, you want a neat template. So I say, tell the computer to the software, you do not give me run pointer. You give me only, let's say, for these plots, give me only cube of A log. So now, even if I move, it will not change the data. It will only show the data for A log. Okay. Similarly, for cube number 2, I want here it to tell me, remove the run pointer and give data for AL2. Okay. For cube number 3, I want to remove run pointer and keep data for AL3. Make sense? Okay. Now, all that is okay. The other thing you can do the coding is okay. Now, all of these regions that you have here in the cube number 1, all of them that you have here, there is a feature known as okay, now this is an important point. Look here on the screen. If you see the region on this is not matching exactly with the region on this. This is your right now. Okay, so this is still on. You need to make this also, all of them also on. All of these should be also on A log 2. Okay, and all of these, the first row here also should be on AL03. And the first one should be on A log. Run pointer A log. Okay. Now appreciate this point that this plot is showing the regions were drawn according to this, but they are not matching with this. Why? Because Cube number 1 is A lot. A lot is cytoplasmic cube. In cytoplasmic cube, the marker intensity can be different. As you notice that the CD45 intensity for the lymphocyte in the AL2 is slightly higher compared to the CD45 intensity on the lymphocyte here. We have drawn region on the cytoplasmic cube, but that region is not valid for your AL2 and it is not valid for your AL3 also. Correct? And even you see in the, oh, by the way, cube number 3 does not have CD34, it has CD4 for CD5. Okay, so for cube number 4, so for cube, now there are two things we will do. Now think about the logic. We want to tell the software that whatever region I'm drawing on cube number 1, that should be unique. That should be unique only to cube number 1. Correct? So that when I go to cube number 2 and change the region, I should be able to change that. So the software allows you that feature, and what you can do is go to this region here and say make unique. Make unique. If I make this unique, okay, if I don't make this unique, I remove by a control Z, I undo the feature. If I do not make this unique as I move it here, what happens? It moves everywhere. You see, it moves everywhere. I do a control Z and I go and make this unique. Once I make this unique, even if I move this now, it does not move in the other tube. You see, it does not move in the other tube. Correct? So same way, I will go and make other regions also as unique. And I can, if you see, that I can go directly the population hierarchy and in cube 1 here, select NDCV, and once you make something unique, it will show a cube icon here. It tells you that this is unique only for that cube. And I will now maybe highlight all of them and let's see if we can make them. No, it doesn't allow me unique in this. So I have to go and individually go here, make it unique. I go back to my cube number 1. Unique. Unique for lympho. Unique for mono. Unique for neutrophil. Correct? You see in the hierarchy, here is the hierarchy one. All of these are unique now. Unique means a small tube comes next to it. That's unique. And then in CD34 as well, I make it unique. Now, other important thing is when I was doing the CD34 positive, I realized that in my cube number 3, actually I do not have CD34. I have CD4. See on the screen, CD4. So this region should not be applying to this. What do? I say on CD34, right click, and I say property, and I tell make strict parameter matching. What does it mean by strict parameter matching? It means that when I am drawing a region, on a site scatter for CP5555 plot, which is this plot, correct? So you see here, this is the site scatter for CP75. I have drawn the region 34. Moment I say stick parameter matching. Now, only in the plot which has site scatter 34, it will show that, but the moment you have a site scatter 4, it will not show that. Let me show you again. When the region was drawn on a site scatter 34, now people do not know, remember like 34 or whatever, understand the site scatter and the fluorochrome channel. Because drawing the region 34 on that, in any other view, in that site scatter for CP75, but the moment you tell it six parameter matching, you go to the properties for the gate and say six parameter matching. Now it will not show the region here. I undo my control Z. Now you see the region again. But this is actually C4. This is not 34. So we don't want the region here. So I have to make it six parameter matching. It's an important topic. Would you like to explain in do you understand? Maybe explain in the language. So, maybe just explain it on what I am doing by the big parameter. That if in two different tools, you're drawing a region, the parameter of the region, so we want to apply the region only based on one parameter. Then you use the parameter. Okay. Understand all of you are new to the software, which must be already in right now. I understand. Okay. So if you are very, very new, do not try to understand. Yeah. So those who have some idea of software can play. Something like this. Don't try to remember the steps. You cannot. This is what possible. If you do not make the new one, good question. If you do not make unique, then when you go to cube number 2, and here you move this, then it will move even for the cytoplasmic cube. So, you get the point. So see, right now, if I move this here, and let's say move here, if this was not unique, even this would have moved here. As you will see, for plot, this plot, that I am not very unique for that, so this has moved here. So sometimes there are few to few differences in the population. Okay, Cube number 1, training protocol is intracellular training protocol. This is your active leukemia orientation cube, which has cytoplasmic marker. Cube number 2 and 3 are surfaces. The expression pattern of the marker will be slightly different for CD45. So by making it unique, you make sure that when you make small changes to the region on the plot, they are specific only for that region. They do not disturb the data in other groups. That is the purpose. So even for this one, I can come here, make it here, okay? And generally you will find that between the data will look same because both are showing the same way. But for acute leukemia cytoplasmic you want to make it for unity. Okay, now clear? Okay. Okay. Now, once, so what we will do now, this is done for cytoplasmics. We will go now here and see if we need to make some changes based on what we have seen. But even for NDCE, let's make this first unique. So this is already unique. Okay, cool. So I'll come down here and I need to change this slightly. Okay. Looks okay. Now, even this I want to change slightly. Okay. Done. And I need to make some changes to the regions here. This is lymphocyte, monocyte, neutrophils, monocytes here. Okay, and these are okay now. This is animal CD4. So all these are okay now. 2, 2 and 3 are okay. Okay. So I have my plots ready now. And remember the first plot always was showing the tube name. This is A log, then AL2 dry, AL2, this is AL2. And then everything else is just showing the colors. There's one more thing you can do to identify that the plots are different. Is what I do. Select all plots of page 1 and we will display the background color to BD light blue 15. How do I do that? I highlight all the plots. Okay. Right click, properties, display, background, 1, 2, 3, 4, 5. BD light blue. Now, it's a kind of making the mind used to a certain pattern. That any plot which has this boundary, this is my cube number 1. Even if sometimes you don't have this, you know which is number 1. And I come to, now I come to, I can reduce this in size. So I can see all of them in one page. I highlight all of them. Right click, properties, display, background. What was the color? Light orange, 18. 5, this was 5, 6, 7, 8. Correct? This is my tube number 2. And then we come down again. Okay. What was the color? Burley wood, 26. Right click, sorry. Properties, display, 26, 1, 2, 1, 2, 3, 4, 5, 6. There you go. Correct? Now, what do we do now? We create a analysis hierarchy and print it here. Okay, so in this, I will, once this becomes active, I print this. Now this is permanently printed in my template. Okay, I have all the populations now in this. And I can say show population stats. It will show the population stats as well. Okay. Now, what does it tell you? See here. It tells you that the blasts are 1% of the parent population. 1% of NDCE. A log similarly here, I can increase this inside. Increase this inside for the full page. You can see it better. All this you have to do only once to create a template. You don't have to do it every time. The reason I'm showing you is I could have shown you the template and they will do the data science analysis tomorrow. But once you create a template once, you save it, you don't have to do it again and again. You will see the second page will become very fast. It will become very fast after that. Now this is the stack, this is the hierarchy. And then I can create a multiple tube stats. I open a stats box. In the stat box, again I can say, show me which all populations I want. Okay, in the A log, okay, it tells you in the A log tube, you want maybe all events and then you want your blast, min, mono, gram, and 34. Okay, it is showing you all the ones here. Then you go into the AL2. It is showing all of them, but I say maybe now I don't want time. Okay, maybe I want singlet and I want all of them here. Similarly, here, I just don't need maybe the time on here, maybe I can use the time here. Now it is showing you all the populations from all the different, all the tubes. All the tubes is showing you the population. But A log, AL2, AL3. I can also right click on the properties and change the size. The title size, I can make it 14 here. Okay, then decimal places. For some of them, you can decide whether you want 2 or no. Maybe for grandparent, I can say 3 decimals. You can see that here. Color and font, title, from family. Okay. Okay. I can also add here in the stats. I may not always be interested in mean and median here. I may not be. I may be interested in percentages are already there. So maybe, okay, I say, give me, I don't want forward scatter mean. I don't even want this. I don't even want side scatter mean. Maybe for NPO, okay, I say, let's look at the mean. Okay, and we look at, let's say, the CV. Okay, I look at just the, I'll just show for one marker, maybe for simplicity 3. If you want to find something fast, you can just type CD. If you want to go to CD3, it's showing you all of them. CV3, we will just look at, let's say, CV and V. Okay. So now Also in the stats window, I'll show you something very powerful now. I will edit the keywords and I will show you here, let's say, acquisition date for maybe one of the files and time of beginning at acquisition, time at end of acquisition. Now look at the power of this. As if your technology, you have the template, you can always go back and look at when the data was acquired, on what date, what time it started acquisition, what time it ended acquisition. It is all part of the template itself. If something was done on day 1, day 2, all that information is there. Time at beginning of acquisition for AL2, time at end of acquisition. It is showing you all that information there. Okay. You can also add information like sorry, no, yeah. I can put here a $sign and P. It is showing me the parameter parameter 1. Then what is the voltage for parameter 1? Okay, now what it tells me? The parameter. Then I go, for example, I just take one more parameter. Parameter suppose equal to 5. And I go to parameter 5 voltage. Now look at this. What it tells you? What it tells you is, see here, parameter is forward scatter area, but the voltage in the A lot and the cube leukemia, second cube and third cube, somebody has seen the voltage cycle. It's a good quality control. It's a good quality control for you to check. C voltage, parameter number 5, because parameter 1 is forward scatter area, parameter 2 is forward scatter height, parameter 3 is side scatter area, parameter 4 is side scatter height, and parameter 5 is fixing. So fixing is the parameter name, and for the parameter 5, the voltage is 472 for the first cube and 471 for cube number 3. Usually, there should not be a reason to change the voltage between the different cubes. You should not be changing that. Correct? But if every time you will not be sitting in front of the machine. But if you have this in your template already, and if something is changing, then you can ask your scientist, the technical officer, what was the need for change? Why did you change? Sometimes that can help understand sample acquired in day 1 or after 2 months, maybe follow-up sample or some different sample. You see something is shifting usually, you can quickly go and check what the voltage is very much different. What was happening? Okay. So now suppose this is what I am interested in. I am looking at now. I have all the percentages with me. And what do I see, for example? In cube number 2, the monocytes are 3.73%. In cube number 3, monocytes are 4.1%. In cube number 1, monocytes are 4.2%. So everything is matching by now. That means the data is consistent across the different cubes. Okay. Now that if I am happy with my template overall, okay, I am happy with my template. What can I do now? I can, this becomes my master template and I call it as file, save as, I'll say save experiment without data for now. and I call it as after acute leukemia template. Okay, now see the font. One side template. Once I save this, it goes into my experiment and it causes a master acute leukemia template. Correct? Okay. Master acute leukemia template. Now next time, before that, let me just explore the Excel file of this screen. Okay, so what I have just done is in a folder, I have my okay, so next time now, when I open the master template, in this master template, I go to file, import FCS files from the other case, and I highlight all three of them, and I say open. Now you see all your data has automatically come into place. All the data with all the stats, everything is into its own space now. Now only you need to go and check, this looks okay, yeah, this looks okay. Maybe this, yeah, which also looks okay, no more change, this is okay. This one, lot of blast. So maybe I need to change this region. Here I make it like this, and maybe this is a little bit more of not many monocytes. If we are really no monocytes, maybe keep here or just take it away. Doesn't matter. And lymphocyte, done. And here also lot of CD34. Okay, done. Now look at this. When I look at now in this row number 2, because I have clearly 34 positive population, actually I do not need this blast. Okay. Now what is the problem with this blast? This blast is not a pure population and is showing some mature population. So, but these are my here neat and clean 34 positive. So in this case, it's just here that for the second row here, I will say show me only CD34 positive. Okay. Now what does it show you? Now how do you know this NPO is negative or positive? I refer to the internal control. What do you think looking at this? If my lymphocyte is here and the blasts are here, do you think the blast is positive or negative or impure? Look at this. If you are in doubt, maybe along with this in the same plot, you add your internal negative. Now you look at this. NPO positive should be here. But this is the lymphocyte. What do you think? Is the blast positive or negative for NPO? Negative. Is the blast positive for 79A or negative? For 79A? Is the blast negative or positive for 79? Because yes. Okay, guys, now we are coming to analysis. So everyone focus here. Let's focus here now. And now doing the analysis. The template is ready now. Now see how we are going to do the analysis. Now see here now. Internal lymphocyte control formulation compared to this three for the blast segment population. Positive or negative for 79? Positive. Correct? You don't need to draw any quadrants or anything. Simply by looking at the internal control, that is positive. And how do you know that NPO has been added? Because my neutrophil, which is my internal positive population, it is positive for NPO. Correct? Now look about this. What do you think about 34 and 19? 19 is strong positive. In WHO classification, you use the word strong and dim. What do you mean by strong? Look at this blast. The expression of the blast of 19 is somewhat similar and even more than the normal lymphocyte. That is strong. And what do you think about CD7 and CD3? This is the blast, this is the neutrophil, and this is the internal normal T lymphocyte. The blasts are clearly CD7 negative and surface CD3 negative. Correct? And then cytoplasmic 3. Without the internal controls, you may think, oh, this is sitting on 10 to the power 3, this may be positive. But the moment you look at the internal control, you know, oh no, these blasts are similar to the internal negative control. So this is just a high background, but the expression of cytoplasmic 3 on the blast is negative. Okay, because even my normal B cells, the normal B cells are showing some bit of cytoplasmic 3. What is the diagnosis? This is 34 positive, 19 positive, 79A positive. This clearly delineates ALS. Okay, in the same case, if we now go to second Q, now the beauty is because we have already done your template, your analysis becomes much faster. So I go here quickly, and in this again I try to see, yeah, I do need to move the region. Okay, rest looks still okay, that is fine. Here as well, I can say just show me the this is CD34, okay. And the monocytes I can take little away. I don't need these monocytes anyway. In this case, it is purely positive. And now here, same way, what does it tell me? What do you think about? Okay, now what do you think about CD13 and 33? If, as I said, if you are in doubt, you can always just add at least lymphocytes, if not everything else, just add lymphocytes. It will help you in deciding. What do you think about CD13? CD13 is positive or negative on the blast? Yes, positive. Correct? This is positive. 117. Now, what do you think about this 117 pattern? This is not positive. When you see this fishtail kind of appearance, that fishtail appearance is coming because of the spread of C3 psi 5.5 into the P psi 7. Because in the morning of that spread. Correct? This is that spread. So as much it is going up, the same it is going down. That is your spread. That is not genuine positivity. Then what about C33? Positive. Correct? Clearly nicely positive, and I know that neutrophils are internal positive control, lymphocytes are internal negative control, and nicely positive here. Then HLA-DR, neutrophils are the internal negative control. There are some B cell or T cells which are showing activation and which are HLA-DR, but HLA-DR for the blast is strongly positive. CD14 is negative. Now, if I put that monocyte little bit back here, I just move it just in this area so that you can see. Now, what do you think? This is that little bit of monocyte. This is your HLA. This is 33 and 14. Correct? See this? Monocytes are 30 and 14 positive. Monocytes also are HLA-DR and 14. Okay. That is how the way to interpret in acute leukemia is by looking at your population of index, which you may define either on the site set of the plot in the last region or as a clear cut 34 positive population. In comparison to the internal reference control population, internal reference negative control, and internal reference positive. Correct? Now we go to tube number 3.
Pardon, Dr. Paris, the time is about 5 minutes left.
Now you look at the tube number 3. Now here we do not have to make any change because when we made the changes in tube 2, those changes in the region automatically apply to tube number 3 because we made the unique gate only for tube number 1. Okay, all these gates are looking okay. And now in this, but now here there is no CD34 region. So here I should simply say for properties, I should say make it only for class, correct? And it is not showing CD34. Now what do I see? Again, just for as I did earlier, I can make in this also. If nothing else, at least I can add lymphocyte because it gives you ready comparison. What I can also, if you want, we can bring in the lymphocyte population. I can have this, and in this, the lymphocyte population, I can say, bring population to front. I can say in this plot, bring the blast population to front. The blast will come in the front. See, it was like this earlier. If this is better, you can see like if you want to bring blast in the front, you can do that. How will you do that? Click on the plot, right click and say which population you want to bring to front, which population you want to front. You can do that. So what it tells me, the blast are CD5 negative, CD19 positive, CD4 is negative, CD10 is strong positive. What do you think about CD8? Is it positive or negative? CD8. CD8 is negative. Even though it is going up like this, it is also going down like this. It's a fishtail pattern. In the fishtail pattern, this you are seeing because pen is very bright and that is spilling into the APC-H7 channel. Okay. Then what do you have here? CD20. Would you call CD20 as positive or negative? Positive. Correct. And CD5 is negative, you already see. So it's a very nice case. 34 positive, 19 positive, 10 positive, 20 positive, cytoplasmic 79A positive, and among the myeloid markers, what was positive? 13 was positive and 33 was positive. Remember? Here, 13 was positive and 33 was positive. So this is the VEATLS, which is 13 and 33. Okay. So once we have done this, the next case, again in the template, import all the three FCS files. Once your template is done, everything goes very fast. Okay. And you come down here if you want to look at percentages or stats. Now, in this case, what happened? Now let's review this. The last thing, then we'll close it. See here now. Data was acquired on 28th August 2019 for all the three tubes. Time at beginning of acquisition was around 10:40. Okay, beginning of acquisition for AL3 was this, for AL end of acquisition was this. Parameter is forward scatter, all voltages were 216, parameter 5 is 50, all voltages were 216. Which is how it should go. Okay, the same way generally I have now shown only two parameters, I use all the parameters there. Yes. So I keep this as a part for every sample. Even if I am running 3 samples in a day, it will show me when it was acquired, what was the routine. So when you classify quality control is an instrument to see difficulty FNB. Okay. This helps you in understanding any variation which could be coming between the two tubes of the same sample. Okay. Or if you find that this month the data was very good, after 15 days the data is not good, then you can ask open the figure to open either you use the instrument to have all information, or if you are 3 month analysis of it, you can check some information. Okay, these are little more. I was trying to reach your version of the software, advanced feature of software. This is very helpful. Once you start getting used to it and you forward differences and want to rule out the experimental condition and the variables in your example here. Now, one more thing to look at this. Remember I showed you the CD3. Look at the CD of CD3 lymphocyte. This is A1 for you. Tell me. Sylvie was asking me on the table, what happens if my instrument with the TV with a P is coming 7% and it fails, TSNT fails. So I was telling him, even for something like a lymphocyte CD3 expression, which is a very tight homogeneous population, look at the CV of CD3 on the lymphoid population. This is the lymphoid population. Even on the lymphoid population, the CV is around 76%. Okay, you can say maybe this CV let you get a robust CV. So we go to edit stats and say should be robust CV. So we want to get so I go to CD3 and say should be robust CV. I'll remove CV. Even the robust CV for lymphocyte for CD3 is 56%. So when the biological variation within the same sample is 56% in the same sample, then if your instrument CV for that channel for ATCX7, whether it is 6% or 4% or 8%, will it matter much? No. So it is done. So I mean the biological sample would still be a good analysis. But what is important is if your instrument gives you showing a trend increasingly going up, you need to figure out what is before it goes very up. This is really just to show you that. So the software can allow you to see from biological CV perspective and keep it. You will just not. What is for CV code and normal improvement? Is it changing over here? If it is not in this thing, you find out everything right. Okay, so I think I end here right now. I will open it for questions. Maybe something which somebody has not discussed well, and you want to quickly show you again and improve it. But the whole purpose was to show you how you create a template from scratch, completely blank. Okay, otherwise if you don't see these things, you need to see everything, you don't know how it will be. That was the whole purpose. Okay.
Thank you very much to Dr. Pares for your great explanation. For all the participants,
Yes. Yes.
Bagi para peserta untuk workshop pada sore hari ini sudah selesai. Kami mengingatkan kembali bagi para peserta untuk mengembalikan kembali laptopnya ke bagian belakang dan para peserta dapat mengambil break di luar, dapat makan di luar atau dibawa kembali ke dalam dan kembali melanjutkan ke web workshop di ruang sebelah. Terima kasih. Bagi para peserta yang memang daftar untuk web workshop. Terima kasih.
Info driver. Kalau info drive oh, aku tidak pernah terima. Jadi memang web memang terbukti. Aku habis kopi break. Jadi apa saya
Selamat sore, selamat datang kembali kepada para peserta dry workshop pada sore hari ini. Kepada para peserta, saya persilakan untuk mengambil laptop terlebih dahulu di bagian belakang untuk kelengkapan untuk melakukan workshop. Terima kasih.
Everyone? Is everyone back from team? We start? Okay, to start?
Thank you so much, Doctor.
So we move on to the second session for the last one. We must have seen some good data acquisition, how you check voltages, pain index, right? In the web lab, when Dr. Saka was at home. So I talk now about the data analysis. So the way I designed that is we all have the cases that I will showing. Okay, so now what focus on it, I'll show you what should be good look like. How I do analysis and do data analysis for instance, I'll show you that with few cases. And so that you understand the logic of where we want to go, what we want to achieve. Then I will start with a blank worksheet completely and show how those plots and regions and waves, ISAC, everything works really there. Okay, and then hopefully if you have some time in the end, you can practice on your laptop as well. Those cases are there, then you can practice there. Right? So we have 90 minutes, so I try to show the first 30 minutes. I will show you some, 20 minutes, I'll show you some cases. Then I'll show you how to create a template, and then you create the model. Okay, good. So what we have here is, we all see here, look here. What we have here is, for example, a classical data mapper, data analysis, a template that one could be using for acute leukemia analysis. Now here, what you have here is a time plot and a CD45 plot. We use the time plot to ensure that the fluids during the period of activation are stable. If you see a lot of signals that are going up and down here, this green is not stable, this is not stable, then you know that there is some problem in your data activity. Okay, where these colors are coming from, I'll explain you very short. Then what you see here, this is the time plot. Then what you see here is the plot of forward scatter height and forward scatter area. In this plot, what we are trying to exclude is this population of doublets. This linear population is the population of signals. Okay, wherever if you see the larger area signal with low height signal, that is your doublet, and we exclude that from the analysis. Here then the third plot you have is on the forward scatter, side scatter. Here the intent here is to exclude this debris, and we call it, I like to just call that as NDCE, non-debris cellular events. NDCE. You can call it whatever name you want. I just have to call it as NDCE. And you try to include in this all the different populations that will be there. Now, then comes your main plot of site scatter CD45, where you draw populations of interest. Now, here I have drawn a region on the low site scatter, right, 45, and then this is my lymphocyte region. Okay, this is the lymphocyte region, and I have here a monocyte region. There are not many monocytes in this sample, so I don't have to worry about monocytes. There are some, this is very likely maybe a bone marrow sample. There are not many mature neutrophils. Predominantly what we see the blast population of low site scatter, low 45, okay, 10 population. So this is your blast population. I also like to include another plot which has a site scatter and CD34, because many times when 34 is positive, it is much easier to identify your blast on the 34 plot. The reason being, in the plot, in the plot that you see on site scatter 45, often it is very difficult to draw the line between the blast region in the gram region and the blast region in the mono region. It becomes difficult to draw that. Let's say now suppose I move this here. Now you see it can be quite hard at times to see where this population is ending and that one. It's a very arbitrary cut. So if 34 was completely negative, I would still use that. If 34 is nicely positive, then I would still go ahead and use this population, to qualify my blast. So this top row is what I call as is a row of defining your regions of interest, defining your populations of interest, which includes the blast population, and it also includes the internal reference lymphocyte, internal reference monocyte, internal reference neutrophil population. It includes all of this. Then the second row is the row which includes all the different markers which you have added in the tree. And in that, the intent is primarily to look at your population of interest, which in this case is CD34, and maybe I like it's a good idea to keep just one internal negative population, for example, lymphocyte. So it's then we have a weak population. But if you are more comfortable not keeping that population there, you can remove even the lymphocyte population and you can see the plant. Then you just see the plant. Okay. So when I do it, I like to just keep it the blast. But when you are relatively new to flow, it's a good idea to keep lymphocyte there. So you have some anchor population there to call what is negative, what is positive. Then I definitely like always to include this one row, exactly same marker. As you see, the marker that you see here, same MPO79A, same way you see MPO79A here. Exactly whatever you have the markers in the row number 2, the same markers you keep in the row number 3. Do not change that. So that visually itself you are able to see what is internal negative, what is internal positive. So it's a good practice. If you follow these things, then your analysis strategy and eventually will be much faster. Okay. So this is your row of your population of interest, then this is a row of your population of controls. Internal lymphocyte, internal neutrophils, these are the population there. Now, for example, now we look into this, 34 is the blast population. If you were to simply look at this, it may be troublesome for you to say whether this is a positive or negative. So you do not perceive going all the way, this population is going all the way up to close to the 10 to the power 3. Okay, so you may be confused whether this is positive or negative. The only way to know that the high level of fertility is when you look at the internal count. And if you are able to visually see this, well, this is overlapping with this, so this is a very likely MPO negative. If you are not in the beginning able to make that visual appearance in your mind, you can just add at least an lymphocyte population here, and now it will become easy for you to define. Okay, no need for any quadrant or anything to be 18% positive, 12% positive, 23% positive. So those percentages can change depending on how you place your project. Okay. If you want, the software does allow you to place quadrants. You can always place your quadrants. Okay, you can always place your quadrants like this, and and then define whether you want to place a quadrant like this, or you want to make it more staggered and take it up and down. Like you can make that. All those features are there. And there will be situations where at times you will use this quadrant, but by and large, in routine diagnostic phenotyping, this is generally not required. Or remove this for right now. Okay. So now what do you think about, for example, now, do you have any doubt about CD79A being positive in this case? It's clear-cut positive, CD79A, right? It's nicely positive. Similarly, 34 and 19 compared to my internal control, clearly positive. Very. This is what I mean by strong 19. Look at the normal B cell. Look at the expression of the plant. Very strong, right? Similar, even more than the normal B cell. That's a strong CD19 that we are seeing. Then, for example, CD7 and CD3. Here, these are the normal T cells. These are 7 positive, CD3 negative, very likely some normal NK cells, which are coming from the lymphocyte here. Okay. And then you see what we call as blast. Blast we call as blast are CD3 negative. Yeah, CD3 negative. You see the right sitting in the middle of this is your internal negative population is very likely to be a big cell. Then look at the cytoplasmic tree. Now, without the control, you may be inclined to think that this cytoplasmic tree is positive. Keep sitting at the part 3. So the moment you look at your internal control, now you know even the B cells have the same level of expression. is just the high background that you are seeing in the cytoplasmic. It's a part of a staining to the thing that why you getting high cytoplasmic. And I've seen that especially the choice of buffer you use, the fixed inform buffer, whether or not you're adding the BSA to the wash buffer, all that can have an impact on the background that you see in the cytoplasmic. So irrespective of that, at least it tells you that whatever you are seeing here is background and not a true expression of cytoplasmic. So based on this cube, we learned that this is 79A positive, 34 positive, 19 negative, very likely to be a lineage error. And the fact that your reagents are working well, your neutrophil is showing nice white and clear, okay, is telling you that your scaling is well. Now what about this? This 34 positivity is very likely coming because some of the neutrophil region cells in actually include your blood. Okay, that is the reason why when you draw regions on sites that are 45, these boundaries are very arbitrary. When you draw versus I draw, the third person draw, there will always be some variation in that. So you need to be mindful of that when you're analyzing the data, but at that point of time, interpreting the data, that whether this looks more like a blast population, that this is likely to be just a true leukemic population which has gone into your plant region. Okay, so the regions, just to show you what are the regions, there's LY is a limb, NO is monocyte, GR is GAN, and this is a part. Okay. Now, when you see this, now this is a very fractional software feature. When you see this cube icon next to the region, what it means is that this region is unique to this cube. That means even if I move this region on this cube, it will not have an impact on the subsequent. Okay, just to show you an example, if I move this region even here, in the second cube, this region is not moved at all. It is still there. Okay, so this is quite helpful. For example, in this case, specifically, you will find that there are small subtle differences when you look even at the sites that are 45 plot between the surface staining cube and the cytoplasmic cube. Cytoplasmic cube will always show you slightly lower intensity of CD45 because you are punching holes into the cell membrane. Okay, so that we can go inside. If some CD45, you will not be able to see because of that. So now, if you see here, now let's say suppose I remove this unique feature from here, and then we will remove this unique feature from this. Okay, remove unique. And similarly here as well, in this one there is no unique because I am seeing there is nothing here. There is no cube sign here. So if now I move this, let's say I bring it here, you see, in other cube also can move accordingly. Okay, so while in the cube number 2 and 3, which are both the surface staining cube, now there is no all of that. But the moment I make this region as unique here, now this is unique plot, and now even if I move this back, okay, that one has not been. And now I can go back and move this exactly as per what I want for the Denpasar. Okay, so this is one interesting feature which allows me to have different regions across different views in the same time. Okay. Now, other point of interest here to understand is I have drawn this region around CD34 positive. Now, in my panel, cube number 1 has CD34, cube number 2 also has CD34, but cube number 3 does not have CD34, it has only CD4. Okay? So how do I avoid that situation? Now, if suppose in this plot I say that, so for this marker, for this region, when I go to the properties, when I go into the properties here, it has this feature called strict parameter matching. If now look at this, if I remove this big parameter one, then any region which you have drawn on the side scatter per CT psi 5.5 plot, the same region will get appeared even in the cube where there is no synthetic force. You see, even in the cube where is a side scatter CD4, which region is appearing, which is meaningless. Because I want that region to be there on side scatter 34. So the software then has a feature which allows you by qualifying this as a unique, and it extends the uniqueness of the region to strict parameter mapping. That means this region will be shown only, and this region will be applied only when the actual parameter also matches along with the parameter. So these are some very important features which allows me multiple analysis in the same template, and one needs to be in. Now we have analyzed the cube number 1. Now we go back to the cube number 2 here. Now what do you think about this case now? If you are just looking at this 64 and 13 plot, what do you think the blast would be? Now this is showing on gated on 34 and this is gated on 25. So I'm showing that on the top of the plot, I mentioned always, what is the population of interest it is showing. On the first plot, I like to also just mention the cube name, that this cube is the AL0 cube. Okay, so these are following some good principles which makes your analysis easier later on. This is from cube number 2. This is gated on time, gated on singlet, gated on non-debris cellular events, and then gated on whatever population you are showing. Now, if I were not to keep the lymphocyte population here, if I was to exclude the lymphocyte population from the display, and I ask you to tell me whether these blasts are 13 positive or not. Historically, what you will do, you have an unstained cube, you will try to draw a quadrant and see whether it is 20% or 30% positive. That, again, as I said, in a population which is as heterogeneous as this, that can be very arbitrary and different people can get different effects. And from a practical perspective for a clinician, whether it's in 13, you are calling as 28% positive or 36% positive, it doesn't really matter. Important thing is, are you calling this 13 positive or negative? And to qualify that positivity, what is important is compared to your. So first of all, you need to ensure that 13 has been added, the reagent is working, as I can see from my internal positive lymphocyte control. And then I look at the internal negative lymphocyte control, and this, if you can mentally compare it, it's okay. If you have trouble, in that case, you can just add the lymphocyte population and tell you clearly that yes, I have a CD13 which is present, and it's a big expression of 13 that I'm seeing on the box. Okay, I mean, if there is a practice and you really want to know what's the quadrant or whatever, you can do it. You can go back and you can make a quadrant here. Okay, and now after having made the quadrant, you can just, for the sake of it, you can remove your, if you want, you can remove your default population from here as well. And if you see in your, if you go into your stats, it's generally upper left is 26% is positive in this. You see it? Oh, it's not. I have to hover it on that. So if I hover it on this, it's a 26% is upper left, and the lower left is 70%. I mean, but as I said, it's important to show that this is positive. And I generally they do not like to use what it's not. 34 is clearly positive. What do you think about 117? Will you call it positive or negative? Negative. Here, this fishtail appearance, whenever you see, this is generally the result of spread coming from 5.5 into the P side 7. I showed you in the morning how the spectral lower causes spread, then it was a problem. So this is not a dual positive population. So if in this case you were to draw a quadrant like this based on this lymphocyte population, and you were to say, even if I keep it here, okay, and you were to say, oh, my upper right is how much? 10%. So you may call it 10%. let's make it bigger in size, and somebody will draw that somewhere like this. Okay, and then you will take it as 2017. Somebody may call it like this. And now the next we go with that. Now, yes, so if you are happy with it, 20%, you will call it very. But if it's for slightly more up there and you are 21%, so that percentage will not determine whether it is negative or positive. You need to see how the population is looking like, and on that basis, you know that this is where the knowledge of fundamentals of climate design and the structural spillover spread, you know, the cyber and biomechanics will indicate that there is this, and it's a very bright signal that you were seeing on the before. So that is the reason why we are saying this way. Now, go to see 33. What do you think about 33? Positive or negative? What do you think about 33? It is positive. Clearly positive. That's signal you are seeing. In this case, we know the 3 millimeter, we are seeing the P1, and now we are clearly seeing the 13 and 33 are accurately also. HLADR is strongly positive. That level of HLADR positivity can be seen essentially in only 3 meters. B, mono, or dendrite itself. Classic positive dendrite. So far, and every day we keep an eye on the internal controls. 64, neutrophil expression. Neutrophils, HLADR, should be negative, they are negative. So the internal controls are always very helpful with any leukemia analysis. If you are not looking at the internal controls, then you are very likely to fall into your trap and lose one time. It's very, very difficult to have your internal controls in the same analysis. Now we look at the cube number 3. What does it tell us? Okay, what about CD5? CD5 is clearly negative. Okay, now and then 19 is there, CD4 is negative. This is just that pin shape appearance. This is again some amount of P57 spilling into complete cytoplasm. Adjacent detector on the left side. But because it's a very bright signal, therefore it is having a little spillover around. But anyway, even if you were to do a quadrant, you will not find it more than because it will be not substantially positive. It's clearly negative. Now, what about CD8? Same way, because the appearance is now, this is where it will be really big. Now, let's see, let's get a quadrant here. If you want to do a quadrant, it's somewhere here and you will try to draw a quadrant. Okay, I would be possibly there is a compensation error. There is a compensation error, and if you would correct for compensation, the population will come down. Okay, but knowing very well that because this whole pattern is coming like this. Okay, that fishtail pattern, we see the percentage by this is somewhere close to 70. Now, I'm not going to call the CD8 positive linear error. No, it is not. It is not a CD8 positive linear error. Now let's see if we want to now know this is the cube number 3. We come to cube number 3 here and we try to look at properties. We go into scalar values, and this is CD10 and APCX7. Go into spillover and we look at APCX7 minus APC. This is the final here. Okay, we improve from APC. Here we go up. Am I getting it now? Yeah. You see now? That going to stay the chairs have actually the spillover effect. So the way else can you have spillover it's not only that what other spillover can commonly stay into APCX7 channel? If you remember now a spillover guide, on the common emission spectra, it is the P size 7. But now the question is, what is the P size 7? P size 7 has CD19. P size 7 has a CD19. So it is this CD19 P size 7 also which could be having this huge spillover impact into the APCS 7. We are going to take your advanced class, advanced group, Dr. Yana told you. You are familiar with Flowcytometry. So the importance of spread is to understand not only from the channel that you are seeing on the plot, but you look at that what all fluorogram can spill into this channel. And if the signal is very bright in the channel, you can still have a spillover impact from spread there. That is the main lesson you need to understand here. Okay, so this population will improve when it come down, even when you change the P size 7 spillover into APCS. Even when you correct for that, you will see improvement there. Okay, so this is clearly again negative on the interpretation. What about CD20? Positive. Now let's apply the same knowledge we learned in the morning. Why is this not a spread? Why is this not a spread? If I ask you that question. Then, okay, what is filling into? It is filling into V450, correct? The channel of interest here is V450. Now you have to think what all can spill into V450. Will Fitzy spill into V450? No. When you go back, let me see that is still open. Okay, come to that. Think of that. I don't want to go and change plots and all that. We'll take another time. So in B450, the only thing that will spill over slightly will be the B5 channel, and B500 is sitting on the right side of B450. Okay, so there is nothing which is in this tube which is really going to spill. And B450 is the B500 is 645, which is already anyway what you see is a very dim to negative signal. It's not a bright signal. So it cannot be an impact of spread that you are seeing here. Now, okay, so this is now look here. Why do you think that the glass have a higher spread in the CP 5.5 channel compared to what we see in the normal infrared? Clearly, we see a higher spread, although it's clearly negative, but the spread is higher. And now you have to think what all can come into CP 5.5. Well, P can spill into CP 5.5, but there was no marker in the P channel here. So that could not have contributed to it. What could have contributed here is the very bright P size 7 signal because of adjacent detector. 5.5 is sitting on the left of the P size 7. P size 7 is 780 nanometers, 7.5 is around 690 nanometers. So because of that, you must see a certain impact of spread. Okay, so this is how one would, do not worry if you're not getting everything now. But this is the concept you need to bear in mind as you start analyzing the data, and that will tell you how good or bad the data quality is. But what we need to do. From a diagnosis perspective, very clear. 19 positive, 10 very bright, 20 is positive. We have already seen 32. We are 79. Classical with 13 or 23. Average. Okay, we'll see one more case, and then I will show you exactly how we create this template from that. In the template, although you can also have the stats and gates population, this is just showing you all of them as 2 by 2. This can show you exactly the A0, what is the percentage of glass, 77% monocyte, and it can correlate across the different tubes. Whether the normal population are the same percentage of that tells you about tube to tube consistency of the sample. Whether there has been some error on that. For example, lymphocytes in the A0 tube are 11.2%, and as I go down in the Second tube, there are around 13%. In the third tube, there are around 15%. That's fine. You know, it's not that in one tube is 11%, other tube is 14%. Then you know something is wrong. There will be an indication around that. In the stats window, again, there are multiple options that you have. Now, one approach is, one thing is simple. Okay, now let's go to the stats and give it a little bit. Now see here. Now what I like to do here is, all the tubes I'm able to see, and actually I can select which all population I'm interested in seeing, whether it's as a percentage or whether I want a certain stats around mean, median, CV, whatever I want to look at. You can edit that and choose that. Now that's generally there, in all the software, and this can give you a quick comparison whether the percentages are matching. The grants are 10%, grants are now. You see here somewhere in the gating strategy, in this, between this is different. And you may want to go and correct that. This is 29%, and between Q2 and 3, we have consistency, but in Q1, the grants are 10%. Okay, so there is some inconsistency in the way somebody has drawn the region in tube number 1 versus Q2 or Q3. Also, the problem could be that Q1 was a cytoplasmic tube. In cytoplasmic tube, generally the site scatter goes down. And it's possible that some of the granulocytes have lost their granules and they're merging with the blast population. So that's how you can, troubleshoot, your different populations here. Now also, the software allows you to edit keyword. I find it very useful feature as a pathologist when you're trying to review your data. You can add information around acquisition date, time of beginning of acquisition, time of end of acquisition. You can also see the voltages at which the data was acquired. It becomes again a good tool that within the same day, different samples, within the same sample, different group, was by error or by mistake, somebody acquired a different voltage. And could that be the reason to explain why 34 is working in this tube in the same day versus other? You want to make sure that it's voltage. Okay, so all that can be included as a part of your template. So in a typical, suppose the 3 tube acute leukemia template, you can have less, you can have more. I would generally have, 1 page for each of the tubes, and then I'll have 1 page for each of the tubes, and then 1 page for my analysis hierarchy and the data, and 1 page for the stats. I mean, I can, depending on how many pages I want more stats, I can even add more to on that. So that, once it becomes my analysis template around this, then I can save this as simply as my analysis template without data. Okay, you just do it without data, you can save this. And then once you open this and you are now looking at the next page. Now see, this is your template ready without data. Assuming your panel is the same consistent panel, now if within this I import the Excel files from the subsequent page that we have acquired on the day. Now let's see, and I'm going to this space. Okay, now see what happens. All the populations are populated, and your stats are there, and hierarchy is there. You can go now, of course, you will need to go back and look at whether the regions are appropriate or not. Maybe this one I need to increase it. So analysis template once properly created, save you time later on. And in this, I have not shown you to create a report. You will actually create a report here. But important thing is now, what do you think about this? Are you happy with this or going to change this? Maybe a little bit more or less, we want to make any difference. This will work quite well. In the site kind of modified, yes, you will need to change. And you can see from the 34 plot. Now, what I do is, if I am in confusion, I just take all of them out and see what are my main populations of interest. Okay, so here I can see this clearly in the mature interest site. So I just move this on the mature site. It's possibly this is one population. So you can have grants over here. I'm making an assumption here. It's arbitrary region. And then Anyway, because 34 is strongly positive in this case, I need not even worry about this except for the cube number 3. So cube number 3, I do not have 34, so I need to play with this. But here, maybe even, okay, let's just leave this here for the time. And then we go into the plot here and I say, this is our 34 positive. Okay. That's what 34 positive population. And I do see here that there is some neutrophil I have left. If you really want to fix this, you can come here. And now, I got some neutrophil there. Okay, so now, same way we do the analysis now. All the same plot, all markers are there. What do you think about NPO in this case? The blasts are NPO? Negative. 79A? Negative. 34 is strong positive, you know that. What about 19? Negative. This is the spectral spread. Now CD7. Strong positive. 7 is there and 7 is there as well. You can see this 7 is almost as bright or even brighter than the normal T cell. This is the normal T cell. These are your red. Okay, and then what about cytoplasmic 3? There's a half log shape. Now this is the cytoplasmic 3 is tricky. You know, honestly, it's there in that in a one flow channel, but I'm not particularly super happy with that. It's okay because there are so many other values of the standardized field which will use it, but we have seen to use the right buffer, which is the domain and the BSA background goes down. I would have trouble making a very strong interpretation of cytoplasmic 3 positivity. But what I do, you know, we try to interpret in the context of what that population is otherwise. You know, you are like the company you keep. The company is speaking is about bright CD7. So I would be inclined to call it cytoplasmic 3 positive only because of that. There is a small shift I see compared to the internal one, but if the 7 was completely negative, I might just call it that. Okay. Okay, so this is where you're making a leap of faith there. And the neutrophil is a high background, so we have seen, as I tell you, this is more of a staining artifact. Otherwise, the neutrophil also did not be shown such a high background. Okay, now, so this phase here, 7 is strong, cytoplasmic 3 is there, separation is. Now, let's look at some other markers and let's look at what happened. Now, because this was a unique gate, whenever I change the gates to this, it did not make any impact here. Okay. So I will have to do the results again for this one. This is the lymphocyte. That's mono, I can leave it there. And maybe this is the one I'm interested in. I need to just make sure that my blast region is nicely identified with 34. Okay, done. So what do you think about 13? See, without the lymphocyte angle, it becomes suddenly long. Okay, so this is where either you need your quadrant, or the way I understand what I like to have is I like to have rather if you have to have another internal. This internal control is very important, very critical. So clearly now you know 13 is positive. Okay, 34 positive, 117. There is a now. You think that this 117 is a spread? You think it's a spread? No, it's a distinct cluster. There is a valley, there is a gap here, and then you have a cluster. So this 117 is there in a subpopulation of cells, but it looks like to be a very distinct cluster which is positive. Then 33 again is positive. Wow. Now I do not know why this is now this once I start seeing P here with a CD14 positive, with a CD117 positive. Is it 14 positive? Looks like 14 positive. Okay. But then I see there are other cells also, maybe are these monocytes. I do not know whether they are monocytes or not. Okay, even if I exclude this out, if this would be very unusual for protein. Now, but think about that, if these were monocytes, what other marker should have been positive? HLA-DR. Then HLA-DR should be positive. And this case, this is not, and even if I bring this back here, you see HLA-DR anywhere, let's see. Does it show HLA-DR? If it does, so there is some HLA-DR, not only this much. There is a very small percentage of monocytes here. There is a CD14 positivity. Now, it's very hard to say whether there is some screening issue, but I just noted right now, there is some positivity that I'm seeing. And now, can this be coming from some spread? Unlikely. It would be very unusual for a TLL to be positive. The only variable I would like to use or exclude in such a cases, is there any high amount of cell viability issue in this sample? Because if the sample viability is compromised, that can lead to non-specific. And look at the forward scatter, side scatter of the tube number 2, this is tube number 2 versus this. Which one looks better? This looks better preserved forward scatter, side scatter compared to this tube number 2 and tube number 3. They have very different scatter. Now let's look at what does the troubleshooting tell us. I'm very curious, I have not seen this issue earlier. Now let's see what do we have here. Do we see something which is different here? Okay, acquisition date is still the same for all of them. So that's not showing any problem. The voltage are same. So it's unlikely an impact of age of sample because the data has been acquired on the same date for all the 3 tubes. Sometimes it does happen in a lab when you acquire the cytoplasmic as a screening tube on a Friday or a Saturday, and you decide on a Monday or Tuesday to do your subsequent tube, and that's where maybe the sample quality is gone down. But in this case, it cannot be explained by that fact. There's something else, or there is a genuine some CD14 in small percentage, or is it that we are dealing with a small myeloid subflow which is 117 positive, 14 positive. So I'm not getting into that part, but this is how you will need to think and train your mind around that, and how you will want to do that analysis. Okay, so the T lineage is very clear cut in this case, and among the blast cell, this is which tube number? This is tube number now 3. Okay, in this as well, I see some signal in P side 7, some signal in CD8, which is okay, APC at 7. CD4 is clearly negative. And these are the blasts here. So I can see there is some population of, so there is a possibility that we are actually sitting there. Now let's remove all of this. Do you think that there might be 2 different blast populations? Maybe there is this as one population and this is another population. Correct? And if both of them are clearly, if I put this as, although I am naming it in mono, but don't think about it mono as of now, and I think about this one and call it as one of the blast population, and we have these 2 populations now. And now we look at this. First of all, we want to make sure are both of them blast, are both of them showing CD34 -. Yes, both of them are showing 34 -. We see that. Correct? So what do we see? That in the blue population, it is almost entirely 34 and 117 positive. Whereas that orange population that we were seeing is only a small population with 117. Correct? And similarly, if we go back here now and we review that, maybe because everything was in 34, we were not able to pick it, and maybe now, even in this case, now this is still showing a kind of one population. So in this here, MPO is clearly negative in the entire thing. The MPO is still clearly negative in both the populations, whatever they are. But very clearly here you see there are two different kind of clones. The blue one is the one which is predominantly more myeloid or monocytic one, and whereas what you see is the orange one is clearly more kind of a peeling it. And if you see, yeah, so here is wrong with the symbol as well. If I add to this the lymphocyte control now, we need the lymphocyte. Let's add the lymphocytes here for a reference. Let's see here. Okay, when I look at the lymphocyte reference, this population is showing up 19, this is showing up 8. I certainly would have trouble with this. I would have trouble. I would still want to go back and see whether there is some biology in this kind of center. It's unlikely that something like this will show for whatever reason. So just leave it like that right now. If something is off, what are the qualities? The same populations are showing, looking at multiple populations, 13 also, 14 also, 8 also, something is easy about it. Something is easy, something is not right. You might just ask for a sample. And the funny part here is this plot, let's see something funny from the time plot here. Okay, singlets, still quite neat and clean. It's comprising of 88% population. Okay. Slightly odd thing was scattered one bit odd compared to the cytoplasm cube. This just shows a very low forward scatter and slightly high side scatter. It's a very difficult compromise biomutant pattern. But I have no explanation to explain why that biomutation is there only because of protein, but not in the okay. I leave it at that. This is not, you can go and deep dive and this is a lab, you can see what could have happened to the sample. Something getting or around that. So this is where having an analysis strategy and a data analysis strategy, it includes all this information, gives you an opportunity to troubleshoot much faster. I have seen situations where many times we are able to simply looking at the data that we can, oh, it is different with every sample that is different. Okay. Now we have two options now. One is I can show if you would like to see how we create this kind of template from scratch. You can do that. The other option is you can look at similar some more cases, or you can try to now create something that is on your own. Familiar with the practice software. You can try to do it on your own and you can just address. We have we have Masanam, Adi, Abhita, there and there. You can come to your address. What would you like to do? Or you want to just see more cases? Right? Yes. So I think you should practice some more. What would you like? More cases? More cases like this one. Okay, so now, so we have, this is a different case now. Now in this case, we now solve it as again save as and just save experiment without data and call it template. Okay, save experiment without data. And in that so-called template, I open it and I import more FCS files. Let's look at some other case. Open. Here we go. Right? You have all the data again. So we can go back. Okay, so we need to fix the time block. So that means this data has taken longer to acquire and include it completely end to end. The tape looks homogeneous, looks good. Let's look at this. Yeah, singlets are about 95%. As long as they're somewhere around 90%, I know sample is not particularly long. This is okay sample. Okay, you can see here around. Yeah.
I had a question earlier today where it was about that. I need to bring it back in.
So you're saying that it will take completely flat. So see, if you want it, it's at the time when you're acquiring your data, if your area scaling is correct. Area scaling is correct, then it will be completely done. If the area scaling is not appropriate, then you will see it is slightly shifted compared to the 45-degree angle. That's what will happen. So it does not fill your data. Even if it is a little bit shifted, it doesn't fill your data. What we are still so CSNP does that, but you can sometimes improve on the CSNP as well if required. CSNP should be able to take care of that. It will take care of only for that standardized IBD assay, which is running in a plate color format. So, yeah, but sometimes we do see it's not a neat clean for IBD. It will be tilted slightly one way, and then we are able to do it. So now in this plot, are you happy? Are you happy with this? Now, if you are looking at this and you're having struggle where to draw the region, this is an arbitrary cutoff. If you are having trouble around that, one good way is, okay, anyway, I will need to bring it down a little bit, and then you go into the sites that are 45 plot, and here, what you want to see is that, in this plot, by excluding these, so you rely on to make sure that whatever is your lymphocyte are the smallest there, that you are not cutting into the lymphocyte population. That is how you can decide on where to draw the boundary on the forward scatter side scatter. So you draw a region on the lymphocytes neatly, and then see where these lymphocytes are lying in the forward to the side scatter. If you have been able to see the bottom of the lymphocyte, then you know the cutoff is okay. That's how you define the lower limit of analysis threshold you are putting on the forward scatter to ensure all lymphocytes have been included. If you are not seeing a clean boundary. Now, in the side scatter 45 plot, what do I see? Okay, lymphs are nice. There are very little monocytes anyway. I can see there is possibly a big chunk of blast. There is some neutrophils. I can just leave it like this. Doesn't really matter. Okay, and that blast in this case as well, fortunately, we do have a big chunk of 34 positive population. And we now see where we want to draw this. Now see, there is this heterogeneity here. Now, if you are having trouble around this, you could make this density plot. Let me see, I go here and make this density plot. Okay. It is telling you where possibly the boundary here could be. If you do not like density, you want a contour, you can go and make a contour plot. And within the contour, you can have, let's say, a contour line color there, and now you see where the boundary is going to be. It becomes just visually much easier. There are various options you can play with it. You can also have a dot contour hybrid around that. Sometimes this thing looks quite pretty, and you can see, now you see this is where the valley is, and you may then want to come into this valley and drop your region there. So these are just some tools, which is, some people by default like to keep the contour and you can do that. I think that is depending on how it goes, how when you're doing your flow, it's a comfort level account. So within this 34, now let's look at the markers. What do you see here? Oh my God, look at it, even your lymphocytes are having very the lymphocytes have no business to be an MPL are still brighter. Something is really, really wrong. Go back and check your thing. Go back and check your body. As the 60 voltage is gone, from here, 5 voltage, 50 voltage. 34 is clearly positive, 19 is negative, internal positive control. What do you think about CD7? It's right in the middle of the population. Some of you may call it A positive, but I don't care. About that, even if you call it CD7 positive, it's not changing your diagnosis, it's not changing your okay, but everyone call it negative. Now, cytoplasmic, this one would be tricky. This one would be indeed very tricky compared to your internal control. It's seen as positive. Okay, it's very similar that population is positive. Neutrophils, we do not have a high background, but this will be putting you in real type C. Real type C. Okay, so if you want to call it NPAT, will you call it NPAT? No, I will not call it NPAT. Okay, it's a very the cytoplasmic tree is not as bright as a normal T cell. Plus, I generally hesitate to call anything specific for tree if I do not see a nice CD7. This CD7 is low. If this CD7 was sitting somewhere here, I would be more inclined to give respect to the cytoplasmic tree to know. But without the CD7, it's very dangerous to it's not super bright. I don't very worry about it. Maybe it's a tabulating cytoplasmic tree or whatever that is. But it clearly tells that yes.
I was just going to say, if you look at the neutrophil CD3 background, lining up with that, so that it's sort of another internal control we could possibly compare it to.
Right about that, but we have seen in this panel, in this training protocol, neutrophils always really high that.
Yeah.
So, and then, so it does become difficult, you're right. That is another internal reference. If you reference on the neutrophil, the blasts are not more positive than that. Refer around that. But it generally points out to the fact that you need to overall maybe improve your training protocol to get rid of the fact that training protocol is not fully optimal. And as we know the fact that we did with a certain buffer, in the wash buffer, we are simply using cheap fluid, we do not have DSA and DFC. In some other settings, when we added DSA, we reduced the background there in the water. So, there is a need to optimize the training protocol in this case. Going to the subsequent tubes here, what we look at here now, of course, this is surface tube, so you will need to adjust your gates again. So if everything is a little messed up, just take everything out. This is, you put your lymphocytes here. The blast will be handled by your anyway, clearly the plot in the CD34 because it is there, positive. So I'll just use the blast coming from here right now. And I can see it's clearly 13 positive. They sum to 16 maybe. 117 is strong positive, 33 is strong positive. Epinediar is there. The strong epinediar, but 14 is negative on this population. And we have all of these internal negative lymphocytes. So there is no doubt about the myeloid lineage here. We clearly have 33, 13, 117, and NQ which is positive. Although the part which cannot be explained is why your lymphocytes are having very high value. That cannot be explained here. Okay, but but I would still consider if you look at here, this is the internal control, and this is the 34 positive cells that clearly have an NPO, which is clearly half log rather than what you see in the normal lymphocyte. It's an NPO positive. So more classical AML, but these are real cases. These cases have not been picked to show only what it is all bad ugly population. This is a real world you see. So you find your population in that ugly world of what it will come there. And right. So if you look at here in the population, because you do not have 34, so you will need to go back to the regions here. Lymphocytes are appropriately drawn, and so you move here, and okay, now let's see. Let's get this one. Okay, so we have captured most of the blast. What are blasts showing? They're showing a pattern on CD15. There's some bit of CD15 there. Maybe a little bit of CD4, which is not unusual for some AML. That can show it for CD4 here. And I think that's about it. So you have clear-cut acute myeloid leukemia. And let's look at some of the percentages and what do they tell us for the difference. Now let's look at the lymphocyte percentage across different populations. Let's look at the lymphocyte percentage. A lot had a lymphocyte of 6%. The Q2 had around 5.6% here, and the Q3 had again around 5.6%. That's it. Across the three groups consistently, there is no differential in the, some cells being lost in cytoplasmic staining or purpose. It's looking quite okay around that. Now, now let's look at the data around. As I say, the controls that we are looking at, date, okay, everything on the same date, time of beginning, okay, time like end of admission here, then voltages are same. So generally, I right now shown you two voltages. I try to keep in my template voltages for all the time. 8 colors and 2 for 1 for 7 cycle. It tells me quickly whether something is wrong with it. And when it is compared across the different days, different months, whether there is something huge drastic change. Okay. So this was the template part. Now we can so I should have saved that once. I can see it as the signal is 9. And I can go back to the template now again. And in the template, we can import. See, import now. Whether it's the basic or the add the few, they're added a few. Because this was not an template, they're added a few. So, I think it's better to have that template in the case, mine will just call it as save as without data, then call it data. Okay. And once that is saved, we go to save. And this is now empty template. And in this we will import FCS files. Save. Here you are. Okay. Go up. Check here. This looks okay. Singlets look fine. About 9, they are 98%. Cells. There is some dead cell there. You can see this is the population that you see. You can see it better than it is. This is the dead cell population. And then when you look here, okay, this is fine. And we see here, this is the glass, that's some small neutrophils. And the normal lymphocytes in green. And again, I look at the glass here, maybe now this glass is going to be corrected. I bring it here. Okay, so we are very much covered. What do you see here? Oh, 0 NPO. But how do you know NPO is work or not? This is where your internal staining in a small amount of neutrophils and that neutrophils are showing NPO. But why are they appearing CD39 18 positive? Because the very bright signal here, very likely when you ran your comp controls, it was not as bright as this, and therefore it is undercompensated. Not very often. But here you can see, compared to your, this is your normal T cells, these are your T cells, and 79A is a strong 79A because the blast 79A is going even closer to the normal T cell expression. 19 is clearly positive, so is 34, 7 is negative. Okay, this again is, I would imagine this is something we are saying, this is a kind of a compensation issue. The NK cell, this is a diagonal. Whenever you see a diagonal, this is either a viability issue or a compensation issue. In this case, this is very likely to be a compensation issue. Okay, and then, so this is your blasts are negative. And this, I would ignore this population and I would figure out where it is coming from. Maybe it is coming from some dead cells there. And you know, if you try to, the possibility there could be some dead cells that are gone, some non-viable cells that are showing it to be. But otherwise, you know, it's a 1934-79A. Go to the subsequent tube here. In this tube, again, I can change this here, or because I do know for a fact that in my tube number 3, there is no 34, I just rely on this glass gate. I just keep it here and here, because right now this is gated on 34. So I change this gate to display population to glass instead of 34. Okay. So 64 and 13 negative. I need to get the grants in picture so that we have some idea of the internal positive controls. Okay. The positive controls, what is 64 and 13 negative? 34 positive, 117 negative. There is no internal positive control for 117 here. 33 is internal positive control. Maybe you can call it 33. It won't hurt. You can call it 33. HLADR is strong positive. 34 is positive. Tube number 3, 19 is strong there. 5, 15 are negative. 4 is negative. 10 is strong positive. 8 is negative. 20 is positive. Correct? So, the image. Clearly. We look at our internal populations, normal lymphocyte in A log is 1.8, here 2.69, and here 2. Okay, pretty much the same. And again, date same for all of them. Voltages are again same for all of them. So consistent. Nobody has played or grouped up with the voltages. Nothing to explain there. Then time save as without data. Calling is 7. Okay. Experiment is 7. 5 import of your time. 7. Here, check the plots. This looks okay. Does this look okay? Yeah, percentage is 92%. Pretty much okay with this. From here, this is interesting. See, now where would we want to draw the line here? Very hard to say. So either again you make this also into some kind of a density plot and see if it is giving you some indication. You put contour, and within the contour we will here. So let's see what it shows up. Not really helpful. Okay. Let us just take a leap of faith anyway, and let's go there and see what the classify is. So get your lymphocytes clean. Let's see all dark. We are conducting my white cell. Here, can do up here to get your particular target cells. So there is some bit of NPO. It is coming from blood or coming from lymphocyte. This all the lymphocyte. Suppose I take just this population. Okay, it reduces little bit. So NPO is there, 34 is there. There is now. What about this CD7? Is this a spread issue? Is it a spread issue? Or is it genuine CD7? How do you decide that? You have to see what all can spill into APC channel. And if that is strong positive for this population, there can be APC at 7 which can spill into APC by time down. But this is the green population, APC at 7, which is bright positive. The orange population is anyway APC at 7, negative to 3. So that cannot contribute to a strong APC signal here. What else can still into APC? A little bit of persistent Cy5.5 can still into. What do we have in Cy5.5? 34, super bright. Now, let's look at this. If we keep 34 here and we keep CD7 here, does it give you, what kind of, does it give you a data which is like a proper that spread? No, it's not the twin-tail spread. The major cluster is here. Now, if you here go and make this plot as a again contour or the say density. The density is not here and where it is given. This is a genuine CD7 expression that we are seeing. So this is a genuine CD7. The CD7 is definitely expressed in this way. Cytoplasmic 3 and 3 are negative, and our internal controls are by and large perfect here. NPO is super bright. So maybe this is a question to look at. Now, if consistently your NPO coming is bright and going all the way up, you may want to review when was the last time you optimized your setting. As per the protocol. Maybe you realize that it's done 4 months ago or service, visit has happened, a PM has happened, and never been into it. Now, this is where if you are not sure, go back and run a normal blood sample. And showing much higher NPO like this. Whereas when you initially set up the thing, it was not like that. I have a normal blood data, maybe I'll show you that as well, how the NPO should look like. Then you know something is wrong in a second. And that's how you will be able to troubleshoot and look into that. In this case, even if it is super bright, it is not killing your analysis. Because you know your NPO is, whatever the NPO is there, the genuine NPO that you're seeing. But this kind of gives you indication of what could be going wrong in your overall thing. This is too high in NPO. It's too high. But here, of course, compared to the internal control, this was nicely positive. Then we look at the tube number 2 here. In the tube number 2, we have 34. But as I said earlier, because tube 3 and tube 4, we want to kind of look similar. We can add some glass here. So nicely, I can see as I'm adding here, 117 is coming at a strong cluster. It's myeloid. 33 may be DIN positive, but it's there. Little bit 13, I may just call it negative in this case, or we can call it DIN positive. It's okay. 64 is negative. Grams are positive. 64 as they should be. 14. What about 14? So let's see if we showing this. We try to find some population is somewhat in the mono area. Okay, so my DRN 14 is there, and my 32 is there. So I know my reagent has worked. It is not that the reagent has not been handled. The reagent has worked nicely monocyte, but yes, my population of interest is 14 negative, and it is HLA-DR negative. Correct? So overall, it is adding credence only more and more to AIM. Why AIM? NPO is there, 117 is there. 43 is there and some 13 is there. But clearly, of course, what is clearly present is an aberrant CD7. Now let's look at some other important markers that we have here. So there is some bit of 15, which is there. 4 I am going to call it negative. 28, all that are negative. That's how we will expect this case to be. Okay, we go back and look at our controls. Lymphocyte population. What about lymphocytes across different tubes? 13%, 19%. So we can go back and check whether there is some drawing the region. And because if you remember why it is, because if you remember in this, I had removed the lymph only to this half of it. Okay. Once I bring it back here, now see, this is also around 19%. So, these are some good checks. If you include these things, I forgot about that I removed the region. But when I looked at my stats, it gave me a quick indication, something is wrong. Go back and check it out. What's happening? And similarly, you can look at the PNG voltages. 316, 471. You see, it's kind of consistent across the different people. Your data has been acquired over the same data. Okay, I think we'll do one more case and then we'll go with. Sorry, so I need to see that. Very bright 45 overall. Okay. Now let's look at this. Is this appropriate? No, this is not appropriate. We will need to change that. Okay, this is just showing 65% and we know very and now here the last. Okay, so here this is clearly heavy. Yeah. Wow. Nice. Any potential diagnosis like on the side of the body? Should we? No. Correct. And now this is the case where 24 is negative. And you know monos are often 24 negative. Monos are often 24 negative. And so of course you cannot rely on this not one meaning. Okay, so now this was created on 34. I need to bring this back to glass. Properties, glass, 34. Neat case. See, very nice case. MPO clean, not going to the axis. Lymphocyte control negative. Glass are clearly MPO positive. 34 is negative, 19 is negative. Neat and clean case. Look at the cytoplasmic 3. Even in this case, neutrophils are here, monos are here, and mass are here. You can see that. We will not call it cytoplasmic 3 positive, although it is higher than the B cells that you see here. Then you look at the tube number 2. Here as well, you will need to now change the regions. This is the gland. You know the diversity in the egg here. This is the lymphocyte. And for the glass. Okay. So this is the way you just pop it there and it adjusts to the population. Snap to great kind of thing. So anyway, see to that. Just to see there are any inside. Oh wow, beautiful bright 64. Bright 64, heterogeneous 13. 117 is by and large negative here. 33 is strong positive. So, and 14 is heterogeneous as well. HLA-DR is positive. And along with 64, if you put here CD14, again you can see here. So all fitting in the mono pattern, bright 33, bright 64, heterogeneous 14, heterogeneous 13. Okay, image. Any other co-expressions? No more co-expressions so far. Okay, and look at CD4. We do know the normal monocytes are CD4, and this is possibly just the same population. And yeah, so clearly the monocytes are CD4 and the bars are CD4 as well. That's the only additional information we're getting from tube number 2. But the diagnosis was done But the diagnosis of AML was done, if you ask me, just on the field number 1 itself. Field number 1 with clearly negative markers for specific P and specific B, with NPO clearly positive, in the name. The tube number 2 added the value that this AML is very likely to be mono, which anyway the sites that are qualified for filing was also highly suggestive of the mono pattern. The tube number 3 did not add any diagnostically contributing information. In this tube, actually should have run only tube 1 and 2, that would clearly tell you the AML will negative frequency. Correct? Nothing else stop now. And we'll see if there is any preference around this or anything specific feature you would like to see of how that is done and what is done, then we can look into that. And any other common laws, anyone laws.
How about if we have two populations based on CD45, clean and positive CD45?
Yeah.
Should we analyze in both or only in clean area?
So I would analyze them as two separate populations first and see if they are showing a distinct immunophenotype profile. If they are showing the same profile exactly, I will just draw one big region around it. But I will start with two different regions around. If you can see clearly two different clusters in life that are qualified, I will draw two different points.
Definitely the CD45 negative has no expression, especially in our.
Oh, if it has no expression, it's very likely to be a electrolyte cell. It could be a electrolyte cell. With the bone marrow, it could be a electrolyte cell. Then you don't worry about that.
Okay.
Then you don't worry about that. Then that's not a combination you will include in your analysis.
And then it will make a difference with the morphology. Because in morphology, the number of flash in the middle or in peripheral flash, like the flash only 70%. But if we look for in the CD45 field, it's only 20%, for example.
Yeah, see, in bone marrow, I do not rely on the blast percentage coming from the flow. But the bone marrow can be hemodilute, there are multiple things which can happen. There could be NRBC, unlike RBC, many things can happen. In blood, it is still reliable, it is a paper blood sample, so there is nothing for hemodilution to happen there. But in blood as such, these are unlike RBC that can distort your blast percentage, even in the paper blood sample. So percentages of blast is ideally come from your morphology. If nearly more than 20% blast in bone marrow, it is okay. You know what you are dealing with leukemia. But if in the bone marrow sample or blood sample, you are seeing 14% clear-cut blast, 34 positive, 14, 45, it could still be an acute leukemia for percentages. Absolutely. Okay. Good. Yeah, I think it's going to be. Yeah.
Thank you very much, Dr. Pares. Is there any question more from participants? So, because of time limitation,
in case you want to practice it more, we are still around. When you want to, the cases are all there in your laptop. You want to practice for more than half an hour, we are around. You can take it. Okay, thank you.
Thank you very much. So this is the end of our parallel workshop session, which also ends our whole event for today. As a master of ceremony today, I would like to say thank you to our honorable speaker today, and also would like to say thank you to all participants for taking time to join us here today. We look forward to seeing you on next day. Good afternoon. Om Santi Santi Om. Assalamualaikum warahmatullahi wabarakatuh. Shalom, Namo Buddhaya, salam kebajikan.
Yang izin mengingatkan kepada para peserta apabila sudah menyelesaikan menggunakan laptop dapat dikembalikan ke bagian belakang dan saya ingin mengingatkan kembali bagi para peserta yang merasa kehilangan bros dapat diambil meja MC. Terima kasih.